Thanks, Giuseppe, for introducing the housekeeping rule. And let's start this session on vectors with the first presenter, that will be Enrique Quesada Moraga, with a presentation on illustrating strategic approaches for entomopathogenic fungi-based Xylella fastidiosa vector control. Floor is yours. You have 10 minutes.
Thank you so much. Thank you to the organizers for selecting this talk, and thank you also to all the people here in the room. Well, this talk is just a view of all the work we have performed in the frame of the SEAL project regarding the use of entomopathogenic fungi for the management of the Xylella fastidiosa complex problem. Why entomopathogenic fungi? There are three key points that make entomopathogenic fungi an excellent tool for trying to go against this problem. The first one is the mode of action. The second one is the natural presence and distribution of these fungi, even in the olive crop agroecosystem. And the third one is the role of entomopathogenic fungi that we will see in the next slides. Regarding the mode of action, entomopathogenic fungi are unique among entomopathogens because they act by contact. So it's the only group of microbial pathogens acting by contact. At adhesion of the conidium, there is a process in which the infective propagules cross the barrier, the integument, and finally they develop the infection process within the host, and finally even they use the cadaver of the host to outgrow, as you see here, just in this Philenus spumarius infected by Metorrhizium brunneum. Secondly, as said before, they are part of the microbiota of the forestry and agroecosystem, and particularly we have demonstrated in our previous work that entomopathogenic fungi are present almost in 80% of the soils of the olive crop agroecosystem in the Mediterranean area, but not only in the soil, also in the cover crops, in the plant, in the olive tree, mainly in the leaves, and in many pests of olive crop. So they are part of the olive crop agroecosystem. Indeed, we have tried to use different strains against Xylella fastidiosa vector obtained from the olive crop agroecosystem. This is a real ecosystem services. And finally, the role of entomopathogenic ascomycete, because during the last century, they have been demonstrated not only to be entomopathogens, but also to have relationship with the plant at epiphytes and, very important, as rhizosphere competent and endophytes. So this is, on the overall, why we have different strategies for trying to manage the problem of the Xylella fastidiosa. The first one is using entomopathogenic fungi by direct sprays over the specimens. This will be a typical mycoinsecticide against the insect vector. But in this case, we should research, we should investigate 2 points. First one is the direct mortality on the vector population, and secondly, whether there is a change or modification of the feeding behavior that could interfere with the transmission of the bacteria. But regarding the role, the other role, we could have also an effect via plant, through the plant, because as they are endophytes, we could manage the covers to have them endophytically colonized by different strains, and to have also an effect in the vector, again, directly by reducing the population, or indirectly by trying to modify the feeding behavior and to alter the transmission of the bacteria. And in the frame of the BASIL project, we have investigated all these strategies. First of all, regarding the direct application of the fungal inoculum against the nymph population, we have evaluated different strains, either in Spain, in Italy, in Brazil. In the case of Spain, we have used only strains coming from the olive crop agroecosystem. against nymphs and adults with very, very good result in terms of mortality. And we have even performed field experiments to try to reduce the nymph population and therefore the adult population at field conditions with very good results in Córdoba. Experiments again, you see the evolution of the population of the controls and on the treated insect and the relative variation in the density, the Pebbling formula, that means that there are very, very good results, not only against nymphs, but also towards the adult population. This is the direct effect. It means using the fungus like a myco-insecticide. But in this case, we have recently demonstrated that when there is an infection process, we have also a modification of the feeding behavior. You could go to the pitch talk poster number 88 by Tamara Hernandez, that you will see what important modification of the feeding behavior of the adults has been detected when they are infected by entomopathogenic fungi. Regarding the other component, I mean the use of endophytic strain of entomopathogenic fungi, again we can have a direct mortality towards the adult and nymph population where they feed on endophytically colonized plants. In this case, the target had not had contact with the conidia after a spray, but by feeding on infected or colonized tissues of different cover crops with the conidia, you see here with very important mortality rates in different important plant species of the natural cover crops in Spain. And even we are developing in our group a strategy for controlling, this is quite important, a strategy for controlling the olive fly, the key pest of the olive crop, is also very important. There are soil treatments beneath the tree canopy. Is very effective either in autumn and in springtime to reduce the Auchenorhyncha populations in the covers. This is also a quite important issue to try to reduce the population of the vector. Also, we can have a modification of the feeding behavior through the plant, as it has been investigated in collaboration with other groups of the Xylella consortium. We have not only mortality, even 50% mortality rate of adult feeding, even on non-distant leaves and on treated leaves. And we have a modification, as you see here, of the feeding behavior of the vectors. And the key question that the Basil project is still ongoing is whether this modification in the feeding behavior has an impact on the transmission of the bacterium. In experiment performed by Natalia Gonzalez here in the region, in Bari, we have two repetitions with different results. In the first one, we have a significant reduction of the transmission of the bacteria. In the second one, we have not. And even, and this is the last studies during the development of the project, we can have an effect by the olive crop. We have demonstrated that the olive crop in the studies of Clara Lago can be endophytically colonized by our strains with percentage, quite important percentage of colonization in targeted leaves, 65%, and even in distant leaves, that means systemic colonization by the fungus. And we have very good, we have also important events regarding the feeding behavior of the adult vector. As you see here, improving, reducing xylem contact and interruption, and delaying xylem ingestion. Whether this has an impact on the transmission remains to be studied. Therefore, if we have a view of the general context of the life cycle of this of the vector, in this case Philenus, we could use these strategic options against the nymphs in the cover. Very important in this stage. I mean, before adults go to the plant, but even using the olive crop. And for the take-home message, and this does not occur with insecticide. We have a cumulative effect. We have hidden mortality of the population, mortality via plant, alteration of the feeding behavior, and remains to be clearly investigated whether there is a reduction of the transmission of the bacteria. I would like to give my deepest gratitude to all people of the work package 5 of the Xylella of the Basil project, and thanks a lot for giving me the opportunity to be here.
Thank you. We go directly to the next speaker, Daniele Cornara from the University of Bari, with presentation on unraveling the interaction between Xylella fastidiosa and its insect vectors. Over to you, Daniele.
Thanks, Evelina, and thanks to the organizer. And let me start my presentation with some reading recommendation, especially for people that are approaching Xylella for the first time and young researcher. These are 2 reviews that are must-reads. The first is from Sandy Purcell, and there's Paradigm of Xylella, and it's a description of the fascinating story of scientific discovery around Xylella. And the second is a review by Chatterjee, 2008, and there's Living into Words, that express a fundamental concept, at least for me, that Xylella is not just a plant pathogenic bacterium, but is actually an organism, a bacterium, living in 2 very different and very distant worlds, 2 kingdoms, the plant and the insect. The insect is actually not just a flying syringe, is a host, and the success of Xylella is actually depending on how well the bacterium is adapted to the host, and how even plant pathogens that are spread by insect vectors are able to manipulate the physiology and the behavior of their vector. The better they manipulate the physiology and the behavior of the vector, the higher the chances for them to spread and to establish in an environment. And this kind of manipulative effect are kind of like being described specifically for persistent plant pathogens, but Xylella is indeed a persistent pathogen, but Xylella is also unique in being non-circulative. So actually, Xylella in the vector colonized the cuticle lining the foregut, so the anterior portion of ectodermic origin, and never interacts with internal tissue of the vector. So we have a persistent pathogen, but it's a non-circulative. So we have a big question around this. Is there for Xylella the chance of a possible manipulative effect? So what is the interaction between Xylella and the vector? It's kind of a simple question, but trust me, it's very complicated. So we have to undertake a very long journey of 10 years, and this is the first time that I present publicly the results, about if and how Xylella manipulate the vector behavior. And we try to tackle this from multiple sides, exploring different kind of effects. The first question was, is Xylella able to manipulate the vector probing and feeding behavior? And we investigated this through a technology which is called electrical penetration graph. Then we investigated possible changes in the metabolic rate in case of presence or not of Xylella in the foregut. Then we investigated the energy storage, so we quantified the lipid levels. And finally, last but not least, we evaluate the gene expression between bacterium positive and bacterium negative vector. And I don't have time to go in detail, so you should kind of trust me. First thing, Xylella indeed alter the feeding behavior of the vector. Actually, when an insect vector has Xylella, it struggles a lot in initiating feeding. It causes a problem in powering the big cibarial muscle in the head of the vector that permits actually the xylem sap uptake. So the number of feeding events is reduced, but at the same time, when they are able to initiate a feeding event, these feeding events last longer. And if you see the graph there, negative, positive insects, that graph on your right, on top, It's a graph comparing bacterium negative and bacterium positive insects and the frequency of the contraction of the cibarial pump, this big muscle in the head that permit feeding. So in negative insects, the frequency goes up and down and it's very dynamic throughout the feeding process, while the red line, the positive insect, the trend is similar, but if you see, the frequency is more constant throughout time, as it is not dynamic as it happens in negative insects. Second aspect, energy storage and metabolic rate. Actually, you see on the left, there is in bacterium positive insect a drop in the content of triacylglycerol, which is fundamental for energy storage. So it's a lipid that is fundamental for energy storage in bacterium positive individual. And actually there is also, particularly in female, like an alteration of the metabolic rate. So it's like the presence of Xylella sort of accelerate the metabolism, making like the life and the normal functioning of the vector more energy demanding. And then finally, what surprised us that the gene expression experiment was very consistent with all the results that we got in the lab. We observed significant differences in the gene expression in bacterium positive and bacterium negative. We had like differences between the two, infective and non-infective, in muscle functioning, immune system, lipid storage, and even chemosensory protein. Basically, we observed in bacterium positive individual an overexpression in the head of this myosin light chain 1, and myosin is an essential protein for the contraction of muscle. So actually, insect with Xylella inside has this overexpression because of probably is like pumping this cibarial muscle is more energy requiring. Another thing that was like a striking difference for us is this chitin deacetylase. Actually, Xylella is a chitinase through which can exploit the cuticle lining the foregut of vector as a carbon source. So to grow, actually, they can degrade the insect exoskeleton from inside and use these nutrients. And the overexpression of this chitin deacetylase just means that there is a cuticle remodeling in bacterium positive vector because of they are probably responding to this bacterium chitinase. Regarding the immune response, what we observe is that actually the vector at the beginning recognized the presence of Xylella inside, but actually there is no activation of a strong immune defense, immune response against the bacterium. Our hypothesis here is that symbionts for insects are essential for their life. Without symbionts, they would die. So in case of activation of this immune response against the bacterium, this would be detrimental for the vector itself because it would kill the symbionts. Regarding lipids and chemosensory protein, actually the trend is sort of a shift from a normal behavior, like exploratory behavior or normal life of an insect, to sort of a sick mode, survival mode, where all the basic physiological functions which are not essential for living are sort of suppressed. You see that there is a downregulation of a vitellogenin-related gene, and vitellogenin is essential for the egg production, so reproductive function. And what is also like very intriguing for us is this downregulation, the last one on the right bottom, of this odorant binding protein, which is a chemosensory protein. Chemosensory protein in insects are associated not just to volatile, but to perception of whatever chemical cues. So are essential for the perception of the chemical cues surrounding the vector. So for the vector to recognize the environment, for the vector to recognize plant and how good plants are or how bad plants are. So very long journey, 10 years to a possible answer to our question. Actually, yes, Xylella makes the vector sick. It's a simplification, I know, but makes the vector sick. So actually what we notice is that there is an exploitation by Xylella of the vector to which the vector responds with this sort of host compensatory response. The vector is sick and the presence of Xylella like change the physiology of the vector itself. So it seems after 10 years we have a cool answer to our question, but actually it's not. This was a long journey, but actually the answer to our question implied that we have to undertake an even more long, probably unexpected journey because of all these effects were measured over a short time and lab condition. But what this could mean that Xylella make the vector sick is like that Xylella can even change the population dynamic of the vector, can change the way that the vector move, perceive plant, transmit the bacterium, and even their susceptibility to stressors, from environmental stressor to pesticide to Enrique fungi. So it will be an unexpected journey, but for now, just let me thank the wonderful people with which I undertook the first journey. So my group at the university and the co-leader of my group from EPSP CNR, Michela Chiumenti, and then all the labs that collaborated to this, Rodrigo Almeida and Berkeley, Alberto at CSIC in Madrid, Cristina Marzaki from CNR in Turin, all the project that funded this research, and obviously my thank goes to you and for your patience, and question will be for later and for coffee time. And now we go to Cecilia Parise from University of Torino that will present her work on RNA interference for the control of Phyllones spumarius.
Thank you very much. Can you listen? Yes. Okay. Today we will draw some light and shadow on the application of RNA interference to control Phyllones spumarius. So let's start. Just a bit of state of art. What is RNA interference? RNA interference is a post-transcriptional gene silencing mechanism which is triggered by a double-strand RNA molecule, and it regulates the expression of the corresponding transcripts. This mechanism is widely conserved among eukaryotes, and it plays an important role in defense against virus and transposons. Okay. I'm sorry. How does RNAi work in insects? So normally it is achieved by delivering double-strand RNA molecules that are about 100 to 400 base pair through different delivery methods that can be cell transfection, artificial feeding, microinjection, and plant-mediated delivery. RNAi in insect was widely explored, first as a functional genomic tool, then as a pest control tool, since that the application of exogenous dsRNA molecules could be a valid alternative to chemically pesticides. Why? It has some advantages, like it's highly specific to the target organism because these dsRNA molecules are designed on the gene of our target. The molecules, the dsRNA molecules, rapid degradase in the environment, low incidence of resistance have been reported, and normally these molecules are also easy to project and design. Has disadvantages, there are some variability in the RNAi efficiency among the target organism in between order and also in between species in the same order. There are also some efforts have to be made in the selection of the target genes. There are some gaps in risk assessment and legislation. And when we talk about subfeeders, there are also some field scale, some problem in the application, in the field scale application of the dsRNA delivery. So, the aim of our study was to characterize RNA in Phyllones spumarius. So we wanted to characterize, to describe the presence of a core RNA machinery, and that delivering a dsRNA was triggering a silencing response. We want also to identify some target gene for RNA-based control, and we wanted to test different dsRNA delivery methods, since that when we talk about subfeeders, this is kind of a problem. Let's go to some results. This is a result from our first experiment where we wanted to assess if this technique can be applied in this insect. So we have tested through microinjection in adults, and we have seen that the target gene downregulation was reported even for the adults and in nymphs. In the lower graphs, you can see the gene expression in nymphs, where the delivery method for nymphs was not microinjection, but was plant-mediated delivery. So we gave to the shoot the dsRNA molecules, and the nymphs was feeding on the treated shoots, was able to acquire, and the target transcript was silenced. The gel is just that the proof that, these dsRNA molecules was present in the plant material that we have used. Okay. Then we wanted to explore like 6 different target genes. We have tested 6 just through microinjection, just on adults. And in these 6 genes that we have tested, 2 of them, one dsATPase and the other one a TRPML, dsRNA, have been chosen for a transmission assay involving Xylella that we have developed with the group of Daniele and all our colleagues. And our insect was first microinjected with the dsRNA molecule targeting ATPase. Then our target organism went through Xylella acquisition, and then to 2 phases of inoculation. Here, I reported the more interesting result that we have found. That was that in positive insect treated with dsATPase, the bacterial load was significantly lower compared to the control. This was a result that we are proud of, and we are happy with that in that experimental setting. And this one is one experiment where we decided to assess if dsATP, this was targeting ATP synthase beta, was also giving some sterility effect in Philenus spumarius. In other Hemiptera have been reported that silencing ATP synthase beta gave some sterility. We wanted to see if the same was happening also in Philenus. So we have checked that in 2 years, and we have seen that the number of laid eggs in the dsATP-treated insects was about 40, wherein the control was 400. So we can say that kind of a sterility effect was present. At the molecular level, you can see that at 35 days post-injection, there was downregulation of vitellogenin that, as was explained before, is mainly involved in the oviposition and in the oviposition process of Philenus. So, some key results that we can say from our experiment are that Philenus spumarius has the key RNA core machinery genes, and that is capable of processing these dsRNA molecules, either in adults and in nymphs, as we can see seen in our first experiment. Among the tested genes, we have selected 2, ATPase that was giving mortality in our target, and ATP synthase beta that was giving sterility in female of Philenus. We have also noticed that silencing ATPase followed by Xylella fastidiosa acquisition resulted in a significant lower bacterial in our insect vector, and we also have seen that plant-mediated delivery of dsRNA is a promising technique for targeting sub-feeding insects. So, some final consideration. This induced mortality and sterility can contribute to reduce the vector abundance in an IPM frame, and also this plant-mediated method can be promising for these xylem feeders or sub-feeders, even if the field-scale application is still challenging and no answer are present for our insects. But the exogenous application of the sRNA is avoid the problem of GMO, and just the past month, I think that the use of a dsRNA-based product have been approved in Belgium to control Colorado potato beetles. I was very fast. Thank you to everyone for the attention. Thank you to EFSA for the organization. Thank you to all my colleagues from the EPSP Torino, all my colleagues from Bari, and all of you for the attention. Thank you very much.
Our next speaker is Crescenza Don Giovanni, researcher at the Agricultural Research and Experimental Center Basile Carmina, with a presentation titled Integrated Agronomic Management Targeting the Juvenile Stages of Philenus spumarius. Over to you, Enzo.
Good morning to everyone. My topic is the evaluation of alternative solution for the control of the juvenile stage of Philenus spumarius. Xylella fastidiosa is transmitted by feeding insect vector, and actually Philenus spumarius is the main vector in Europe, and they is ubiquitous, mobile, and at adult stage move on the olive canopy or the woody plants. They overwinter as eggs, and during the stage of nymph stay on winds, and then they move. So the best windows to target the vector is when they stay on the ground vegetation. Actually, the tool applied for the control of the juvenile stage of Philenus spumarius and the other spittle bug are prevalently soil tillage, followed by mulching. But mulching is less effectiveness than soil tillage. Then we can use also other method, physical method or chemical method, based for pyroweeding, also herbicides. Herbicide is very effective, but is technical not recommended because it is very impactful. And actually, for the control of the juvenile stage of Philenus spumarius, only one product in Italy is authorized. It is based on potassium of salt of fatty acids. So, in order to try an alternative solution for the control of juvenile stage of Philenus spumarius, we have carried out a lot of trial using natural substances based on pelargonic acid and maltodextrina. You have tested also entomopathogenic fungi, cover crop, and ground cover management in farmer condition. In the framework of BioVexo project, we have tested in 3 field trials pelargonic acid at 2 rates, and we have obtained good results, in particular with the highest rate. This activity were carried out in small field trial, and then for 3 consecutive years, we have tested the highest rate in a large-scale trial. Also, in this case, we have obtained an effectiveness about 50%. Then, in 2025, in 4 field trials, we have tested maltodextrin, this is a polysaccharide, and have a physical effect on juvenile stage of Philenus spumarius. We have tested this product at 2 rates, and in each case, we have obtained a good result with Abbott index ranges from 14 to 18%, and the product showed an effectiveness highest than the benchmarked product, in particular when it was used at the rate of 2 liters per hectare. These effects were observed against Neophylenus campestris and also against Philenus spumarius, with similar effect in different conditions of high and low density population. Also, we have tested different kind of cover crop based on crucifere, fabaceae, alone or in a mixture or facelia, this beneficial crop, used alone or followed by mulching, compared with plugging soil and with undisturbed ground vegetation alone and mulching. In different trial, in small field trial, and then all the large-scale field trial. In this condition, the best results were obtained using a mixture of different cover crop. The goal is to modify the structure of the soil, the biodiversity, and so to create an habitat unsuitable for the juvenile stage of Phyllenus spumarius. In all cases, plugging give the best result, followed by mulching and using of cover crop. Among the cover crop that give the highest result, the mixture, we have carried out a large-scale field trial using also during this year facelia, and also in this case we have obtained high effectiveness in all treatment that we have tested. For 2 consecutive years in 2 field trial, we have also testing entomopathogenic fungi, in particular Metarhizium anisoplia, that were selected by groups of Professor Bosco. They have tested in early stage of the peak of Phyllenus spumarius when they reach the 1st, 2nd stage, or at the late stage at the 3rd, 14th peak, and we have observed a good reduction of the density population, both Phyllenus spumarius and Phyllenus campestris, without a difference between the time of application. In general, the results were higher than the benchmark product based on Boveria bassiana. In a large-scale field trial, we have also compared plugging soil with mulching soil in 16 newly planted olive cultivar of Lecino and Favolosa for 3 consecutive years, and we have observed highest effectiveness of plugging, while mulching is less effective. In synthesis, we can conclude that plugging is the most effective tool for the control of a juvenile stage of Phyllenus spumarius, followed by mulching, but they are less effective. The use of alternative low-impact solution, such as pelargonic acid, maltodextrin, entomopathogenic fungi, and cover crop, show variable performance. Their efficacy appears to be highly depending on apricot on time, microclimatic condition, and vector population pressure. But this tool can be integrated in EPM management strategy in alternative to the soil tillage that is an impactant tool, or to integrate the effect of mulching weeds. Thank you for your attention. In particular, thank you my colleague for the big efforts for carrying out these fields.
Thanks, Enza. And now is the turn of David Makowski from University Paris-Saclay, with this presentation, direct control of insect vectors of Xylella fastidiosa, a systematic review and meta-analysis. And I would like to remark that David is going to present the results obtained by the EFSA expert working group on vector control.
Buongiorno. So thank you for having invited me to this very interesting conference and beautiful place. So my talk today is about the evaluation of a range of control measures against insect vectors of Xylella fastidiosa using systematic review and meta-analysis. No. Ah, yeah. Okay. Too fast. So this work rely on the selection and evaluation of trials comparing treatments against untreated control. So basically we collected trials including at least one untreated control group and one treated group. We use observation of survival rate of insect vector in these groups and calculated the percentage of reduction of surviving insects. Okay. Yeah. So the work proceeded in several steps. So we started with a systematic literature search. Then we selected the paper based on a range of selection criteria. We extracted data. We put the data in a common data set. We performed a statistical analysis, and finally we evaluated bias and uncertainty. So this Prisma diagram shows the flow of papers during the selection procedure. We started with more than 1,200 papers, and then we selected 77 papers for systematic review and meta-analysis. Most of the papers were discarded because they were out of scope, or because they did not include relevant data, or because they did not include rigorous untreated comparators. This figure shows a number of references for each vector species. So you see here that we found references for a large range of vectors. However, most of the references were found for a relatively small number of vectors, especially Phileneus spumarius and Ma narva fimbriolata. The blue points correspond to the number of references for use in the full data set, meaning for the systematic review, and the red points correspond to the number of references used specifically for the statistical analysis, for the meta-analysis. The slightly lower number of references for meta-analysis was due to the absence of data or missing data in several references. When doing a meta-analysis, we have to extract effect sizes, individual effect sizes, and their standard error. So here the effect sizes correspond to the percentage reduction of survival. So we extract data for each study in turn. So here the red points correspond to the individual effect sizes, and the red bar correspond to the individual confidence interval. We extract data for all trials, and after this we get a relatively large number of individual effect sizes and confidence intervals describing the efficacy of different types of control measure. Then we proceed with a statistical analysis of these effect sizes, and we estimate a mean effect size, here represented by the blue points. We also calculate the confidence interval of the estimated mean effect size using a random effect model and weighting the individual studies by their accuracy. We do this statistical analysis for 6 different categories of control measure: synthetic substances approved in the EU, synthetic substances non approved in the EU, non-synthetic substances, for example, plant-derived products, entomopathogenic fungi, entomopathogenic nematodes, and predators. For each category, we get a mean effect size, the confidence interval in blue here. And if the confidence interval does not include 0, we conclude that the category of control measure has a significant effect on the survival. If the confidence interval includes 0, we conclude that it has no significant effect. So this figure show some of the results. The labels on the left hand side of the graphic show the names of the control measure category. Between brackets, you have the number of data. The name at the top of the graphic corresponds to the name of the group of trials included in the analysis. So here in this figure, we show the result obtained using all trials. The red points here correspond to the individual effect sizes. The blue points correspond to the estimated mean survival reduction, and the blue bar corresponds to the 95% confidence interval. So here you can see that several categories of control measure reduce significantly the survival rate, especially the synthetic substances approved in Europe, showing an efficacy higher than 50% reduction. Other categories also reduce significantly the surviving insect rate, especially entomopathogenic nematode and fungi. Predator, on the other hand, does not show any significant result and has a high uncertainty. We perform a sensitivity analysis to check whether the conclusions were impacted by the type of trials included in the data set. So on the left, you have all trials analyzed together. On the right, you have the result obtained with a restricted data set without lab experiments. But overall, the conclusions were not substantially influenced by the type of trials. We repeated the analysis for each type of synthetic substances separately. Here you have the results. We can see that significant reduction were obtained with different products, especially with acetamiprid and for other products as well. Finally, we evaluated the risk of publication bias. Publication bias occurs when some type of results have more chance to be published than others, especially when significant results have more chance to be published than non-significant results. To evaluate the risk of publication bias, we can use this type of graphical presentation. So here you have the accuracy of the studies presented on the y-axis as a function of the effect sizes presented in the x-axis. So the most accurate study are at the top, the least accurate study are at the bottom. The study showing the highest reduction level of surviving rate are on the left-hand side, and the study showing a low reduction or no reduction at all are on the right-hand side. The black points correspond to the actual observations extracted from the papers, and the white points correspond to the estimated missing data. So when you have asymmetry in the black points, this asymmetry reveals high chance of publication bias, meaning preferential publication of significant results and a low chance of publication of non-significant results. So here the statistical analysis reveals that there is a high chance of publication bias because several studies are missing. And the studies that are missing are those with a low reduction of surviving rate or no reduction at all. Further analysis shows that there is about an overestimation of about 4% of survival rate reduction, especially for the study assessing entomopathogenic fungi. So in conclusion, this analysis shows that there is a mean reduction of insect survival by at least 50% with different categories of control measure. The highest reduction were obtained with authorized synthetic substances, but other type of control measure show a reduction significantly higher than 0 as well. Effect of predator and of several specific substances are highly uncertain, and there is some evidence of publication bias, meaning that probably some effect sizes are overestimated. So this work was done in collaboration with EFSA working group members working on this topic. So here the members are shown in alphabetical order. Thank you for their contributions, and thank you for your attention. Thank you.
Thank you, David. Our next speaker is Vinton Thompson from the American Museum of Natural History, United States. And his presentation is titled Extreme Polyphagy in the Vector Philenus spumarius, Overview and Implications for Xylella fastidiosa Spread.
Good morning, everybody. You say good morning. This is an American tradition. Actually comes out of African-American tradition, call and response. So you're awake, and we're all waiting for coffee, but we have a couple more speakers. It's great to be here today. This happens to be almost exactly to the day 56 years after I initiated studies on Philenus spumarius when I was a first-year graduate student at the University of Chicago in Illinois. Started out studying color polymorphism. Didn't think it was of particular importance, but here we are 56 years later. Philenus spumarius is really an extraordinary insect, and I want to harp on one aspect of its extraordinariness today, and that is its extreme polyphagy. They feed on lots and lots of things, probably almost everything. The work I'm about to describe was published 2.5 years ago. I did this in collaboration with two colleagues at the University of Sussex in Britain. This is the headline of the paper we published, and these are my colleagues, Claire Harkin, who was a graduate student, a PhD student at the time, who worked on this project, and Alan Stewart, who started out with me a long time ago working on Philenus spumarius color polymorphism. We could not have done this without all working together. This is Philenus spumarius. You're all familiar with it. One of the worst vectors you could have ended up with in terms of its versatility. You see a spittle, a nymph, and an adult. And here's one of the extraordinary things about Philenus spumarius, and I think a major contributor in reality to its extreme polyphagy, and that is its distribution. It's all over virtually the entire temperate Holarctic area, all the way from Ireland to East Asia. We don't know exactly what the boundary is in East Asia. We know it's abundant at least as far east as Kyrgyzstan. The records for China are there, but they're extremely vague geographically. It was introduced into North America a long time ago. We don't know when, but probably by the mid-1800s. There's a large distribution in eastern North America. There's a substantial distribution in western North America, separated by an area in which it doesn't exist. It's the dry plains area. And you'll notice there's a little island there, and that's the Wasatch Mountains near Salt Lake City, where Philenus spumarius has a substantial population. It also has been distributed to a number of islands, Hawaii, Big Island, to New Zealand. It's been there quite a while and it's quite abundant. It's in the Azores, and most recently it's been introduced to Iceland, probably in the last 25 years or so. Initially, when I started this work, the Bible of Philenus spumarius, and the biggest and most important monograph that's ever been produced, was this monograph Meadow Spittlebug by Weaver and King out of Ohio State. It's now more than 70 years old, but they presented the first comprehensive host list for Philenus spumarius. They had 383 hosts. There was also an unsubstantiated assertion in the literature that it had more than 1,000 hosts, undocumented, turned out to be true. We set out to compile a complete, comprehensive list as we could of Phalaenus spumarius hosts. I'm not sure there will ever be any such thing because some of the literature is so hard to find and so obscure. We had two primary sources for this. My contribution was sourced from an unpublished world database of spittlebug hosts, Cercopoidea hosts. It now has almost 10,000 records, of which just a little bit short of 3,000 are now for Phalaenus spumarius. The database is ironically not complete for Phalaenus spumarius. It doesn't include, for example, all the records from the Bridget project, which was the second major source of this study. And this was a project in which, a wonderful project, in which people solicited from ordinary English gardeners, English UK gardeners, I should say, include the rest of the UK in there, to go out and look for spittlebugs in their gardens. And they got thousands and thousands of reports. Britain is a great place to do this study. They don't have, for example, Leperonia coleoptrata. It's relatively easy to look at nymphal spittles and know what you're looking at, particularly if you eliminate grasses and the Neophalanus species that might confuse things. So this was a particularly benign place in which to do this major study. So we combined this data, the Bridget, unpublished Bridget project, and material from my database, which included mostly published sources, but also unpublished sources from people who had sent me information about 40 species that I had found myself. And this is the summary result. And this is really an extraordinary result. We documented 1,311 host species in 613 genera, 117 families, and by far, by a factor of 2 or so, Phalaenus spumarius is the most prolificous insect herbivore of all time. The second runner-up is fall webworm, and even its 636 hosts are biased because it includes artificial lab hosts. When we compounded these results, we did not include lab hosts. We filtered out things that were based on single isolated specimens. So I think it's a pretty good record. Most extraordinarily, the Bridget survey, these English gardeners found about half of all known species, and they found 358, or observed 358 species new to science. What do the results look like? Well, we published a gigantic paper in PLOS One, has a 29-page table listing all 1,311 hosts. I fought for that, by the way. Some of the reviewers and the editor said, let's put it in supplementary materials. I said, no, the most important thing about this report is really the list and not the things we have to say about it that are auxiliary. I wanted a list that people could search in a PDF, and it's quite useful for that. So if you want to know if Phalaenus spumarius has a host, go to this PDF. It's free access online, and you can search it for any Phalaenus hosts or potential hosts that you're looking for. It is organized by plant family. Within that plant species, genus and species alphabetically. It has the stage, nymph or adult. It has the geographic areas in which this host has been reported. It has the citizen science background for this, whether it's new to science, new to Britain, or duplication of an existing British record. And finally, selected references, so you can get a real documentation of where this information came from. The 4 highest families in terms of species numbers of hosts were composites, Asteraceae, Rosaceae, Fabaceae, and Poaceae. They together make up a little more than 1/3 of all hosts. This was not surprising. Phalaenus spumarius thrives on legumes, but they're not the top host group. The thing that surprised me most in these results were how many species. We had 73 species in the Poaceae and the grasses. So grasses are not Phalaenus spumarius' favorite host. I think most of you know that. They like herbaceous dicots, but there are a lot of grass host species. Where are we now? This is the new information I'm going to give you today. Where's the host count? I updated it last month. There have been many additions since we closed the count for our paper in May 2003. Some of the major papers that have supplemented the host count since then are listed. Many of those colleagues are here today. The count is up as of May to 1,430. So Phalaenus spinnerius is still the reigning world champion with no competition. 671 genera now, just added a couple of families, so up to 119 families. And here I think is probably the most important piece of data for this meeting. What's the intersection between Xylella hosts and Phalaenus spinnerius hosts? So we have 1,430 known Phalaenus spinnerius hosts. I went in and counted from the latest EFSA count, and you can see at the bottom, this is from the database for natural infections for the most rigorous determination of the finding of Xylella. And I count in that list 519 Xylella natural hosts. 211 of those, about 40%, intersect with Phalaenus spinnerius. So there is a lot of intersection between the host of these two species. Not good for you.
Apologies.
Oh, I'm stuck.
Vinton, I think the time is
Oh,
over.
time is up.
Thank
Okay.
you very much.
Thank you very much.
So, and now I invite Francesco Volpe from the University of Turin and his presentation on entomopathogenic fungi as multi-target strategy for the control of Phalaenus. So, floor is yours. 10 minutes, please.
Good morning to everyone. I'm happy to be here. I'm grateful to be here to share with you some results obtained during my PhD about the use of entomopathogenic fungi against Phalaenus spumarius. The management of this insect is crucial to contrast the spread of the bacterium, and we know that it's important to hit the target at the nymph stage. To date, the vector management is done with tillages in late spring against nymphs and with insecticides treatment against the adults. But often the efficacy of this treatment is affected by the recolonization of the treated areas during the season. Entomopathogenic fungi can be a viable tool in according with European policy to contrast the vector. And the aim of this work was evaluate the direct pathogenicity of 20 IPF strains against all stages of Phalaenus spumarius. We started this work with a screening of 20 IPF strains against nymphs. We treated Vicia plants with a conidial suspension. On each plant there was 10 nymphs, and we evaluated the survival of insects 4 and 8 days after the treatment. These results show that 6 strains reduced significantly the vector survival below the 10%, and the 2 most effective strains were both Metarhizium anisople strains. We used the most interesting IPF obtained in the previous trial in trials against the adult stage. In this case, we treated directly insect in a net cage. After the treatment, we put insect in these cylindric isolators and evaluated the survival 5 and 11 days after the treatment. The first trial showed that 3 strains reduced significantly the vector survival below the 25%. And also in the second trial, 2 strains reduced significantly the vector survival below the 25 strains. Also in this case, the most effective strains were both Metarhizium anisople strains. We tried also to evaluate the effect of entomopathogenic fungi against eggs, Phyllonosporius eggs, in a preliminary trial in which we treated egg masses, dipping them in conidial suspension. We used one of the most effective Metarhizium anisoplia strains, the strain of the commercial product Naturalis, the negative control. After the treatment, we put eggs, treated eggs and untreated eggs in growing condition and check them every day to evaluate the survival and the hatching. During the trial, we observed that untreated eggs hatched normally, while eggs treated with Bovaria bassiana and Metarhizium anisoplia showed a delay in the development, but also often there were the typical mycelium of the fungus used of the treatment, for the treatment. Sorry. The preliminary results showed that Metarhizium anisoplia, CN-V2I, significantly reduced the survival of eggs. At the end of this trial in lab condition, last year we tried to use the most effective Metarhizium anisoplia strain in field condition against nymphs. We tested this strain in Olive Grove in Crispiano. In this trial, we randomized 6 species in 5 blocks. We evaluated the effect of Metarhizium anisoplia BP1Q, the effect of the commercial product Naturalis. We used the mowing as positive control, and in this trial we evaluated also if there were differences between one treatment or 2 treatments with APF. The results in this slide on the right in the graph showed that all APF treatment significantly reduced the population abundance, and the mowing was the most efficient treatment. Interesting, the fact that the 2 APF application didn't significantly reduce the vector abundance compared to 1 application. Now I will spend a few words about the endophytic capability of our strain. In literature, it is known that some entomopathogenic fungi can colonize plant tissues and behave as endophyte. Some of these entomopathogenic endophytic fungi have some indirect effect against insects, like showed for Phyllonosporius also this morning. For this reason, to identify strains that exhibit both direct pathogenicity and indirect activity against Phyllonosporius could be an important improvement in the use of APF against Phyllonosporius. For this reason, we checked the endophytic capability of our strain in Cichorium intybus plants. We treated Cichorium intybus plants with a conidial suspension and evaluated the presence of the fungus 3, 7, and 14 days after the treatment in treated and untreated leaves. The results show that the most effective strains, the most effective Metarhizium anisoplia strains, showed a good endophytic activity. The strain BP1Q showed also a good activity, a good presence at 7 days, also in untreated leaves. For this reason, these 2 strains are good candidates for a dual action approach, an approach in which we combine direct effect and indirect effect against the target. But how we can use APF against Phyllonosporius in field? We have probably 2 key time windows. We could hit the target in spring. We have promising field data available about this. But we could also evaluate the possibility to hit the target in autumn. In autumn, in this time, we could hit both the adult stage and the eggs in field. And often in this moment of the Phyllonosporius life cycle, the adults show low fitness and mobility. We could also combine the treatment in spring and in autumn, also with the tillage, that is at the moment, the most effective treatment against Phylloenus marius. Naturally, further studies are required to evaluate the possibility to use APF in autumn, and also to evaluate if how our strains have some indirect effects against the target that could represent an important boost to the use of APF in the vector management. I think that if we combine the different treatment, if we use also the time window in autumn, we could reduce the vector abundance in a significant way during the years. I would like to say thank you to EFSA for the support. Would say thank you to my supervisors and to all the staff of the Basile Caramia Center that help us in the field trial. Thank you. Thank you for your attention.
Thank you, Francesco. And now we are going to start the pitch talks, which I remind you that those are the 5-minute talks with posters. And the first speaker of the pitch talk is Rocío López Gómez from ICATESIC, Spain, with a presentation on Draculacephala robinsoni, a potential vector of Xylella fastidiosa in Europe, distribution and population dynamics across the Iberian Peninsula. Please, Rocío, start.
Good morning, and thank you for the opportunity to participate in this session. Today I will present our work on the distribution and population dynamics across the Iberian Peninsula of Draculacephala robinsoni. This species is a sharpshooter native to the eastern United States, which was first detected in Europe in 2021, specifically in southern France and northeastern Spain, and since then its distribution has expanded rapidly in both countries. This introduction is especially important because various species within the genus Draculacephala are described as vectors of Xylella fastidiosa in North America. However, the ecology and population dynamics under European conditions remain largely unknown, and for this reason, the objective of this work was to assess the current distribution and population dynamics of robinsoni in Spain. To address this objective, systematic field surveys have been carried out in various regions, as the first introduction seemed to be in the region of Catalonia, specifically in Girona. This region was sampled once a month, and additional sites and regions were sampled with similar environments to detect the presence of the insect, specifically the regions of Galicia, La Rioja, and the Valencian community. Two sampling methods were used: sweeping net and debug aspirations, and habitats susceptible to colonization were surveyed, including permanent pastures, cereal and alfalfa fields, and the canopy and ground cover vegetation of various woody hosts such as vineyards, apple orchards, and olive groves. As the results, beginning with the distribution, robinsoni has not been detected outside Catalonia, but it has expanded to the north and to the south, with the southernmost record corresponding to the Barcelona province, and the northernmost in France, Occitanie, near Montpellier. However, the largest populations remain in Girona. In this region, on herbaceous hosts, the highest population densities were recorded in rice and perennial ryegrass fields. In the case of rice, around the month of October, and for the perennial ryegrass, it supported the highest populations after the rice harvest, or where the rice crop is not present. 4 major population peaks were observed per year, representing probably 4 main generations, but additional smaller peaks occurred during the winter. So these results confirm that the populations are well established in Spain and can maintain a stable and abundant population year-round. For the woody host, robinsoni was detected in the ground cover vegetation of olive groves and apple orchard, and in the canopy, on the canopy of a vineyard, an nursery vineyard. The higher density were recorded in the apple orchard, but it was specifically important the presence on the vineyard as is a host of Xylella. In this case, 3 population peaks were observed, and this result indicate that robinsoni is not restricted to the herbaceous habitats. To sum up, Robinson is currently well established in the northeastern Spain and continues to expand locally. Also is established in both herbaceous and woody ecosystems. And although the species has not been yet detected outside Catalonia, continued monitoring and further studies on its ecology and potential role as a vector of Xylella, specifically in association with grapevine, will be essential to properly assess its phytosanitary risks and support future management strategies. And to finish, I want to thank my research group, or partners of the project, of course, EFSA, and all of you for your attention.
Thanks, Rosia, for the talk. And now it's the turn of Tamara Hernandez-Lau, that will present her work on Metarhizium bruneo direct exposure that reduced survival and feeding in Philenus.
Thank you. Thank you also for the opportunity to present my work here. As a brief introduction, as you know, Philenus spumarius is the main vector of Xylella fastidiosa in Europe, and because effective treatments against the bacterium are limited, vector control remains like one of the main management strategies against one of the main management strategies. So, among the available biological control agents, Metarhizium bruneum has shown promising results against Philenus spumarius through indirect application, as Enrique told us before. However, its direct effect on the insect's feeding behavior and survival are still not fully understood. Therefore, the main objectives of this study were, first, to assess the feeding behavior of Philenus spumarius on Sonchus oleraceus and Olea europea when sprayed with Metarhizium bruneum, and second, to assess the mortality of Philenus spumarius when spread with Metarhizium bruneum. For this purpose, Philenus spumarius adults were treated with control or fungus solution and maintained on untreated Sonchus oleraceus plant for 72 hours under controlled conditions. After this incubation period, feeding behavior was recorded on both untreated Sonchus oleraceus and untreated olive plants using the electrical penetration graph technique. Following the recording, mortality was assessed daily for 10 days. And finally, microbiological analyses were carried out to detect the presence of the fungus in the insects. The EPG technique allows us to monitor the insect's feeding behavior in real time. By recording electrical signal generated during feeding, we can identify different feeding activities such as xylem ingestion, for example. And regarding the result, I will focus only on the most relevant findings. Looking at the total time spent in each feeding phase, both control and treated insects, and in both in Sonchus or olive, spent most of their time ingesting xylem. Regarding the sequential variables, treated insects on Sonchus oleraceus reached the xylem and initiated xylem ingestion significantly earlier than control. In olive, although differences were not statistically significant, the same trend was observed. And as for mortality, Metarhizium bruneum significantly increased the mortality of Philenus spumarius in both in Sonchus or in olive, without significant differences between two assays. And as a conclusion, the direct application of Metarhizium bruneum altered the feeding behavior of Philenus spumarius, promoting earlier access to xylem ingestion, particularly on Sonchus oleraceus. A similar trend was observed in olive, where treated insects also showed longer xylem ingestion events. This consistent pattern across host plants may indicate a compensatory response to fungal colonization. In addition, Metarhizium bruneum significantly increased the insect mortality, supporting its potential use in integrated pest management strategies against Philenus spumarius. Thank you for your attention.
Thank you. And our next speaker is Pierre Rouaillet from the University of Turin with the presentation Uncovering Viral Partners, How the Virome Shape Fitness in Philenus spumarius.
Good morning, everybody. So, first of all, I'd like to thank the EFSA and all the partners for having brought to life this very beautiful conference. And now, if you may, I'd like to proceed directly to the core of my research, which is about the virome of Phylloemus spomarius and its effects on the host fitness. So you may wonder, why studying the viruses? Because as far as I know, I'm kind of the only one studying the virome of Phylloemus in this conference. But usually when we talk about viruses and insect, we mean plant viruses. But insects are only seen as vectors. But there's a huge gap in the knowledge of viruses within insects. And in this matter, a colleague of mine from the EPSAP from Torino has done the first characterization of the virome of Phylloemus spomarius. But there is still a lot to discover for the species, but also for the interaction between the host and the viruses. So my work is in the continuation of the work of my colleague. So to continue the characterization of the virome of Phylloemus and also to evaluate the effects of this virome on the host fitness. So how did we do? First, we went to some locations where my colleague did the sample. We collected insects and we extracted the RNA. We sent it to sequencing. We analyzed the nucleotide sequences and then we identified the viruses. So this was for the characterization part. For the fitness, basically, we were kind of lucky because we found that one of the population was virus-free. And this is gold because when you want to do some comparison, it was perfect. Because we extracted viruses from the other population and we injected it inside this virus-free population. After we've done that, we gathered 15 couples from each of the population and we put them on cylinders with a chicory plant inside, and we monitored some characteristics, some parameters of fitness. So for the characterization of the viruses, here's the list of the viruses we found. And you can see, so there is only Chiari and salt because obviously Arnasco is virus-free, so there is nothing to see. And in comparison to the work of my colleague, we found 3 new viral species. For the fitness, the first parameter was the longevity of the insect. So since this was done for the F0, there is not the comparison with the infected population because it was a very traumatic process for them, and so there's no point to follow the survival. Basically, every curve was the same except for Bellino, but this population is a bit tricky because we take them from a location 2,000 meters in the Alps and we put them in the plain. So it's most likely the reason why the population collapsed so quickly. For the numbers of eggs that was laid per females, we have the same trends. Every population was kind of equal except for Bellino. And then when the things begins to be interesting, it's for the F1 with the eggs and the hatching rate of the eggs. Where you want to look is the 2 bars on the left, which is Arnasco and Arnasco infected. So the virus-free population and the virus infected. And I wasn't expecting that, but it seems like injecting viruses into this population increases the hatchability of the eggs. So, why not? But this continue for the survival of the larvae, where the infected larvae survive more than the non-infected population. So, for now, I still lack some data about the viruses and the other parameters of the F1. And so the work is in progress, and I hope maybe next year or another moment we will discuss about that. And yeah. This is it. Thank you.
Thanks, Pierre. And our last but not the least speaker, Julia Lopez Mercadal, from postdoctoral researcher from Agroscope, with a presentation on assessing vector competence and Xylella fastidiosa transmission in Switzerland on different plants.
Thank you very much, Daniele Neverina. the slide. Okay. I'm pressing hard.
There's some issue.
You see? All right. So, Xylella fastidiosa is absent from Switzerland, but is also considered as a quarantine organism like in the European Union. Due to the risk of introduction of Xylella fastidiosa from the neighboring countries, we proactively evaluate or are evaluating the competence of the commonly widespread potential vectors in Switzerland. For this, we aim to perform acquisition and transmission analysis of these widespread insects that I will show you afterwards, in between the agriculture, forest edge, and forest species. Okay. So to perform our analysis, we did this acquisition and transmission test inside biosecurity level 3 conditions during July and August of 2025. These plants that we used for the analysis were seedlings from local producers. To achieve our objective, we collected the insects from the field and we focused on 3 insect species, Afrophora alni, Philainos spumarius, and Cicadella viridis. We made these insects to feed on 2 different feeding systems. From one side, from source plants infected with 2 Xylella fastidiosa strains, and from the other side, using artificial diet. For these experiments, we used 6 plant species of interest for Switzerland, for the agriculture, for the forest, and for the forest edge. After those insects were in contact with Xylella-free plants, and then we analyzed the insects and the plant for the presence of Xylella fastidiosa. And what did we see in this first stage of our project? So regarding the survival of the insects, we saw that Philainos spumarius and Cicadella viridis had a great survival rate. More than 70% of the insects survived during the whole experiment. Acquisition rate was low from plants, actually less than 2%, but acquisition increased during DSAs with artificial diet. Both Xylella fastidiosa strains were acquired, and regarding the transmission, it was only occurring when we used the artificial diet system, and only with Xylella fastidiosa strain, and transmitted to Quercus robur and Rubus fruticosus. Finally, the first conclusions that we could draw after this first stage is that we should take into account the role as potential vector of Afrophora alni and Cicadella viridis in the Swiss context, that forest and forest edge species can play an important role as reservoirs of Xylella in the landscape. And I believe that those results can help to understand how Xylella could spread in the Swiss environment, and that this could serve as a stepping stones for targeted surveillance and early detection in Switzerland. Thank you very much.
We encourage you to see Julia's poster. Thank you. Okay, now is the time for questions from the public. We would ask you to introduce yourself very briefly and where are you coming from, so the speakers, they know with whom they are interacting, okay? Okay, here, the first row, can we ask the hostess to bring here the
Apparently I got the power. Francesco Porcelli, Università degli Studi di Bari, Aldo Moro. Simple question. Do we have a vector threshold suggesting the opportunity for transmission management?
Yeah, it's about transmission. Anyone want to reply to the question of Professor Porcelli? If we have a threshold for vectors? I can reply. It was like a long discussion, even with David and Evelina in the working group, and actually the honest response is we do not. It's like it's one vector is enough. Obviously, the theory is the lower the number, the better it is, but we have no threshold so far.
Thank you. Next question. Here. Yeah.
Can you hear me? Okay. I'm Reinaldo, I'm Brazilian, but I work with Saudarelli and Saponari groups here in Bari. And I have two questions. First question is for Cornara. I'd like to know what you think about the behavior that you find in insects. Do you believe this is kind of Xylella is controlling the insect, secreted some one or more factors to keep this behavior, or it's a secondary affected because the insect is infected with Xylella?
I think that it would be very cool to talk about manipulation, but it's not. I'm a scientist, I have to admit it's not. It's just, to me, incidental effect of the fact that when they occupy the foregut, it's like they reduce the lumen. The lumen of the foregut becomes narrow, so it's very hard for them to feed, and so you see all these cascading events. Plus you have that chitinase and degradation of the cuticle, maybe that is like detrimental to insects, but it's not a manipulation. Then, the effect that we could have, yeah, are effects, but it's not manipulation.
Okay. My second question is for the other speakers that have been working with entomological fungi. If you tested like non-target insects to see the effect about these fungi.
So I think the question is for all the speakers that work, like Enrique, want to reply?
Yes, it's necessary to do it. I don't see the person. But the strategy for application is quite important. As you have realized, we may apply different strategies. It's not the same applying the inoculum to the soil, trying to colonize the plants, in which case the non-target fauna is almost no contact with the inoculum, and probably the most risky, but using a microbial strategy is spraying all the covers or even the olive canopy. But in these cases, we have to, of course, we should have to compare the possible effect of the fungus with a chemical, because sometimes we pose many questions about the microbials. The microbial is non-risky for bees, but we have to use a control. We have two possibilities: not doing anything or using an insecticidal control, and to compare which are the comparative effects. So first of all, the strain, selecting a specific strain, the more specific as possible. Second one, the strategy for application, trying to use, I have shown there is a cumulative effect. And third one, we have a problem, we have to solve the problem, we have to select between using pyrethroids or using a microbial. So let's make the same questions always for all control measures.
There is another question from this. Can you bring microphone here?
To the first row.
Thank you. Is it working?
Yeah.
Well, it's not really a question, it's a follow-up on the response. Just to explain, if you would bring a plant protection product to the market, then these tests you were asking are part of the data requirements. So they would need to be done before the active substance can be approved and before a plant protection product can be authorized. And what the colleague behind the column said, that the strategy is important. This is essential for getting a dossier through, because the first step is to get the active substance approval. But this means that in this dossier, you need to have a representative use. The representative use means a strategy. What is the plant protection product? What is the dose? How would you apply it? When would you apply it? Etc., etc. And this representative use is then used to do their particular risk assessment based on this use. So to get it, let's say, to the farmers, all these questions are part of the regulatory steps to be done. And yes, I mean, it includes bees, etc., etc. Yeah. I just thought it's important to
Question from Giuseppe.
I have a general question. So what is the diversity of Philaenus fumarius population in terms of susceptibility to Xylella infections?
Nice one, thanks. So the thing is that as far as we know, there is no vector strain specificity, but this is something that we should investigate, and we were discussing with Rodrigo. Oh, that would be cool to see. Maybe there are differential effects depending on the vector. There is a very complete work done by Liz Clark at Berkeley, analyzing, for example, how big the foregut is in the different species. Maybe the bigger the foregut, lower could be the extent of the effect because of it reach more time, and I don't know. It's it depends. There is a lot of an unexpected journey. If I can intrude, I have a question for Enrique, specifically. I am absolutely fascinated by a recent publication about the possible emission of HIPV, herbivore-induced plant volatiles, from plants treated with entomopathogenic fungi. So what's the perspective? Is that true? We can recruit biological control agents through entomopathogenic fungi?
Yes, the endophytic colonization have an effect on the chemical ecology of the plant. This includes the covers. So yesterday we showed a poster by Dr. González Mas, in which we show how, depending on the strain, we can have an attraction of not an attraction to the colonized covers. And that's a new possible strategy, an open door to the push and pull strategy that would be very nice. Unfortunately, we have not been able, if you have a new project or something like that, you could, to work together, people from covers and people from entomopathogenic fungi, to try not only this cover or this other cover, but a cover that is an artificial cover coming from an inoculated seed. So we have to compare standard covers with covers colonized, because we could have new applications to this extent.
Okay. I have also a question to Julia Lopez Mercadal. What are the agro-ecosystems most at risk in Switzerland, in your opinion?
Can you please repeat, because I hear it very slow?
What are the agro-ecosystems most at risk in Switzerland?
I would say it really depends on the region of Switzerland that you are studying, because there are a lot of little micro habitats around. It depends if you are in the south, more in Ticino. I would say it's more about agriculture, vineyards, for example. But then if you are in the other side of the Alps, it's more about apricots, apples. But there is much more interest, I would say, about forestry, because they care much about this matter. We have the Alps that really cover much part of Switzerland, and we can find the vectors really everywhere.
Thank you.
Thank you. There is a question from public.
Thank you. That was fantastic to see all the creativity from all these researchers of all ages, so that's very encouraging. I have a question for David. we have that breadth of researches going on the whole chain, on all possibilities. And I really appreciated his effort to be doing this meta-analysis for the management of the vector. David, would it be possible to undertake meta-analysis for all the other aspects of management? We had cultivar, we had, well, many options offered, to then have a meta-analysis of all the meta-analysis together to come to a systems approach. Thank you.
Thank you for your question. It's probably relatively easy to perform other meta-analysis to evaluate other types of single control measures, like different type of cultivars, for example. I don't know, different type of cover crops or soil tillage, or so. I think as soon as you have several experiments available for one given type of control measure, there is no reason for not being possible to do a meta-analysis. What will be more complicated, I think, is to evaluate combination of control measure, because to have a good meta-analysis, you need a number of experiments. And as soon as you are combining different types of treatments together, it's difficult to find a sufficiently high number of experiments assessing these specific combinations. So my answer is that yes, I think it's possible to evaluate a large range of individual control measures using meta-analysis, but probably it will be much more difficult to use the same approach to evaluate combinations of techniques.
Thanks, David. I think there is a question from the public, and then Blanca.
Thanks. I am Budur, researcher. I'm doing my research in Siambari cooperation with the CNR. I have a couple of questions. First, the question, if there possibility to discover more the diversity of microorganisms inside the vector, since there is a few studies about it, some of those microorganisms, it's non-pathogenic for the vector, but it have an influence effects against Xylella. So my question is if there is any possibilities to discover more about these microorganisms.
I think that first, Pierre, I think you can reply to the question.
Okay. But first of all, about the viruses, who am I talking to? Sorry? No? Bo?
There. She's standing.
Ah, okay. Ciao. For the virus part, honestly, I didn't look with the Xylella effect, the relationship. But for sure, if my students truly see differences about fitness and about all the physiology, for sure there will be an influence with other microorganisms. But as far as is my research, we didn't look to any other effects with other microorganisms. But we could actually see, because with one of our colleagues from the University of Turin that was working with entomopathogenic fungi, we had some discussions to understand if there is like room for studies with the relationship between microorganisms, inside the insect. So I definitely did not answer your question, but this is my answer.
I can just try to add something. It was like inspiring myself to like a book on the correlation, you know, all the gut microbiome, brain correlation and all the effects. I read recently a book that let me think about something that is important. These kind of studies are difficult to perform, and most of the time the thing that we publish are correlation rather than causation. It's very difficult to say this variation in the microbiome is causing this. We always say it's correlating this to the other, and we really risk to produce biased results and then to push at the end farmers toward like the wrong solution. So we should be very cautious. So, and the answer to this question is it's very complicated. That is very interesting and very complicated.
And now Blanca, please.
Yes, I just wanted to add something to what David said. So when we were updating at EFSA the utility of the different risk reduction options for Xylella fastidiosa control, we also thought to perform a meta-analysis to evaluate the efficacy on the bacteria. But for doing that, you need the same variable. How do you measure the efficacy of a treatment against Xylella? And what we found is in each study, scientists use different approaches. Some just disease severity, other population levels of the bacteria, measured through different methods. So even we try to propose to do that, it was not possible. So for doing a meta-analysis, first you need to have the same way of measuring the efficacy of the treatment.
Thank you.
Se posso aggiungere qualcosa, scusa? Sì, solo sulla parte ancora di interazione. Scusa. Right. On the part of relationship between the microorganism, just to add something, we have here people that also works on Wolbachia, and actually it has a really deep impact on the biology of Philenus spumarius. So I go again with the side of Daniele Cornara in the sense that it's very difficult to see the interactions between these microorganisms. It needs to be very specific, but it really shadows the effects of many things. So yeah, that's all.
Thanks, Pierre. There is a question from the public, and then there is Rodrigo.
Sorry.
Hello.
Yeah, I have a question.
First?
On the left, and then Pasquale.
No, Pasquale, sorry. There is first Pasquale. Let's avoid
I was trying, sorry. No, Philenus gets sick with Xylella. So it is a no choice situation. So why should I deliberately choose to get sick?
Because of you do not. It's like it happens by chance. I'm giving you my 50 cents. I don't have final data, but it's just, it happens that it acquired a bacterium, and we only think about Xylella in sick plants, but the majority of the plants are asymptomatic. So it just, the bug lands on an infected plant, gets the bacterium. What we notice, and I didn't discuss, is that when Xylella is inside, it increases the frequency of the digestion behavior, that is this sort of coughing, that is inoculation. So for Xylella, it's beneficial. For the vector, it's not. Probably it's just incidental, but we don't know what the effects in the long term are. So these are things. Question there?
Hello, I'm Eli Falcon from Germany. I wanted to ask a question to Rocio Lopez about the vector in the Iberian Peninsula. Have you actually detected Xylella from the samples that you have collected? Or have you looked into this?
Yes, we have looked into this, but as far as I know, there were no positive samples on Xylella.
Okay. My next question is to Pierre Royal. If they have looked into, so since you found one population with and without virus, have you looked into the differential genes expression in the vector, and not in the virus, but in the vector, that may be also explaining the egg hatching behavior because they're promoting the productivity of the vector to basically pass on the virus to the next generation and all, or the epigenetic of the vector. So are you looking into that, or will you look into that?
No, actually, the genetic itself, we don't go deep inside. It was really a characterization work. But definitely, those viruses that we found in the population, we actually found them for the past 3-4 years. So it can be assumed that if
viruses are maintained in the population without altering too much the fitness, for sure they have some benefit. But then about like the gene expression, etc., we are not going today in this kind of matters. But if the project continues, yeah, it for sure might be interesting to investigate.
Thanks. Question from Rodrigo.
Rodrigo? It's at the end.
Let's go.
Very much at the end. Standing. The microphone is coming to you.
Yeah, thank you. It's on? It's on now, okay.
Yes.
So first, the previous question, I would just want to say that there is no evidence anywhere of Xylella being beneficial to the vectors. That's not my question, though. The question is to the lady from Switzerland, and I just want to confirm the slide where you showed that you got transmission of a grapevine infected with multiplex, and that's the question.
Can you repeat? Because from here, it's not, we don't hear well here.
Okay.
Slowly, and if you can repeat.
All right. The question is for the lady from Switzerland.
This we got.
Okay.
No, use the microphone.
No, you need to use microphone. Here we cannot hear you.
I think I got the question. It is if I got transmission from grapevine infected with multiplex. Okay, the answer is, I think we got one example, but now I cannot remember by heart how is it, but if it was only one insect. But if you want, we can check in the poster with the results in front of.
Yes, and we will do that later with Rodrigo at the poster. Thank you. And I have a question to Enza, if I may. Have you monitored the impact of nymphs management on biodiversity?
Okay, sure that soil tillage have a lot of negative impact on biodiversity, and so we have tested other alternative solutions to have other tool to control the juvenile stage. The other solutions do not have the same effectiveness in reduction of the population, but as I said during the presentation, they can integrate with mulching, so to reducing the density population without modify the biodiversity.
Thank you. Thank you very much.
I have a question for Julia. It's a very technical one.
All right.
And is, so you said that you administered Xylella with artificial diet, and I guess it's multiplex and fastidiosa genotypes?
Yes.
Okay. Did you supplement the media with galacturonic acid or pectin or chitin, or did you give the Xylella as it is from a normal plate?
No, I used the artificial diet that I think you also used in one of your essays that you did in 2018. It was the Olidic diet already in the literature. So I put different amino acids in there.
But you grew Xylella on a normal medium, not something supplemented with something else.
No. Xylella was growing in PD3.
Thank you. We should discuss.
So our session is actually finishing, and there is a coffee.
There is Domenico Bosco.
But we cannot not take the question of Domenico Bosco. So
Thank you very much at the very end.
Last one.
No, mine is not a question. I just want to try to add something to the question that was raised before on the potential of other microbes for the control of Philenus. And I just want to tell that besides the big effort which is done by Massimo Giorgini, I guess he's in the room in Napoli, in studying Wolbachia effect on Philenus spumarius together with Luigi Iodice, so they are a good reference persons for that. We also sequence total DNA for microbes, and basically what we found is an obligatory symbiont, which is Candidatus sulchamulleri, but this is not pathogenic at all. This is needed by the insect and another microbe. This work has been done mainly by Michela Chiumenti here in Bari, and we didn't spot further possible microbial partners that are potential pathogens. That's it.
Thank you, Domenico. So, Daniela, shall we thank and finish?
I think that talking about science is the best, but the second best is coffee that I didn't take this morning. So it's coffee break, 30
and
minutes.
Please continue discussions.
Good. Ciao. I need my coffee break.
Yes.
Can have a seat.
Dai, iniziamo.
So the session is starting. Is a session number 5 on impact and social aspects of Xylella fastidiosa, epidemics and management. And we now give the floor, but we ask first the presenters to come here, to sit here. And I would like the first speaker to have the floor. He's already on the floor. So Pierpaolo Miglietta. His speech is on from production decline to structural crisis, olive meal dynamics. Please have the floor.
Okay, thank you very much. Good morning, everyone. And thank you for the opportunity to present this study. The title of the work is From Production Decline to Structural Crisis, Olive Meal Dynamics in Xylella Fastidiosa Affected Area. The study focuses on the consequences of the Xylella epidemic for the olive oil supply chain in Salento. In particular, we examined the decline in olive milling activity and the role that replanting is playing in supporting production recovery. The main idea behind the research is that the impact of Xylella cannot be evaluated only by looking at the loss of the olive trees, but also is necessary to consider the effects on processing infrastructure and the long-term capacity of the supply chain to recover the production. The agenda of the presentation is organized in 2 parts. First, I will examine the decline in olive milling activity. I will discuss the temporal dynamics of the number of active mills, their spatial distribution, the municipal mortality rates of olive mill, and the evolution of the main production variables. I will also present some correlations among the different olive oil supply chain variables, and in the second part, I will focus on the post-Xylella recovery process, and I will describe the annual and cumulative evolution of replanting, its distribution also by cultivar, and the spatial relationship between replanting and some subsequent olive oil production recovery. So we can say that the study objective is twofold. The olive oil meal mortality aim to quantify the reduction in the number of active mills, identifying which municipalities most severely affected by the Xylella fastidiosa, and assess how the territorial distribution of olive mills has changed over time. At the same time, we will also analyze the evolution of the main production variables, which we consider in particular olives processed on the mill's own account, the olives processed on behalf of third parties, and the purchased olives. And at the end, we will analyze also the quantities of olive oil produced. Second, we will focus on replanting and post-Xylella production recovery, measuring the spatial association between replanting intensity and subsequent olive oil production at the municipal level. We also seek to estimate the distance beyond which this relationship is no longer statistically structured. In other words, we can say that we want to determine whether recovery dynamics are confined to individual municipalities or operate across broader territorial scale. We will see that this is the case, the second. So we will operate at the cross-border territorial scale. So this slide shows the evolution of the number of active olive mills from 2014 to 2026 season. At the beginning of the study period, there were 232 active mills. The number temporarily increased to 2047 in the following season, but this increase was not maintained because, as you can see, a clear downward trend subsequently emerged, and the contraction became particularly evident, especially in the 2018-2019 season, when the number of meals fell to 182. The decline then continued almost uninterruptedly, reaching 155 meals, and then 100 at the end, 111 in the most recent season. So less than the half of the beginning period. So overall, the number of meals declined by more than the 50%, and the persistence of this trend suggests that the phenomenon cannot be interpreted just as a temporary fluctuation, but it points instead a structural contraction in the processing capacity of the local territorial olive oil supply chain. The maps, unfortunately, are not so visible, show how the decline in olive meals has developed across the territory of Salento area. During the first years, olive meals were relatively widespread, and many municipalities had at least one active processing facility. Over time, this territorial network, we can say, became weaker and weaker. Some municipalities experienced a gradual reduction in the number of meals, and some of them lost their entire local processing capacity. This is very important because the crisis affects not only the number of agricultural firms or farmers, but also the geographical accessibility of milling services. When local mills disappear, farmers may need to transport olives over longer distances, and this increases costs, creates logistical difficulties, and affects negatively the timing of the processing of the olives. So we can say that the decline in mills may therefore reinforce the agricultural crisis. Lower olive production reduces, obviously, the economic viability of mills, while the disappearance of mills makes it more difficult to maintain or restart olive oil production. These slides summarize the percentage reduction in olive mills at municipal level. A larger number of municipalities are concentrated, as you can see from the graph, in the highest mortality categories, between 75% and 100%. The intermediate categories are also well represented, indicating that the decline is widespread rather than limited in a few isolated municipalities. And by contrast, you can see that the lowest mortality category, between 1 and 25%, includes only a small number of municipalities. This implies that very few areas were able to preserve most of their original processing capacity. The municipal perspective we adopted is relevant because aggregate figures may conceal important territorial differences. We can say that in several municipalities, the crisis has resulted not simply in a reduction of the number of mills, but in the near total disappearance of the local processing system. These slides represent the evolution of the main production variables throughout the study period. The data show a general decline in the quantities of processed olives and produced olive oil. Initially, olives processed on behalf of third parties and purchased olives represented the dominant component of milling activities in the Salento area. Own account processing was also important, but generally involved smaller quantities. The time series shows significant annual fluctuation. As you can see, this is partly expected in the olive oil sector because production is obviously influenced by alternate climatic condition and bearing. However, beyond this normal fluctuation, a clear structural break you can see in the 2018-2019 season onwards can be observed. So after this point, the quantities of processed olives and produced oil remain substantially lower than those that was recorded during the initial years. The sharp decline in purchased olives and also third-party processing indicates a general reduction in the volume of raw materials available to the milling system. So the impact of Xylella is visible not only in the production of individual farms, but also in the overall flow of olives through the supply chain. The correlation matrix here show strong positive relationships among all the variables included in the analysis. The quantities of purchased olives are strongly correlated with the olive oil production, and this relationship is also technically expected, but confirms also the consistency of the data. Purchased olives are also strongly correlated with own account processing, third-party processing, and olive oil production. So when olive availability decreases, the reduction affects all processing activities and both categories of olive oil production. The results of these, therefore, suggest that the observed changes are driven by common territorial factors rather than by problems affecting only one specific segment. And this high level of interdependence also between variables also shows a shock originating in agricultural production and how this can rapidly spread throughout the entire supply chain. This is the contraction of the supply chain to the process of olive growth generation by year. Between 2018 and 2020, replanting activity gradually spread across the territory. So there is an up, the replanting activity. However, the number of interventions was still limited and the spatial pattern was relatively fragmented. A strong acceleration occurred in 2021 with particularly high concentration of replanting in some areas, especially in the northern part of the study area. The years 2022 and 2023 represent an intermediate phase with a lower and more uneven level of activities. In 2024, a new increase can be observed together with an expansion of the areas involved. We can say that the overall process is still discontinuous rather than progressive, and this suggests that replanting may be influenced by the timing of financial, for example, incentives, administrative procedures, investment capacity, and also the availability of certified safe nursery material. This is the annual maps which show when replanting activity occurred. Is a cumulative perspective which allows us to observe how the new olive growing structure has progressively developed across the territory. And the cumulative maps provide a clear representation of the role of the overall regeneration process in the Salento area. And unlike the annual maps, which show the interventions carried out in each specific year, these maps illustrate the progressive accumulation of replanting activities. So the distribution from a spatial point of view remains highly uneven. Some municipalities show a relatively strong regeneration process, while others continue to record very limited replanting activities, as you can see from the white part of the maps. This difference may be related to farm structure, access to financial support, and also local production potential, but also by the intensity of the damage caused by the Xylella fastidiosa. This cumulative perspective is relevant because newly planted olive trees require time before they become productive. We know this, it's obvious. So for this reason, the effect of replanting should be evaluated by considering both the spatial approach and the time needed for new growths to enter production. This slide shows the spatial distribution of replanting according to cultivar type. The cultivar selection is a key element of post-Xylella fastidiosa reconstruction because it affects disease tolerance, agronomic performance, and productivity in terms of olive oil quality. The maps show that the cultivars are not uniformly distributed across the territory. Some municipalities are characterized by strong concentration of specific varieties, while others show a more diversified patterns. To examine the spatial relationship between replanting and production, we used the variographic approach. The first step consisted of a direct variographic analysis of cumulative replanting and olive oil production. This analysis allowed us to verify whether each variable had a spatial structure. In other words, we can say that we assessed whether municipalities located close to each other tended to show more similar values than municipalities located farther apart. Once spatial structure had been confirmed, we calculated the cross-variograms between cumulative replanting and production during the subsequent 2 years period. The use of a time lag is important because replanting cannot produce an immediate production response. These slides present the central result of the spatial analysis. The two cross-variograms compare cumulative replanting during 2018-2019 and 2018-2020 with olive oil production in the subsequent 2-year period. The two curves display a largely similar pattern, and this suggests that the result is relatively stable and does not depend only on the specific time interval. The horizontal axis represents the distance between municipalities, while the vertical axis measures the joint spatial dissimilarities between replanting and production. We can say that as distance increases, the spatial association becomes progressively weaker. It remains statistically detectable up to 35 kilometers. At this distance, the variogram reaches the seal, and beyond the seal, geographical proximity no longer makes a significant contribution to explaining the joint spatial pattern of replanting and production. Shifting to the conclusion, the results show that the Xylella crisis has evolved into a structural supply chain crisis rather than remaining only an agricultural production shock. The persistent decline in olive meals threatens the processing capacity that will be required if olive oil production recovers in the future. At the same time, replanting remains spatially uneven and discontinuous, and this highlights the need for stable financial support and greater coordination among interventions. We can say also that the association between replanting and subsequent production is strongest within 15 to 20 kilometers and remains statistically structured up to approximately 30 to 35 kilometers. This means that the recovery dynamics operate across connected sub-regional areas rather than being confined to an individual municipal level. Consequently, policy action should be coordinated across neighboring municipalities, taking into account this territorial scale identified by this spatial analysis. I want to thank you for your attention, and also I want to thank Frantoio Le Aree Associati that gives us the opportunity to process this data. President Michele Librandi is among you. So I want to thank them specifically, because otherwise we will not have the opportunity to give these evidences of the tragedy that our territory had in the past, and the hope for a renewal phase, we can say, renewal phase. Thank you.
Thank you for the presentation and for saving some seconds. So I give the floor to the co-chair, and Claudio Sopranzetti from Oxford University. He will present the topic on coexisting with Xylella and anthropological perspective.
All right. Thank you. Thank you, everybody, for listening. I'm just waiting for the PowerPoint to start. All right, yes. Okay, so I would like to start this presentation with a question that I think you also might have, which is namely, what can an anthropologist offer to a conference like this? Why are we here? Why this panel? What is the contribution? And to answer it, I want to ask what happens when epidemiological knowledge that we heard a lot about enters a territory made not only of host plants, vectors, and bacteria or climates, but also of farmers, landowners, and mill workers. And I want to do this through one word that is also in the title of my presentation, and this word is coexistence. Now, coexistence is a difficult word in the case of Xylella, as you know, because the bacteria still is legally and institutionally a quarantine organism in the European Union, and the logic of prevention, containment, and eradication remains central to plant health policy. This is obviously necessary in many contexts, especially in areas of new introduction, buffered zones, and active expansion. So let me be clear from the beginning. I am not using coexistence as an argument against containment when this is possible. But I think it's time that we start also to consider a set of different questions. What happens when Xylella is no longer a threat approaching from the outside, but an established condition of life as we're hearing in a territory? And the practical question is no longer how do we prevent Xylella from spreading, but how do we farm, govern, and live with it? It is in this sense that I use this word coexistence, not as some ideally peaceful and harmonious condition of living with the bacteria, but as a difficult, uneven, and often conflictual set of practice with which people inhabit the landscape when the pathogen has become de facto endemic. My argument here for these 10 minutes is quite simple, but I think it has quite profound consequences. Human conception and practices are not external to these processes. They are part of the epidemiological landscape itself. How people think about trees, how they classify them, how they decide whether they're alive or dead, as we see in a second, whether to remove them, to abandon them, to replant them, or simply to leave them there, has profound epidemiological consequences. This practice shapes the landscape in which the bacteria, the vector, and the host plant interact. We always talk about this triangle. The missing element is the human organisms in that triangle. So the point is not that we should add a little bit of social context or impact or stakeholder participation after the scientific analysis is complete, but rather that if we ignore these practices, we risk misunderstanding a large part of the very system we are trying to make sense of, therefore drafting policies that are built on this misunderstanding and ultimately failing to limit the effect of the epidemic. So let me give you a few examples of how this misunderstanding created and the consequences they can have. In the infected area of Salento, one essential distinction emerged again and again in my research. This is the distinction between productive and biological death. Here you see two trees that we can both identify as dead, but it's quite different what people mean when they see a dead tree here. By productive death, I mean a condition that you see there in which an olive tree may still be biologically alive, but from the point of view of agricultural production is no longer viable. It requires too much labor and too much care for too little return. The tree is not dead in a biological sense, as you see, it still has vegetation, but is dead as a source of income. This distinction matters enormously because much of the conflict around coexistence is really a conflict about these two things. Is this tree dead or not? For some people, and we heard from them, productive death is simply death. If a tree is no longer produce enough to sustain agricultural labor, then it should be removed and replaced. For others, this is not the case at all. A tree might be productively dead and still be worth saving. So the language of saving is often because this is what they are looking at to save. Because various reasons, family history, landscape. So this difference is not merely symbolic. Conceptualizing a tree as dead when it stops to generate profit, which is completely right, or dead when it still vegetates, which is also right, produce different forms of action that end up affecting the vector population, the epidemiological pressure of the bacteria, and the adoption and refusal of phytosanitary measures. And these are the these actions are what coexistence actually look like down there. So in the affected area, for instance, I have encountered three broad forms of coexistence that I quickly go over here. The first is through abandonment. This is perhaps the more widespread and at the same time the least discussed. Many of the small plots that you see here have been abandoned and they're changing into something else. And this obviously has consequences for fire risk, for land value, for future agricultural recovery, for oil mill, as we heard, but also for epidemiological surveillance and for the management of vectors. The second form of coexistence is through replacement. Here, the infected trees are understood primarily as a failed productive system. So the answer is to remove and start again, often with this new variety. And as we heard before, about 3 million trees have been planted so far. So this, of course, implies a different landscape, different labor regime, and a different epidemiological condition. The third coexistence is maintenance. Some people, again, who see this productive death not as death, try to keep heavily affected trees alive through pruning, through all different measures. Of course, from the point of view of maximizing production and making production, this is a nonsense, right, in that sense. But however, at the same point, from the point of view of heritage, memory, or tourism, this is becoming a quite diffuse form of coexistence with epidemiological consequence. So I don't have the time here to tell you more about this emerging model, and the Q&A are here for that. But I hope this outline shows you that coexistence does not mean one thing, but a set of different practices. What I want to stress, however, is that these are not simply opinion or attitudes. They are epidemiological consequential practices. They affect the epidemiological landscape of this area. They matter because abandonment changes vegetation and land management. It affects CO2 level, ozone level, soil radiation, and so on. They matter because replanting changing host distribution and density and affect future epidemiological risk. They matter because maintenance changes the persistence of infected tree, therefore effect on the vector, on the level of bacteria, and so on and so forth. This is why I think coexistence must be taken seriously as an analytical, epidemiological, and policy category. A quarantine frame is indispensable when the problem is introduction, spread, and containment. But in territories where Xylella is already endemic, coexistence is not the opposite of epidemiology. It is a terrain in which epidemiology has to operate. And this means that anthropology, I think, can contribute to discussion for plant health, not by replacing biological analysis, but by helping to describe and analyze the human practice that shape the condition under which an epidemiological process unfolds. In this sense, what people think of olive tree become materially important and epidemiologically relevant. My role is not to say if one is right or one is wrong. My role is to analyze the practices and bring something that can be taken into account. These are not just meanings, they are a way of organizing action, and action in turn reorganizes the epidemiological landscape. So to conclude, I would like to return to the question with which I began. What is anthropology doing here? My answer is that plant epidemics always occur in landscape inhabited and transformed by people. They happen in fields that are cultivated, left wild, abandoned, inherited, sold, irrigated, replanted, contested, loved, or set on fire. If we ignore these practices, then or consider them simply stakeholder afterthought, we do not make epidemiological analysis more scientific. We simply make it less complete. Plant epidemiology already depends on human practice, whether we take them seriously or not. And perhaps this is also what coexistence requires. Not simply accepting the presence of the bacteria, but learning how to live in the transformed landscape without eradication in the horizon. Thank you.
Thank you, Claudio. I ask to the next speaker, Mola di Bari, to take the floor from a European Commission Joint Research Center. She will present farmer preferences for integrated pest management strategies against Xylella fastidiosa, evidence from Apulia and Alicante.
Thank you, Chair, for introducing me. And while the presentation will be uploaded, I also want to take the chance to thank the co-authors of this study, Jesus Barrero Orle and Jose Antonio Gomez Limon. So let's see if it works. Yeah, great. Okay, so this study is on farmers' preferences for new technologies adoption to control Xylella fastidiosa. The main objective of the study was indeed to elicit and quantify the farmer preferences for integrated pest management strategies to control the bacteria in the two affected regions of Apulia and Alicante. The key research question was actually which IPM attributes do farmers value the most, and what level of public subsidy is needed for them to adopt these techniques and to achieve broad adoption. And of course, we have the comparative dimension of the two different regions and also two different cultivars, the olive trees in Apulia and the almond trees in Alicante. But the most important question is why farmer preferences? Because it doesn't matter how promising a new technology is, farmers need to be willing to adopt it. So we have listened to many interesting presentations about effective control technologies in the last 2 days, but it doesn't matter how effective is a technology. It's important to understand what are the drivers and the barriers for farmer decision making in order to then be able to design successful IPM interventions. And here we take advantage of a branch of the economics, the behavioral economics, that studies how the psychological, social, and cognitive factors can influence the decision making of a person. And here in particular, thank you very much. Yeah, I've been losing my voice in the last 2 days, apologies for that. So here in particular in this study, how farmers for preferences under uncertainties. This study has been pre-registered in the Open Science Framework platform. So what we did, we collected data in Apulia with 400 farmers and Alicante with 200 farmers through a questionnaire. We collected data on farm characteristics, Xylella awareness, knowledge, and risk perception. The discrete choice experiment was the main core of our survey, and I'm going to explain it in a moment. And finally, some socio-demographic. So the discrete choice experiment is the methodology used for this study. It's a survey-based method used to elicit preferences by asking respondents to choose between hypothetical alternatives that differs in their attributes. So since we don't have a lot of time, I will show you the practical example. So this is a choice card. Respondents in the questionnaire face 6 different choice cards. In each card, you have 3 active IPM programs. Option A focuses on the control of insect vector, option B on the increase of plant resistance, and option C of a combination of both. These techniques presented to farmers are real technologies developed and studied by the Bexil project. We also have other attributes. We show them like the reduction of chemical pesticides, the minimal threshold for collective adoption, and finally the subsidies per hectare and per year. This is the model specification. Again, I don't have time to go into details, but thanks to this model, we can analyze the observed preferences or observed heterogeneity of the farmers. We can derive the choice probability of one choice among the whole pool of choices presented to the farmer. Thanks to we interacted with some covariables, so the other questions in the survey for the 3 active IPM programs and the subsidies. And finally, most importantly, we derived the willingness to accept. So how much subsidies a farmer is willing to accept in order to implement these new IPM programs. Very quickly, some descriptive analysis. In Apulia, almost the totality of farmers are aware of Xylella, while in Alicante, 78% of the farmers are aware. We collected data in 4 different provinces in Apulia and 2 different areas in Alicante, the infected containment areas and disease-free areas. Some data about the production loss. So as you can see, in Apulia, almost half of the sample had production losses above 75%. Like in Alicante, this percentage is very small, so only 10% had such a big loss. Regarding the spending measures, the spending on Xylella measures, here again we can see how in Apulia 60%, around 60% of the farmers spend more than 100 euros per hectare to control Xylella, 15% more than 500 euros per hectare, while in Alicante again the situation is different and 60% of the sample spent less than 100 euros per hectare. On perceived risk, in Apulia, farmers at the moment feel more prepared to manage a new Xylella outbreak. And again, in Alicante, this perception is different, with the majority of the farmers feeling slightly or not at all prepared. These are the results from the multinomial logic model. As you can see, the three different active IPM programs are all significant and positive. What does it mean? That farmer prefers them over the status quo. I forgot to say that the status quo was the opt-out option, so they were able to opt out at 0 subsidies and keep with the current situation. And here the willingness to pay is indeed negative, meaning that farmers are even willing to, the willingness to accept, meaning that farmers are actually willing to pay to implement these practices, and this makes sense given the fact that them already, especially in Apulia, they spend money for already to control Xylella. The new resistant varieties, that was one of the attributes for the plant resistance, show as well a significant and positive estimate. The subsidies, of course, are positive, and the peptides are slightly less preferred than other techniques. Same in Alicante, results are very similar. One key regional difference in Alicante, farmers do not like to reduce pesticides, so they need to be actually incentivized to do that. Also in Alicante, farmers are more sensitive to subsidies, meaning that the estimate is higher, and also here the willingness to accept is less smaller. These are the interaction effects. I don't have time now to comment them, unfortunately, but please reach out if you're interested. Let's go directly to the key takeaways of this study. So there is a strong preference for active IPM programs, both in Apulia and Alicante, with the combined strategy being the most preferred. Again, in both countries, resistant varieties are preferred, and in Alicante, farmers are more sensitive to subsidies. And finally, a key regional difference is that pesticide reduction is a driver in Alicante, but not in Apulia. Thank you very much.
So thank you. Yes, have a drink because. So I ask to the next speaker, Fabio Gatti, Wageningen University, to take the floor, and he will present the conservation dilemmas, Xylella fastidiosa, and the future of futures of olive farming.
Thank you very much. How is it? Non va? Come non c'è una slide? Gliele date?
No, non ci sono le slide.
Non è possibile. Le ho consegnate.
We can solve this problem. If you have the slides, we can go to the next presenter.
Okay.
And then you can come back.
Yes,
okay?
let's do that.
So no problem. Okay, so I ask to the next presenter, Filippo Positano from European Food Safety Authority, to present the 3 years of the EU's Plant Health for Life campaign.
Thank you very much. And good morning, everyone. I'm going to present our Plant Health for Life campaign, so an EU-wide campaign on raising awareness about the risk of plant health. Yeah, let's start. So the idea of our campaign was simple. There is basically not enough public awareness about the risk on plant health and how this can be prevented. So as citizens, basically, or as travelers, for example, when we travel, we bring back plants or plant products like seeds, for example, and we don't know the risk that this can have. So our campaign was based on the social research, informing how these behaviors can affect plant health and the environment around us. And it comes from a mandate of the European Commission. Yeah, the main objective was indeed to raise awareness among different target audiences. They don't want to show up.
Okay.
Yeah. Here they are. So basically, we have identified these three target audiences: the curious traveler, the home gardener, the hobby gardener, and the conscious young parent. The curious traveler is the citizens that has a passion for plants, and when they travel, they can bring back plants just, for example, as a souvenir, but they don't know exactly that this can bring risks with it. Then our second target audience was the home gardener, so it's people who has a passion for gardening, for plants, and they can be, in a way, multiplier of our key messages. They can have an impact, let's say, on their communities by sharing good habits on plant health and yes, how you can prevent risky behaviors, let's say. And then we have identified the conscious young parents. These are parents who have, who can be, let's say, they could spread our message about plant health with children because they care about sustainability, they care about the future of their children having the environment, let's say, in their heart. What are our key messages? So we have identified three key messages, three simple messages, something that people, citizens can relate with, and they can be actionable, so something simple. Everyday relevance, so how can plant health, our small habits, simple actions, how this can be, they can affect our environment, as simple as, for example, not bringing back plant souvenirs when traveling outside of the European Union. Our second key message was about responsibility, so how all of us, citizens, as parents, as travelers, we have a role. So everyone with their actions can have a role, so we have a shared responsibility. And finally, preventive actions, so how these simple actions can protect plants, they can protect diseases from spreading. Our campaign started in 2023, and it started with 12 countries participating. And now, 2026, this is our last year, the last year of our campaign, and we grew to 33 countries from Europe, as you can see, and from the European Union, let's say, and beyond, so to Europe and accession countries. So it's a very interesting, let's say, the value was indeed in this growth and in the network that was created between member states and between also well, the authorities, let's say, in the national member states. Our approach evolved from 2023, 2024 in the beginning with the basics, let's say, of our communication campaign. So by raising awareness about our campaign, about our key messages, how everyone can contribute. And it developed to advocacy. So last year was a year of advocacy. So we tried to mobilize the citizens, schools, and networks to spread our messages. And this year is a year of consolidations where we are reaping the benefits, let's say the fruits of the campaign. To give you an overview of the activities of last year, we had more than 700 hits on media hits, so appearances on TV, radio, in national TVs, more than 50 events in person, meeting people and communities. You can see schools, for example, or other networks that we have activated. On social media, we had more than 1 million of users reached through content creators, and quite impactful was also the out-of-home advertising, so the messages that we have posted in airports, for example, so in key travel hubs in Milan or Amsterdam airports, for example, but also on streets, like strategic points on streets. And what comes next? The impact, as I was saying, was not only measured in a quantitative form, like in the sense of the reach, social media, out-of-home advertising, but also in terms of quality. So we had focus groups, surveys to understand if our messages resonated, so if our public and audiences were finding our messages clear and appealing, which was the case, and more than 77%, so it's quite a good, I would say, part of audiences recognized the key messages. And our campaign recognition grew from 2023 to this year, to 2025, to almost 40%. So this, as I was saying, this is our last year, so it's the last year of the campaign, but a lot remains in terms of campaign assets, so a lot of material that was made available and translated in national languages. It can be coloring books for children, but also a number of visuals and assets that can be reused. Also, the results of our campaign can inform future research, also in the future activities on public awareness on plant health, so the insights can be useful. A very important part, as I was saying, was the collaborations that were established through these years among countries, member states, among authorities, associations. So I think this is a very important heritage of this campaign. And looking to the future, the collaboration with partners is something that will stay and will help us build on future activities. Thank you very much.
Thank you for the contribution. And so if Fabio Gatti is ready, please have the floor. Yeah, I know. Your presentation is ready.
Okay, sorry for the delay and the inconvenience. I hope I keep my 10 minutes. Yes. Okay, thank you very much. I'm Fabio Gatti. I'm currently finalizing my PhD at the University of Wageningen in a group called Knowledge, Technology and Innovation, and I'm currently also on a temporary appointment in the Sociology and Anthropology department of the University of Mons. And the story I'm going to tell you today is titled Conservation Dilemmas, Xylella fastidiosa and the Futures of Olive Farming. Let's see. Yes. Okay, so I guess most of you know this, but just to set the scene a little bit, the importance of olive trees in Apulia is very strong. We have 60 million olive trees out of 4 million inhabitants, so there's about 15 olive trees per person. We produce almost half of the olive oil of the whole country, and then olive farming is on top of the economic importance. It has also a strong identitarian and cultural value, let's say. Okay, we are also the first region where Xylella fastidiosa was detected in the European Union, and where the eradication measures were implemented, or attempted to be implemented at least. And I also want to stress that as we have heard before, that the crisis of Xylella is caused a structural crisis, but what I want to stress is that, in fact, the olive sector was already in structural crisis when Xylella fastidiosa arrived. So I'm not going to go into detail, but I have one paper in which I talk of how the structural crisis, to some extent, was a co-factor in the development of the outbreak. As a brief historical background, this is a heated story because at the very beginning, when eradication were implemented, there was a big reaction by local populations that basically disagreed on this measure and tried to stop eradications and so on. And this is also a story of distrust towards science. Some scientists, and some of them are even in this room, were under investigation, were pressed charges, and I'm really sorry for them, but this is part of the historical evolution of this epidemic here. But it's also a story of saving trees. Claudio mentioned this before. Basically, since the 2015, 2016, everybody wanted to save olive trees in Italy. Okay, you can find Rodrigo Almeida wrote this famous perspective on science: can Apulia's olive trees be saved? And many other organizations, journals, and so on framed this attempt to intervene in the epidemic in terms of saving, okay? And that's why I call it conservation dilemmas. And when I think about saving, I always think about this quote that I want to read with you. This is from another PhD thesis from anthropologist Heather Swanson, and she talks about salmons, okay? She doesn't talk about olives. But I think if we read this together, you can have a sense and a feeling of what happened also in Puglia. So Heather Swanson says, in the 90s, just as I was becoming aware of my social, political, ecological surroundings, salmon runs hit all-time lows and fishing seasons were cancelled. The situation was so dire that several salmon populations hovered on the brink of extinction. Soon, several kinds of salmons, once our town staple, were listed under the Federal Endangered Species Act. It is hard to capture the sense of emergency that these fish declines provoke. Salmon were not just an economic resource, they were the stuff of our lives. Hardly a day passed when the town's newspaper did not have a story about the waning number of the fish. Although almost everyone agreed that we needed to save the salmon in order to revitalize our region, no one could agree on what to do. A big part of the problem, we soon realized, was that there was no consensus about what should be saved and no consensus about what counted as a salmon. So the main take-home message from this presentation is that in Apulia, everybody wants to save the olive trees, yet what is to be saved is not clear. And what I found from my research and from my fieldwork was that, in fact, we have different trees. The olive tree in Apulia is not the same entity. Depending on the stakeholders and the actors you talk to, you will encounter different olive trees. Okay? One is the productive olive tree, so the olive tree as a production asset that creates income and sustains livelihoods. The other olive tree is the symbolic and emotional tree, okay? The one that is connected with family memories and the one that is not strictly related to a productive role, but to some extent still sustained like self-consumption, okay? And then there is a third tree that is growing, I think, also because of tourism development in the region lately, and is the aesthetic tree, the landscape value, or the tree that creates landscape value, okay? And this tree, again, is not strictly related to a productive component, but indirectly can sustain incomes because it sustains tourism and so on. So basically, like, my point is, before asking can Apulian olive trees be saved, we should ask which olive tree we want to save in Apulia. And then, also importantly, we have to define who is this we, okay? And basically, what I call a conservation dilemma is this structural impossibility to sustain at the same time and with the same policy instrument the olive tree as an income-sustaining entity and the traditional olive tree, the one that is attached to memories and so on. So, of course, to each of these three types of olive trees, there are different practices associated with it, so, and policies, in fact. So on the one side, the olive tree that is the productive one, the tendency is to replant intensive or high-density plantations. Of course, the subsidies go very much in that direction as well, so the politics supports that as a sectoral modernization and so on. So to some extent, this restructuring is for competitiveness and is to save olive farming as a sector rather than olive farming, the individual olive tree. Then you have the landscape value, you have the grafting, that is the practice that is used to preserve the monumental olive trees, the ancient olive trees. And this is a technique that works, but is very costly, okay? And with respect to replanting, it still received some subsidies from the region, but a little bit less, and still, remains a costly intervention. And if you talk with grafters, you also learn that it's impossible to graft all the territory of Apulia, okay? There is just not enough grafters and not enough means to sustain this intervention. And then there is a wide variety, the galaxy of like coexistence protocols that biostimulants, some foliar treatments with partial antibacterial effect, and so on. Again, they don't receive subsidies, basically, and they are also costly. And in a sector that is already under structural stress, basically, all these interventions, with the exception maybe of the replanting, are made by wealthy owners, okay? Are made by people that have an extra income and that they don't rely on farming for their living. So basically, yes, this is what I just said. So the more a set of practices goes in the direction of attempting to preserve traditional landscapes, the more complicated it becomes to sustain livelihoods, to generate income with it. And at the end, policy strongly favors intensive replanting and grafting and coexistence protocols. Some of those have shown some effect, some partial effect, receive less or no public support. So some key questions, I throw them there. Even if treatment or grafting work, they have a very high cost, so more political questions arise. So who will be able or should be able to sustain that cost? Then a corollary question arises, should this low productive traditional landscape be saved at all? And if we answer yes to this question, then we have to start seeing them as cultural heritage, not as productive assets, because the market will never sustain low productivity traditional orchards, okay? Ultimately, the question becomes, what future do we imagine for olive farming in Apulia? More of the same, olive monoculture, more intensive, more controlled, and more managed, or rather something else? And the last slide, it's a little bit broader, and I wanted to take the opportunity to speak in front of this kind of audience, to be the kind of representative of the social scientists here. So I want to make some final remarks. One is that the olive quick decline syndrome is not only a phytopathology, but as my friend Christian Colella calls it, is a socio-phytopathology, and that means that in order to tackle this, we need much more sustained collaborations between social scientists and natural scientists. We as social scientists need to tell better stories that foster dialogue rather than reinforce and exacerbate polarizations. As natural scientists, I think I encourage you to abandon this ideal of the neutrality of science, because I think science, as all social endeavors, is political, both in the effects on society and in the dynamics that shape its production. And technical responses to Xylella fastidiosa are necessary, but not sufficient, because the long-term fate of Apulian olive groves depends on the social, political, and economic conditions that determine which coexistence practices can be materially and financially sustained. If you want to read more, this is a self-promotion slide with the publication that came out of my PhD thesis so far, and thank you very much.
Thank you. So we have now two pitch talks, and we start with the first one by Livia Maltese, University of Bonn. mapping research gaps on Xylella fastidiosa toward a socio-ecological perspective for Apulia. Thank you.
Thank you. Good morning, everyone. It's almost lunchtime, so you just have to endure a bit longer. So to give you a bit of introduction, I have a background in biotechnology and ecology. And what I'm now doing is a PhD, an interdisciplinary PhD combining social sciences and ecology at the University of Mons and Wageningen. I started my PhD last October, so what I will present today is the result of my literature review that I did in the past month. So I guess you're all familiar with these haunting pictures, just some of the many showing the devastation that has been happening in century-old olive trees, olive groves in Salento. Since the beginning of this tragedy, an extraordinary amount of effort has been mobilized against it. So the beginning of my research was focused on asking which kind of questions have been asked until now and which questions have not been asked yet. We often refer to Xylella fastidiosa as an ecological disaster. So my question as an ecologist was how much studies have actually focused on ecology as a field of study. So to answer this question, I performed a systematic literature analysis. I gathered all the papers that have been written on Xylella fastidiosa in Salento. I categorized them according to their research question and according to the field of study. And I came up with 250 papers since 2013. The results are quite clear. So as we can see, 42% of literature focused on pathology, pathogen genomics, and vector ecology, the light blue slivers in the pie chart. And that is what can be defined the host pathogen vector triangle of interactions. Another 39% focused on monitoring, diagnostics, management, and control methods. And 8% focused on the social and economic impact of the disease. So to go back to my question, how many studies have actually focused on ecology? The answer is 6%. But why does this matter? Well, because as was said before, in places like Puglia, the landscape, it's the result of dynamics of interaction between social and ecological systems. So a socio-ecological perspective contextualizes the question, how do we deal with the bacterium, with another question, which is which kind of landscapes and society allow the bacterium to thrive. So to have a truly integrated response, in my perspective, it's really important to combine the very important epidemiological and management studies with a study focusing on socio-ecological condition. That's what I will try and do in my PhD, or at least in part. Since ecology looks at the interaction within organisms and their abiotic system, I will perform social and ecological fieldwork to understand not only how olive trees' health is impacted by Xylella, all of you are very busy with that, but also looking at how management strategies, soil conditions, biodiversity around the olive groves and climate have an impact on the tree. To do so, I'm of course open to any kind of feedback and collaboration, so please visit my poster, number 112, and come and talk to me. Thank you.
Thank you. So the last talk is a pitch talk by Harry Marshall from Forestry Commission Research Agency, UK, and he will present the social dimensions of managing Xylella fastidiosa, insights from stakeholders.
Hello, can you hear me? Can you hear me now? Yeah, cool. Hi everyone. Yes, so I'm Harry Marshall. I'm a social scientist from Forest Research in the UK. But today I'm going to be talking about research that we've done in the EU as part of the Bexel project. Specifically looking at social dimensions of managing Xylella fastidiosa, looking at insights from stakeholders. And so I'm very fortunate to be last in this session because I get to stand on the shoulders of the ones who've gone before me. Oh, sorry. That's all right. Is that better? Okay. So, sorry? Oh yeah, okay, let's try this. So, I don't need to tell you that Xylella is obviously one of the most impactful plant pathogens there is out there. But I guess, as kind of has been hinted already, it's impactful in many ways. It's not just economically or ecologically, but it's also socially and culturally. And so it's also, it has already impacted many parts of the EU, but there are still many parts which it hasn't reached yet. So what we tried to do was to look at areas where both stakeholders have been affected by Xylella, but also areas where Xylella hasn't reached yet, to understand essentially how people's perspectives, what their mental models might look like, and how they interact with both the disease, but also our management responses to the disease, and what this has in kind of implications for preparedness. So we took a kind of qualitative comparative approach, looking across different epidemiological stages, carrying out research in Cordoba, Alicante, and Salento. So an area where Xylella is not present, where it's found in olives, and where it's affecting olive trees, almonds, and where it's affecting olive trees in Salento. And kind of we were interested in that difference between the different stages, so monitoring, prevention stages versus eradication and containment. And so we did this, carried out research over the last couple of years. We did focus groups and interviews with different stakeholders, relied a lot on the support of the Bexel partners, so thank you very much for all of that. And so our kind of sample size is on the screen there, about 50 across the different locations. And so what we found were three distinct perspectives. In Cordoba, the olive farmers were anticipating the potential impacts of Xylella. There wasn't kind of a wholesale understanding of Xylella, and some people were frustrated that they didn't know more about it already and authorities weren't alerting them. In Alicante, where it's very much in the thick of dealing with the eradication of Xylella on almond trees, there's kind of people are struggling to imagine a future, how they're going to continue to practice their agriculture, particularly with almond trees. And in Salento, in the area of the people we spoke to, there was some sort of, maybe not optimism, but people were looking forward and maybe looking to kind of start coexisting with Xylella. But kind of thinking about Fabio's results there, maybe there still needs to be a discussion about what kind of future we're imagining. So kind of overall, what my work as well as the other social sessions which have gone before me kind of suggest that a one-size-fits-all approach won't work. In each different social political context, the way that we respond to Xylella and managing it needs to be different and needs to take into account the social and cultural impacts. And the preparedness framework should integrate these social insights as early on as possible and include stakeholders at every stage if possible. And so that's what we're doing in the Bexel project, working to kind of co-produce some of these IPM solutions, working with various parts of that consortium. And yeah, that's me. So thank you very much, everyone. Thanks to all my collaborators, to the co-investigators, and the conference for having me.
Okay, perfect. Now, can you hear me? Yeah. No? Yeah. Someone? Yeah. Now, yes. Now for the Q&A, a few things. First, if you could introduce yourself at the beginning so that we know who is asking the question. And second, we had a request from the tech team to not tap on the mic. They're losing their eardrum, so be nice to them. Thank you. Yeah. Floor is open for anybody.
Okay, thank you, and sorry for the several questions. I have thanks to Gatti for the three categorization of professional, landscape, and family. Oh, so, what happens throughout the time? So, how was the weight of these three categories in affecting the political choices or something like this? and if we should expect again a race of some of them in the future. This is the first question. The second question is, me as a scientist, I rely on science. But now a positivist approach, so the science will save our lives, is being very, very difficult to manage. Artificial intelligence, all these stories, epidemic. So what do you think about this, and what is your role to mediate in this history, the dilemma between how to rely on science and mediate with the society?
Thanks. Complicated questions, but interesting ones. How these three different types are evolving or have been evolving, I'm not sure. I know that, for example, in Salento, there is very few trees of the family type or hobby type that are still productive. So I know that that kind of position, for example, faded out quite a bit, but because of material constraints, impossibility to keep those trees alive or productive. Although now there is this whole debate about these trees that are resprouting and so on, and of course, from a productive perspective, that is not an interesting phenomenon. They will never reach the levels of productivity that they had before, and it's irrelevant from a productive perspective, I think. But some people use it as an argument that, those trees can still be saved. I don't know, I think it's a very dynamic and evolving situation also because now the symptomatology is spreading north, and therefore the more the symptomatology becomes severe, and therefore also the intensity of the interventions of this intensified care that you have to apply to the trees becomes too costly, then that type of tree or that type of practices will just fade out, I think. I don't know if I answered to your question precisely, but regarding science, that's a very interesting question, I think. Of course, I come from social studies of science, in which, let's say, the positivist vision of science as the domain of knowledge that produces truth, it's hardly criticized, let's say. It's hardly questioned, Science is always situated and is always as a social phenomena, and as all social phenomena is basically is not objective and is not neutral and is not speaking to truth, but at the same time it has some value because it creates knowledge based on empirical data and on something that has to, I'm not saying that we have to go fully to the post-structuralist or post-constructivist side. I think data and empirical data, quantitative data are extremely important, and I think social scientists have to take that into account fully. At the same time, I think science is not a bulk homogeneous. It's not a bulk homogeneous object. Within science, there are internal differentiations and there is diversity. So we have to acknowledge that within science, there are scientific communities that have different perspectives, different disciplinary trainings, different positionings, and that struggle to gain hegemonic positions within the landscape. And I think that's a fact.
I think there's a question from Donato, then we'll go one by one for now.
Yeah, and let's try to keep the answer a little short if we can. No, it's fine. There's many questions.
So since you asked us to introduce ourselves, I'm one of those that have been object of investigation by the prosecutor, as mentioned before by Fabio.
No, anymore.
And Fabio, you do not have to be sorry for that. It has been an interesting experience, I have to say. But my question is to Professor Miglietta, because I'm sorry if you already reported this data, but maybe I missed. I want to ask you if in the amount of olives that are processed every year, as you show there, if you know how much of that are olives produced not in the province of Lecce, but imported from bordering provinces, still Xylella free.
Funziona. Aspetta un secondo.
Okay, yes. So the data are related to only the olives processed, but deriving from the territory of Salento. So even the not on account olives, the purchased ones, derives from the territory. For this reason, we were able to make this kind of analysis, statistical analysis. And there is this correspondence between the replanting activities. This is the most important thing, that the replanting activities is helping in 3-4 years the renewal phase of the olive mill activities. So with the, in particular with the Favolosa cultivar, we are experiencing a new growth in the production of olive oil. This is the results of the statistical analysis. So the replanting are certainly from the Salento area, and also the data produced, the data of the olives processed are from the same area. So there is no doubt about this.
Qua davanti, per favore, il microfono.
Down there is another person since.
Ok. No, era
Yeah. Thank you, every speaker. I am Giacomo Giogannoccaro from the University of Bari. I'm an economist, and I like these sections opening the social science within a hard science floor. So I have several questions, but just to be quickly, to Miglietta Pierpaolo, just to think about replanting, if there are irrigated or not irrigated systems, because the topic now is we are escaping a tragedy and we are opening a new tragedy. This is the groundwater over exploitations, and the new system that is so dependent on the water. And then we have the recent experience in Spain where 2 years drought has put, let's say, a very struggle for production. What do you think about?
Yes, I know that, for example, the new replanting are water intensive, but as one of my colleagues said, we cannot just move from a problem to another. Certainly, the problem of water is a problem, and I think that we can solve this through, for example, water-saving activities or water collection from rainwater. There are some experiences in the Salento area. There are some farmers that are already collecting water from rainwater, and they have a lot of opportunities to use this rainwater to irrigate in different times. So we can collect rainwater in the time in which, for example, we have availability, and then use in a second time when there is drought period. The water is another problem apart from the choice of the cultivar selection, I think.
All right, there's a question up in the front, and then we're going behind, and then on this side, sorry, I will get to you. Up in the front, if possible. Can you bring the microphone up to the front, please? Thank you.
Thank you. A question to Dr. Gatti. Here in the area we have a lot of centennial plants, and you showed very well how we have a lot of centennial plants here in the area. So one of the ways to save them is grafting. But policymakers may know how much it costs, because it is costly, it's different. You give some numbers. So according to you, to preserve those plants that now are here, but in maybe 10, 15 years, they may disappear. So a way to save them, how much it costs per plant?
I mean, the numbers that I know from grafters, is that the intervention costs 250 euros, more or less. If you find someone that does it cheaper, maybe 200, but it's better to rely on someone that really knows what they are doing. Plus, you need this extra 100, 150 euros for the post-grafting management, because it actually requires quite intensive post-intervention practices, because you have to remove all these shoots because once you remove all the branches, basically like the tree explodes for the coming 1 or 2 or even 3 years. So you have to go and remove those shooters. So I would say just for the grafting and for the managing the reaction of the plant, 400 euros per plant.
Thank you.
Thank you. Julia from Agroscope. So I wanted to ask to all of you, maybe just out of curiosity, if you have ever, for example, checked how is the media or the scientists' communication towards the topic shaping, for example, the response of the society against one of these problems, like in this case, Xylella. If you have, for example, compared it with other plant pests that maybe are not that known in the media, no? So I wanted to know if you have any information.
Do you have a question for someone in particular that you want to hear from? No. Maybe people work with stakeholder responses? No, they are.
Is that one? Yes, that's good. Cool. So I'll admit I haven't looked into that. However, I know some people are looking into kind of misinformation and disinformation and how people react to that and how people engage with that. I think that's colleagues. I think it might even be Claudio and Donato. But I think that it's really important. And for example, it came up when I talked about Cordoba and how people are frustrated about the lack of information being provided, and how that they'd like more information, which is quite interesting because usually people don't want to hear about something that's going to threaten their business, I guess.
Yeah, maybe I can respond a bit. So just to give you a sense, for me, my research, now I've been here for about a year and a half, doing fieldwork, extensive ethnographic fieldwork with different groups. And one of the things that's been really kind of incredible is the amount of work that people like Salvatore or Donato do in public discussions. So my sense is that there was a moment a few years back when this was underestimated, heavily underestimated, and it changed quite profoundly. So there is an enormous outreach of information, whether it's through media or direct contact. But of course, as we were saying, the presence of alternative models means also the presence of alternative networks of information. Right? So often part of what is happening is that people are receiving different type of information. and often information that can be very available in a way when you are experiencing such a loss, right? So for instance, if I tell you there is no cure for Xylella, you receive the information differently than if I say to you, look outside, the trees are vegetating again, right? So it's not just about the source of information, but it's also the ways in which information are there understood in this type of relations. So part of the work that I think many people here are doing, it's precisely to do that, right? And I think this is also where us as social scientists can come into the picture, because we go out and talk to people, you know, so you can do that kind of work. But I do think that, like, thinking quite seriously about media circulation, not just as what do we produce, because I think there's a lot of discussion now about what does this side produce as media, but also what media are people receiving. It's less actually studied. Like, where are people getting information? Like a YouTube post from EFSA is done magnificently, but the average farmer is not going on the YouTube EFSA channel to listen to this, you know? So I think that question is a profound one. So, Laura, do you mind just...
No,
it's a response, okay. Okay, do you mind passing it for a second behind? Yeah. And then we go on that side and then to Giuseppe,
Thank
sorry.
you. First of all, congratulations for this session. It was really interesting. I have a question for you. So I think that the three models that you presented are really interesting, and it is really what is happening in Salento. But you didn't speak about the spread of those three models. I think that it is important also to consider how much they are impacting the territory, because mostly of the territory is abandoned. And also, I want to know if you think also to consider the impact of each models on the other models, because the first model really strongly impact the third one and also the second one. So I think that also those part should be taken into account, because the fire especially is really a bad problem.
Yeah, no, I think that's a fantastic question. So let me start from the extension and assessment part. So we're dealing with, in these three models, we're dealing with very, very, very different set of data sets, right? So for the reimplanting, the same data that was presented before is data that was given to us by the Osservatorio, the Phytopathological Observatory. Why they have all this data is that every single reimplanting that receive any funding, this does not cover all reimplanting, but reimplanting with some funding has to be collaudato. We use this weird term like, certified, but it's a strange term in Italian why we use collaudato. Anyway, that's a different discussion. So for that we have data. We have data of extension, we have data of location, and we have data of varieties. So that's quite easy. And currently, as I was saying, there's about 3 million something trees. These are not the number of trees planted. This is the number of things that were taken out and replanted. So the planted is actually higher, because the collaudo is based on what you displant. So for that, we have data. For abandonment, and this is where I think the region needs to step up their game a bit, there is very, very little data available. The only data set I have that I'm using is done autonomously by the Comune of Ugento, who did a map of abandonment. And Ugento is one of the most replanting area that you show, so it's really much skewed on the opposite side, and it's 40% of the territory that has been abandoned. And this is maybe the highest town of replanting in the entire Salento. So the assessment is much higher, but it's hard to find number. And the consequences, the interaction between the model is massive. As you know, tomorrow many of you will go down and you'll see there'll be fires in your drive, 100%. And so this is part of the question, like to try to bring up these models is to also invite people who do policy to say, hey, think about the interaction between them, because this is the first question that was asked to Fabio, none of these models will take the entire landscape, because of water requirement, because of cost. This is not, like the landscape of the future will not look like a monoculture of any of these models, but there will always be a composition. So to think through that question, I think is very important. Sorry, I took a long time. No, there's questions on this side, and then Giuseppe.
Hi, everybody. Carlo Donadio, BG Agro. I think that researchers play a pivotal role in this domain. Not coming from your academic background, I'll try to provide, trying to ask a different question, or trying to depict a scenario that is slightly different from the researcher. I liked a lot of what Dr. Gatti said regarding the neutrality of the scientists. Neutrality is something that works a lot on paper, but in reality, it shouldn't be. Or trying to be in my comfort zone, I will provide, I'll ask you a crispy and direct question: how the researcher role should evolve, and if this role should evolve in this domain, as the three different type of consideration, trees, olive oil, and olives are an asset, a touristic asset, an asset for production, is something related to our heritage. I think that the focus now should be not only on the scientific pure contrast to the phytopathology, but trying to see what will be better located the funding on the producer, and this is quite critical and maybe a little bit harsh, on the transformers from olive oils to from olives to oil, or even, that's very bad, on the commercial side. Who should be funded more? Because if you fund the commercial side, you will increase the demand of olives, and maybe there will be a different view on how to restart the production. If you go into a transformer, you will work on a different, you will fund somebody that will support somehow the producer. If you fund only the producer, you may have a singular, we go only to Lecino maybe. Is it what we wanted? Is this the right view? And again, just to retake the question from the beginning, how the researcher role should evolve in this domain? Thank you.
Yeah, thanks a lot for the question. I think it's a very relevant one. I mean, I think it's an economic question. It's not, it doesn't have to do with the role of researchers, honestly. Like, I don't see how, because I think, so ultimately, my message would be, to save the olive trees, you need to make olive production economically sustainable and economically profitable, okay? So whether you manage to pay pay the producers of 1 liter of olive oil from traditional orchard 25 euros per liter, either you make it sustainable because you intensify, whatever, it has to be economically sustainable. And the fact that 3-4 years ago there was this like spike in prices of olive oil reflected on much more attention to management practices all over the region. This ethnographically you could see it. So when olive oil prices are worth it, people farm olives and farm olive trees and even spend more money to do all this sort of management practices and so on. So I think it's a policy question.
So we have 2 last questions. Let's take both questions and then we respond, and then please find us outside. And Giuseppe first, if you don't mind.
Okay. So my question is mostly for Filippo Positano, because I think I agree with some of the discussion that the replanting, the planting of the landscape need a very deep economic analysis. So you need to think carefully how you replant, look at the resource, the water, a lot of components. Now it's difficult to address all in the discussion, but I'm thinking about new invasions. So for the first time now in Europe, we have a European campaign on the awareness of plant health. There was nothing before. So how do you think this campaign can help for future invasion, for people to understand the risk of the plant pest? That the discussion is more about solution and not about different point of view, for example.
Let's get the other question that we respond together.
Okay. This is Massimiliano Morelli from CNR Bari. I have a question for Claudio. You properly mentioned the involvement of anthropic component in this multidimensional triangle, let's say. And usually for other species, involvement is forced because they are forced by natural laws or survival needs. But in the case of human beings, this is usually the consequence of a choice. Probably now, for several people, is not more a choice, is an obliged choice to be involved. But this was not the same since the beginning of this story. Have you ever had in your study the impression that in this case we have observed a kind of abnormal involvement, abnormal choice to be involved? And if this, in your feeling, may have caused, may have shaped the destiny of this story somehow. Thank you.
Yes, thank you, Giuseppe. Very good point. I think that this was, I mean, indeed, first in Europe, public awareness was very low, and I mean, colleagues, scientists, I am a PR practitioner, let's say, know well that awareness was very, very low, and it continues to be low about public risks. So the collaboration that we have established with member countries and between stakeholders in Europe and beyond is a good basis, I think, to get the conversation started at least, and in a way be prepared for the future. So preparation is a really key component here. Thank you.
All right, super quickly because people want to have lunch. I mean, yes, absolutely. I think Giovanni just left. I think Giovanni is an example of this. Is there. I mean, again, this might be one part of my work that makes this audience roll their eyes, but I do think that part of what is happening, and a way of understanding what is happening, is trying to understand what a giant ecological loss is. Every single person, even the people who are more thinking about production, like Giovanni, because he's a producer and he's an entrepreneur, has an obsession with replanting, an obsession with continuing to put trees. I mean, talk to Giovanni and you will feel it, right? So my sense is that we don't have a good language to think about and deal with giant ecological losses. And this is an experience that we as a species will have in the next, this is why I'm doing this work. We will have this experience of ecological losses everywhere, but we don't have form of collectivizing these losses, right? We have like a relative of us die, we have a funeral, which is a form of collectivization. Here is not available. I was having a conversation with this old lady who told me the first time she saw these big trees being carried out, she says it was a funeral without, what's in English word? Without the people behind it, without the procession, right? And so I think there are a lot of behavior that can actually be understood collectively as this. Similarly, the people that we call conspiracy theorists, this is an obsession with finding causes, right? Like ultimately what they're doing is putting all their attention to believe that there is one person or one group of people who did this, right? And so to me, I think there's a lot of this type of behavior, and some of it is productive, some is not. But in order to tell the full story, I think this needs to be told as well. And I know as people who work in lab, you might be a little reacting to the sense of grief, but I think we need to think about this question, and we need to, I mean, one of the variety that Giovanni is considering as a name is ossessione. And I think it's worth asking him why. Anyway, have a good lunch. Thank you for listening.
Yes. Okay, so we have to wait. It's 1 minute.
Yeah, no, but
So,
I think they were a bit late with the previous session,
okay,
so we can, a couple of minutes more we can. Also because there is no many people in the room now.
And it's working. It's
Yeah.
working. Oh, yes.
Yes.
So, you start with you start with yes, and I take the notes. Okay, it's clear. It's clear for me. Yeah.
A couple of minutes, you said.
Yeah. Maybe it's not. Okay, we cannot switch. Okay.
Okay, good.
Session 6.
Yeah, 4:30. Yeah.
It's going well.
No, it's going well, very well.
Okay.
Yeah.
You have a lot of speakers.
Yeah. Okay. Wow. Oh, we have a photograph. Yeah.
So 1 more minute and we start.
Okay.
Last 2 minutes of courtesy.
Okay.
Because also they were late with the previous sessions.
Okay. But we have lost many people.
Yeah.
This is the siesta time.
Well, people is chatting, these things, and the surveillance is maybe not the most hot topic in the session in the Congress.
Alors, non, alors. Ah, okay.
Okay, so welcome back. We hope you enjoy lunch and hopefully you are not too sleepy now. For today we had coffee, so that's good for our session. This session is about monitoring and modeling Xylella fastidiosa epidemics. Is the first part, is number 1, because after the coffee break there will be another session on the same topic. And this session will be chaired by Valérie Olivier from ANSES and myself, I'm Antonio Vicent from IBIA. So because we know that you are a bit sleepy after the lunch, we choose our most strong, provocating speaker, that is Sybren Vos from the European Food Safety Authority. He is going to talk about harmonization of Xylella fastidiosa surveys in the European Union. So Sybren, the floor is yours, and the challenge to keep people fully awake. Yeah, it takes is all the morning like this. Just push it and wait a bit. It will come.
Yes, good afternoon. Somebody is sleepy or everybody took coffee?
And the micro. Sybren, you are told you have to...
Hello. Do you hear me? Yeah. Okay, so let's start. Oh, the clock has started. So I have a lot of things to say, but I have little time. So I will try to filter down. Yeah. So I will give you a small talk about what EFSA is doing to try to harmonize, to contribute to harmonization of the surveillance activities in the EU. It's not particularly about Xylella fastidiosa. Our work is covering all the quarantine pests that are of concern for the European Union. So we all know that harmonization in the context of the EU is very difficult. We have different climate zones,
We have so many different administrative organizations. We have different expertise. It's a very complex topic to harmonize surveillance in the EU. It's a big challenge. And therefore, we need to have common rules. We need to have common framework. We need to have common tools. And behind all this, we need to train the people that are doing the surveillance and train more. So this is basically the scope of this presentation. So in terms of rules, we are well covered by a strong legislative framework where we have the plant health law, first of all, that indicates the general requirements for surveillance that are very important. And based on those general requirements, we have developed our methodological framework. So basically, it says that every survey should be risk-based and scientific sound. It says that there are reporting obligations. For priority, pests should be annual, etc. It should be done with high degrees of confidence, and it should allow timely detection of the quarantine pest. And then we have specific rules that are common for all. And that framework, legislative framework, allows already to harmonize a big part of the process of surveillance. Then we developed in this context, following mandate from European Commission, an entire toolkit. And that toolkit aims at helping the member states to perform surveys in a rational, in a scientific manner. Basically, we want the member states to have a rationale behind how much they have to survey, how much effort they have to conduct to be able to find the pest, or to not to find, but to be able to declare pest freedom or substantiate freedom of a pest. And this toolkit is developed to allow for comparable surveys in time and in space. Now it's better, eh? I don't need to shout anymore. Okay, so these tools, the underpinning methodology behind these tools is a very simple methodology. It's based on science, it's based on distributions that are very well known in the sampling environment. So what we are doing is that we are implementing this mathematics, this statistics in a framework that we train the member states on using. Of course, to run this methodological framework and the tools, we need to inform well our surveillance with pest survey parameters. So we need to clearly tailor the surveys to the different parameters that are specific for the pests and for the areas where the surveys are conducted. I will just give you a few examples. In the time that is allocated, I can just give you a few examples of this. So in terms of pest survey parameters, there is the first one that's very important, is the spread capacity of a pest. So the spread capacity of a pest, we are in the situation of gaps of information, etc. We try to estimate these values through expert knowledge elicitation methodologies, where we put a certain number of experts around the table, and we try to capture the belief of those experts in the form of an uncertainty distribution. And for Xylella fastidiosa, the last exercise was performed in the end of last year, and we came with a median value of 2 kilometers per year as the capacity of the pest to spread, the maximum capacity of the pest to spread in natural conditions. Of course, this information is dynamic, has to be updated continuously, and new knowledge appears. Ongoing research is also feeding us to revise these values, and we have the EFSA risk assessment for Xylella fastidiosa that will be delivered by the end of the year that will also help us to adjust this information. This spread information, we process it because what we want to help is the member states to survey where the probability of having the pest is higher, meaning we try to support the member states in risk-based surveys. And for doing so, we are using an exponential kernel distribution model that helps us to clearly characterize the risk areas that are around the places that we assume that the pest could be introduced. And for this, there is a new tool available that is called the Risker. If you have interest, just come to see me after this presentation. Other survey parameters that are very relevant is the detection methods. We need to have an estimation of how well the detection methods in the field are performing. To gather that information is very complex. It's a data gap again. And I invite you all to do a lot of research on this topic because we really have gaps there on estimating the performance of visual examination and of asymptomatic sampling, etc. So basically, the questions that we are asking, and we try to resolve this with estimates coming from expert knowledge elicitations again, and the questions we are asking in particular about asymptomatic sampling is, given that the plant is infested by a pest or by Xylella, what is the probability that the asymptomatic sample will also be?
Sorry, Siren. Okay, he's back, he's back.
You give me one more minute. What is the probability that the asymptomatic sample will really be infested? So this is the sort of questions that we want to ask to a panel of experts that are experts in the detection methods, in the sampling, etc., to be able to give an estimation of that performance. And that performance value is essential for calibrating and for being able to do a proper sampling scheme. For Xylella fastidiosa, these ECEs on these different detection methods are planned by the end of 2026. So I will go very fast in the conclusion. So we have a toolkit. It works. It's applied by member states. We are training the member states. We have a lot of capacity building activities, but it should be a dynamic toolkit. So this toolkit, we don't want it to be outdated and to be just a calculator. We want to help to bridge the research with the application in the field, meaning the research results, the new knowledge that is generated by the research sector, by the risk assessment, etc. We need to integrate that fully in our toolkit to help the member states in saving resources in their activity of surveillance. So basically this bridging, and we have heard a lot of research going on, a lot of, and we will hear a lot of subjects dealing with surveillance. It's important that we have at a certain moment this bridge created. And we are now going to focus in particular on pre-detection methods to help early detection, in particular remote sensing, how to integrate remote sensing, eDNA analysis in the toolkit, and we want to focus as well on inventories of trapping to help member states in choosing the best traps for union quarantine pests. For Xylella fastidiosa, we will have to update the whole toolkit and the guidelines following results of research and following risk assessment activities ongoing now. And that's what I wanted to say, 0-0.
Thank you very much, Ibrahim, for keeping the time. Thank you.
Next talk would be with Donato Boscia from IPSP Siena and Siam of Bari.
Okay.
And
no, please.
Database of 13 years of monitoring in Apulia, over 1,500,000 of analysis, a powerful tool for the study of the epidemic of Xylella.
Thank you. What I'm going to try to do now is to give you some key information regarding the large database of produced in the framework of monitoring program of the Apulia region in these last 12 years since the first discovery of the first outbreak. So it's of course a database that has been made by the Regional Plant Protection Service. So as you can see here, the Salvatore Infantino is the first author of this talk. So what happened? That soon afterward, the discovery in 2013 of the first European outbreak of Xylella, the regional phytosanitary authority has implemented a huge monitoring program, still ongoing, to support the surveillance and, where appropriate, action of eradication of containment. So it is, since 2013, through the end of last year, a total of over 1.5 million of plants have been sampled and submitted to laboratory analysis. We believe that this is an effort that is unique at international level. So I think there is and of course, it's a large campaign that cannot be sustainable to long term, easy, difficult also to export, also for questions on sustainability. However, it has been, we believe, it is useful to give a contribution to better understand the complexity of the phenomenon Xylella on the territory. So as you may know already, in this region there are now 3 different subspecies that have been found, and almost 50% of the whole region now is demarcated mostly by Xylella subspecies pauca, but also now with some relevant presence of multiplex and fastidiosa. So the regional authorities deemed it appropriate to make such a large data set accessible to both the scientific community but also to the general public through the institutional website of the region, that is this one, Emergenza Xylella.8, which also provide maps and cartographic representation for each annual monitoring campaign. The files of the database that everybody can download are preceded by an introductory notes containing some instruction for the correct interpretation of the data, which are essential to avoid the risk of misinterpreting the database, risk that unfortunately periodically still occur, because we know that time by time we can read some article or even some paper misinterpreting this data. So the purpose of the monitoring is not to inventory infected plants, but to anticipate the bacterium by identifying new outbreak at the early stage. Consequently, since the monitoring is done mostly in Xylella-free area, in buffer zone that has the denomination should be also Xylella-free, and just in the containment area that is the terminal external part of the infected zone, means the area where the bacterium is not well established yet, but there is a very low frequency of the bacterium. So a very low percentage of the positive is the expected result. And so, of course, doesn't mean that if we have only 1% of Xylella, doesn't mean that Xylella is not the causal agent of the... okay, I think you understood. So this is the synthetic final table of all the data. You can see over 1.5 million, and out of this 1.5 million, only 18,000 samples tested positive in this year. So totaling 1.23% of the total. So a very low percentage. And the majority of this, 83.5%, are olives. So 1,250,000, while and 1.38% of olives were positive. This is just to give you an idea. As you can download, you can consultate in the website also the map for each campaign of monitoring. You can see that the yellow correspond to the visual inspection, so not very important, but the green are negative samples and red are the positive samples. And as you can see, after the very first year, the southern part of the peninsula, where there is the devastating and very high impact of the bacterium, has not been monitored anymore. So that's why the percentage is so low. The database also provides additional valuable insights, as, for instance, evidence of different degree of susceptibility to Pauca ST53. So we try to propose this kind of infection risk index. So, but in other words, we calculated the ratio between the percentage of the positive of each single species and the percentage of positive in olives considered as a kind of reference species. And you can see that there are a few species that with very high risk of infections, like lavanda, like polygala, like acacia, like broom. But there are also many plant species for which the risk is low or even negligible. So geranium, laurel, something like that. So other additional valuable insight are you can draw from this a further confirmation of the etiological role of the subspecies pauca as agent of the olive quick decline syndrome. So because even if the monitoring program it does not interest the infected zone, but there are also few exceptions. One occurred in the campaign 2021-22 when a small part of the monitoring interested also a part of the infected area. This allowed to raise the number of positive to 100 and some that were in the official area up to over almost 3,000. And as we can see, we can observe a gradient because coming from the north to the south, from the Xylella-free area to the area that is getting more close to the very highly intensively infected area, we can observe a kind of growth, exponential growth of the percentage of positive, moving from the 0 in the Xylella-free area to 5%, 6-7% in buffer and containment area, up to 70% in the northern infected area. And when we move further to the south, to the very devastated area, this can rise up to 100%. It was the case of 2017 when 500 out of 500 highly devastated trees of the susceptible cultivar were completely, were all positive. So, for those that are interested in exploring the database, because it's not very easy to surf on it, if you are interested, you can take a note of this link, because the Regional Plant Protection Service at the Mediterranean Agronomic Institute, they prepared a detailed guide or instruction to its consultation. And they published in the frame of the project Regenesi, and as a briefing number 1, and you are welcome to consult, to download this. Okay, and thank you.
Thank you very much, Donato, and I think it's very clarifying information and interpretation of these results. Our next speaker is Martina Zendoya from IBIA, Spain, and the title of the talk is Delimiting and Managing the Xylella fastidiosa outbreak in Alicante, Spain, Insights from Surveillance and Eradication Data. So, Martina, the floor is yours.
Okay, thank you for the presentation, and also thank you to the organization. So I am going to talk about the Xylella fastidiosa outbreak in Alicante, where this slide is not well. Okay. The first detection was in 2017, and it was detected the subspecies multiplex, mainly affecting the almond trees. And also I have put here some data on the current situation. However, in the next session, Vicente Dalmau from the Regional Plant Health Service will explain in more detail this current situation. So I will first start briefly summarizing some analysis that we did from the first surveillance data. And here we analyze the effect of climatic variables on the presence of Xylella fastidiosa in this area, using a Bayesian spatial modeling. Sorry, but there are many things missing in this presentation, but I sent the correct file. So I can continue, but the slides are not correct. No, there are many things missing. I can continue with it if it's...
Maybe we can switch. Maybe we can switch speakers. Maybe while they can solve your slides problem, because maybe it's a matter of maybe they are using the old PDF or something.
Sorry.
So don't worry, Martina. We will have time to your presentation.
Next speaker is Anne-Laure Boutini from ANSES France, and the title is EFSA Project and Detection of Xylella fastidiosa in Important Mediterranean Plants During Dormancy.
Okay. Okay. So hello everyone, and thank you to the organizers for giving me the opportunity to present this EFSA project on the detection of Xylella fastidiosa in important Mediterranean plants during the dormancy phase. A little bit of context: the multiplication of Xylella fastidiosa within a host plant depends on environmental conditions, bacterial strains, and the species or cultivars of the host plant. For trade purpose, an analysis of the plants to be planted is required, and in order to maximize Xylella fastidiosa detection, sampling should preferably be performed during the plant's active growth period, and this period extends from late spring to autumn in temperate zone. For deciduous plants, analyses are often required during the dormant phase when bacterial activity is the lowest, and previous experiments conducted within previous European projects have shown that Xylella fastidiosa could be detected in some deciduous plants all year around, including during the dormancy and the asymptomatic phase. So in this context, the aim of this EFSA project was to evaluate methods to detect Xylella fastidiosa and identify its subspecies in dormant plants of interest for Mediterranean countries, as there is a great interest in dormant wood analysis, especially during the winter and for trade. This project started in 2023 for 2 years. It involved 20 laboratories from 15 countries, from the USA, Australia, Europe, and North Africa. There were 3 main tasks. The first task was to realize an inventory of dormant plants that could be infected with Xylella fastidiosa, and of the methods, including sampling methods, DNA extraction, diagnostic tests, to detect Xylella fastidiosa and identify subspecies in dormant plants. This task was included questionnaires to the laboratories. The second task was to evaluate methods on dormant samples spiked with Xylella fastidiosa. This work was done at CREA in Italy. And the third task was to evaluate the methods and Xylella fastidiosa dynamics on dormant samples naturally infected with Xylella fastidiosa. So the first step was to collect dormant plant materials that were naturally infected with Xylella fastidiosa. Then these samples collected, we organized a test performance study on these naturally infected samples collected. And the distribution dynamics of Xylella fastidiosa in woody stems naturally infected was evaluated throughout the year, including dormancy, by 2 laboratories, so Esther Marcono-Ales at IVIA and Maria Saponari in Italy. I will present you more specifically the test performance study that ANSES organized on the naturally infected samples we collected. So I will present you the process and how we organized this test. So first, we collected samples naturally infected with Xylella fastidiosa. We collected dormant twigs and healthy twigs in France, Italy, Spain, and Israel, from almond tree, plum tree, and grapevine. And the infected twigs were infected with different subspecies of Xylella fastidiosa and different sequence types. So there was a good diversity of infected samples with 3 different matrices collected at different locations and infected with different subspecies, multiplex, fastidiosa, and pauca, and different sequence type. Okay, the panels sent to the participating laboratories included 10 samples, including 8 infected samples and 2 healthy samples. There was also 1 lure different for each laboratory, and it was a blind testing. The homogeneity and stability of samples were verified, and 14 laboratories participated to this test. On the photos, you can see how we prepared the samples. The twigs collected were cut into 2 centimeter pieces, and after homogenization, 10 pieces were randomly selected to form 1 sample. And this is what we sent to the participating laboratories. Various protocols for Xylella fastidiosa detection and subspecies identification were tested at the choice of the participating laboratories. You can see the different protocols tested in this table. And a common protocol for Xylella fastidiosa detection, CTAB DNA extraction, and Harper real-time PCR was proposed by the organizer and tested by 10 laboratories to have a common protocol to all laboratories. And we analyzed the results for all methods and all participants. 3 Xylella fastidiosa detection protocols were tested by a large number of laboratories, including Harper Simplex PCR with CTAB, QuickPeak or DNeasy extraction DNA, and 3 subspecies identification protocols, including Hodgett Simplex and Dupat tetraplex with CTAB or QuickPeak DNA extraction, were tested by 3 or more than 3 protocols. And for these protocols, performance criteria could be calculated, including diagnostic sensitivity, diagnostic specificity, accuracy, repeatability, and reproducibility. This is a summary of the results we obtained. I will go quickly, but some protocols were validated to detect Xylella fastidiosa in dormant twigs. And some protocols were validated to identify the subspecies of Xylella fastidiosa in dormant twigs. As a remark, other protocols were also reliable to detect Xylella fastidiosa or identify subspecies in dormant plants, but they were only tested by 1 or 2 laboratories. As a conclusion, the Harper PCR simplex after CTAB, DNEZ, and quick-peak DNA extractions was validated to detect Xylella fastidiosa in dormant plants, and the Hodget's PCR simplex and the DUPA PCR tetraplex were validated to identify subspecies of Xylella fastidiosa in dormant plants. The results of this work are available in the Fresco report and in the TPS report, if you are interested. And they will also be proposed for the next versions of the EPO PM724 and the ICPP diagnostic protocol for Xylella fastidiosa. And I would like to acknowledge the people from my lab who helped with the preparation of this test, and the colleagues who participated in the other work package of this project, and all the participants of this project. Thank you.
So thank you very much, Anne. It seems that the technical problems are solved, so we come back to the presentation by Martina. So second chance.
Okay, so thank you. I'm sorry, things that happen. Let's see.
It will come.
It will come. So I was explaining that I am going to talk about Xylella in Alicante. So we have the subspecies multiplex and affects mainly almond trees. And here we have some data that Vicente Dalmau from the Regional Plant Health Service will explain later in more detail. And these maps to situate where is this outbreak and how it looks now. So we previously started with some analysis of these first survey campaigns to analyze how the climatic effects can affect to the presence of the pathogen there using this spatial Bayesian model. But we showed that no relevant effect of climatic variables were found on the probability of presence of the Xylella fastidiosa in that area. But we had a strong effect of the spatial component. So to go further on this spatial relationship, we estimate this spatial range. This parameter is the maximum distance at which two observations are considered to be spatially correlated. And we found that we estimate this parameter more or less on 4 kilometers. And here in this plot, we can see the spatial correlation, which is the red line, how decrease with the distance, and the first blue line is the minimum buffer zone established by the legislation, so this 2.5 kilometers. Then we have our spatial range, and the last blue line is the 10 kilometers envelope, external envelope that the Plant Health Service are applying to prevent and to survey around the affected area. So this envelope will be explained also by Vicente later. Also, we analyze some outbreak management plans, and to do that, we start simulating the most likely origin of the pathogen in that area based on molecular epidemiological studies that estimate the first introduction in the mid-90s, and also based on the current extent of the infested area and on the almond tree distribution. So we use this baseline scenario where we simulate the spread of the disease without interventions during over 30 years. So based on this baseline scenario, we simulate different combinations of outbreak management plans, including surveillance strategies based on the EFSA guidelines. with different confidence levels, also including different control measures as different eradications, radius and buffer zone sizes, and also other measures that can reduce the transmission of the disease. And we can see in these plots that the red line is our baseline scenario without interventions, and the other lines are with different combinations of these surveillance and control measures, and all of the management plans reduce the infected almond trees in the area over time. However, only those with very high surveillance effort can eliminate completely the infected trees. So after this briefly summary of previous studies, now I will move to the current picture that we have after 9 years of surveillance, intensive surveillance, and other control measures. So here we can see the cumulative sampling effort on this grid of 1 kilometer cells during the different years, and we can see how the surveillance area progressively expanded in the area, also based on the detection of new positives. This surveillance is heterogeneous in the affected area, also because the regulation, following the regulation, they have to survey more intense around the new positives. So to see if there is a possible extension of the outbreak or just is based on the surveillance extension, we calculate the detection front based on the previously positives detected during the different years and the distance of the new detected detections from each year to this detection front. So we can see in this plot that each year the distance decreases, the distance of the new positives to the previous detection front. However, in 2023, we have this peak of distance, and we can see in the map below that corresponds to these red dots new detections that are outside of the previously surveyed area or in areas with previously low survey effort. So that's why we have this peak in the distance. But apart of this year, again, in the following years, the distance decreases. So overall, we cannot say that there is a substantial expansion of the outbreak. Also, apart of surveillance, there has been applied eradication measures, and we can see here in the maps in the following years how the extension area expands with the new positive locations. However, in the last years, the perimeter of the extension area is more or less the same. So this perimeter is not extending a lot in the last years. And also we can see in the corner some new positives that correspond to the previous slide, where we haven't got a lot of samples in the previous years. So also to see what is the effect on this eradication, we calculate the distance of the new positives of each year to the nearest eradication area. And we can see again that this distance over the different years decrease, with this exception of the peak in 2023 that corresponds with this low surveillance effort area. So just to finalize, some points to highlight is that we show that there is a strong effect on the spatial component on the Xylella fastidiosa presence on this area. Also, the spatial analysis support the extent of the surveillance area around the outbreak, and the survey effort determines the performance of outbreak management plans. Also, the outbreak delimitation in Alicante was achieved progressively over several years, and the survey effort was heterogeneous over time, following the legislation, with greater survey effort around positives. Also, the new detections occur close to eradication areas, and we cannot say that the outbreak spread is substantial so far, but this can be a bias on the surveillance or a low spread of the outbreak. So we need further studies to see if the outbreak is really spreading or what is happening there. So I want to thank the Brexit project and also to the organization and to you for attending. And sorry again for the previews.
Thank you, Martina. And apologies for the technical problems. Next speaker is Giuliana Loconsole from IPSP CNR from here, Italy, with the talk Large-Scale Validation of qPCR Assay for Simultaneous Identification of Xylella fastidiosa subspecies in a wide range of plant species naturally infected. The floor is yours.
Good afternoon, and thank you for this opportunity. And the research was carried out in the framework of the official monitoring program of Apulian region, and focused on the large-scale identification of the subspecies of positive findings of Xylella fastidiosa by a multiplex real-time assay developed by DuPa in 2019. In the European Union, the genetic characterization of Xylella fastidiosa is not only a scientific need, but a legislative requirement. According to the regulation 1201 amended in 2020, the plant species submitted to eradication and containment measures differ according to the subspecies as listed in the Annex 2. Thus, diagnostic method must comply the legislative requirement as listed in the Annex 4, which include some molecular assay to detect the bacterium, identify the subspecies. When the regulation was amended, subspecies identification relied mainly on MLCT analysis and several conventional PCR approaches which differentiate subspecies Pauca multiplex and fastidiosa. Following the implementation of 2024, two real-time assays, including the one of DuPa, were introduced to speed up the procedures for the identification of the subspecies and to determine all the subspecies. Donato showed to you these slides which represents the complex phytosanitary scenario that emerged in Apulia region starting from 2024. Three different subspecies and three different sequence types are in our territory now. The fastidiosa ST1 is mainly present, in fact, almonds and grape plant plants. On grape plants, symptoms are resembling those of the Pierce disease. The multiplex subspecies sequence type 26 predominantly infect almond plants with no specific leaf scorch symptoms recorded so far in the field. And new outbreak of Pauca ST53 are present in the northern part of the region, of the Apulia region. Thus, this complex scenario prompted to the implementation of the diagnostic workflow to integrate the detection bacterium, the identification of the subspecies on positive samples in the framework of the monitoring program. all the analyses to detect the bacterium were in accordance, were performed in accordance with the standard ISO 17025. The process starts with the preparation of pooled samples. The first molecular screening is performed by the PCR or real-time PCR of Harper, and if the samples are positive or undetermined, there is another molecular screening by Harper on the single samples of the positive pool. Thus, the samples, DNA and plant material positive to Xylella fastidiosa, resulted positive to Xylella fastidiosa, and the undetermined samples, the plant material of these undetermined samples, are sent to the laboratories of the CNR EPSP, where further analyses are performed. The subspecies identification is carried out on all DNA samples coming from the positive samples of the field. Between these two here, more than 1,600 positive samples were identified and subjected to the subspecies identification. When we start with this research, we need to slightly modify the reaction condition of the protocol reported by DUPA, because we met some critical issues when we carry out the qPCR using the 60 centigrade as a temperature of annealing the extension steps, because some amplification curves were generated with very low relative fluorescence, with lack of detection in some positive samples at low concentration. Thus, we tested the gradient of temperature, and when lowering the temperature around to 55 centigrade, the amplification curves, the efficiency of the amplification curves for the PAUCA was implemented significantly without compromising the detection of the other subspecies and the specificity. These are the results obtained in these 2 years. More than 1,500 samples were unambiguously categorized according to the different subspecies, and the fastidiosa was found mainly on grapevine, and Prunus dulcis and grapevine, and occasionally findings were in cherry and Polygala myrtifolia. The subspecies multiplex was predominantly in almond, with sporadic infection in plum, cherry, and Spartium gentium. The subspecies pauca, as expected, was exclusively almost associated with the olive, with rare detection in Prunus dulcis and one plant of oleander. We used a triplex format of the DUPA to target the different 3 subspecies. This slide shows the CQ value range obtained for each subspecies across the different host plant. The range is similar in for pauca in olive, for multiplex in almond, approximately similar, and for fastidiosa in almond and grapevine, starting with a CQ of approximately near to 20. In 105 samples produced undetermined results with high CQ value and sometimes with no optimal exponential amplification curve. Most of these undetermined results were produced for almond, and when these samples were retested starting from the DNA extraction, 16.9 resulted positive, 32 negative, and 4 remain undetermined. Most of the positive were, as expected, were plants of almond infected by multiplex. Probably this indicates erratic distribution of these subspecies in the almond plant. The test was also applied on insects to identify Xylella fastidiosa in insect, and on more than 600 samples tested in accordance with the ISO 17025, 11 positive were detected, and 3 with fastidiosa, 4 with multiplex, and 4 with pauca. In conclusion, the optimized triplex qPCR based on the DUPA assay is reliable for simultaneous detection and subspecies identification of Xylella fastidiosa. Decreasing the temperature to 50 centigrade, the sensitivity for the species pauca was improved without affecting the detection for the other subspecies. The assay also proved reliable also in insect samples, and a large-scale application confirmed a clear host subspecies association and supported effective surveillance. Also, the robustness of this assay to differentiate the 3 subspecies was also confirmed by MLCT analysis, which was performed always on new outbreaks and newly identified host species. Although in the province of Bari, all the 3 subspecies coexist in a small area, positive samples did not show any mixed infection so far, including in almond, which is susceptible to multiple subspecies. Overall, this assay represents a rapid and robust tool for phytosanitary management, alternative to the MLCT analysis for a rapid and large-scale characterization of the positive findings of Xylella fastidiosa. I finish, and thank you for your attention. To all my colleagues that share with me the complex management of the accredited laboratory in accordance to the ISO 17025. Thank you.
Thank you, Juliana. Thank you.
Thank you, Juliana. Next talk will be with Carla. So Carla Luis Carvalho from the Institute for Nature Conservation and Forest at the University of Villarreal in Portugal. And the talk will be Xylella fastidiosa, from eradication to containment, the Porto metropolitan area case study.
Thank you. Today I bring to you an overview of our first detection in Portugal. That was detected on January 2019 in Vila Nova de Gaia on the Porto metropolitan area, an Atlantic-influenced bioclimatic zone. This demarcated area has 3 main regulatory frameworks. In the beginning, it was decision 2015-789. Then was the transition to regulation 2020-1201, and in 2024 we had a big change for containment strategy, and this analysis intends to analyze the spatio-temporal dynamics characterizing temporal trends and modeling spatial patterns across the territory, and finding also the dynamics across different host plants and land use regimes to provide a robust evidence base for the epidemiological framework from transition to eradication to containment from eradication. So, it's not working. Okay. Our study area, it's a complex land use mosaic. As you can see, using Carine land cover, we have mostly urban and forested areas. Difficult place to work because many of the areas are bushlands or dense forested eucalyptus forests. And we have three main zones for the containment strategy: the interior infected area, where we don't sample, and in the containment infected zone, we don't want the spread to go away, so we sample there and in the buffer zone. This data this analysis will scope the data survey from 2019 till 2025 with our NPPO, the GAV Action Plan for Eradication of Xylella fastidiosa and its vectors technical framework based on EU regulations, EFSA guidance, and of course ESPM 27 for Xylella fastidiosa. We used the systematic grid survey and REBAS statistical tool to determine the number of samples for each land use type. For agriculture, urban, forest, and semi-natural ecosystems in arboreal, shrub, and herbaceous layers of vegetation for both symptomatic and asymptomatic plants. Here we can see the infected zone. We selected a buffer zone closest to the infected zone, where we survey more intensively. And we can see our land use structure. For the analysis of these data, we used Python and QGIS software, used EverSign formula for calculating the kilometer scales distance, and expansion profiles, and convex hulls to track the outbreak's annual maximum spatial envelope. And we used data harmonization, standardizing land use categories to be more easy to classify based on core inland cover, of course. And botanical taxa with the total samples, more than 100 samples for statistical robustness with Wilson 95% confident intervals. We used also 2+2 contingency framework to comparing symptomatic versus asymptomatic plants using odds ratio and Wilson 95% confident interval to evaluate the diagnostic performance. Our results. Our data set has 15,820 samples in total. We found 512 positive detections from subspecies multiplex ST7 and subspecies fastidiosa ST2. The total, the overall positivity rate was 3.25%, but we can see the positive rate is stable, relatively stable through all the study period. Okay. Plant samples by zones. We have two distinct phases, the eradication phase, where we can see clearly that positive rates reduce from the inside outside, of course, because the probability of finding positive in infected zone is higher. And then we can see for the containment measures, we still don't have samples, a significant number of samples, because it was in 2024, so we are still collecting the data. For land use, we can see mainly forest and semi-natural areas as reservoirs, the main reservoirs for the persistent infection levels, and we can see clearly that in agriculture areas, we have a near zero rates of positivity. So, for species analysis, we choose to analyze for genera And for eliminate statistical noise, we selected more than 100 samples for a taxonomic filter. We can realize that lavandula is the main primary urban amplifier and is a risk driver among heavily sampled ornamental targets, and wild shrubs reservoirs like wild fabacia, Ulex and Genista are the worst, and they potentiate widespread and persistent reservoirs for the bacteria in unmanaged natural areas. But we can see, for example, for quercus and for olea, that there is a near zero positivity rate with higher number of samples. This was our main phases, the initial focus on 2019, and the demarcated area in 2023 before the transition to the containment strategy. The 2025 is how it is nowadays. For our results, the epidemic scale, we have now a total demarcated area with 67,400 hectares with 30.55 kilometers maximum linear extent, as we can see on the right, in the figure on the right. And we have a spatial asymmetry, 12.83 kilometers north versus 19.31 kilometers south. For early radial jumps, we can see the biggest jump was in 2020, as we see in the table. And the convex source represents annual and cumulative maximum detection envelopes, so we can integrate every detection that we had and analyze the spatial data. The genetic predominance, we have subspecies multiplex, of course, and we have a complex introduction because we have two subspecies, so with occasional detections of Xylella fastidiosa subspecies fastidiosa, ST2, indicating different historical events and could be a spillover, a national spillover. And I have to move on. For symptomatic plants, we can see that we have a big portion of symptomatic plants showing positive, revealing to be positive. In the outer ring, you can see the total sample plants. In the inner ring, you can see the positive detections. And for conclusions, we have the data to support transition from the regulation, from the eradication to containment measures. And we have to, the containment is a data-driven proportionate response focused on slowing outwards and protecting high-value agricultural sectors, and for me as well, to protect forest areas, because there is a big point on my country. So if you want to see national overview, you can see the poster 123, and as a final acknowledgements, I would like to thank my NPPO in Portugal for kindly acknowledge the official monitored data, and CIM Bari for supporting my participation in the conference. Thank you.
Thank you very much, Carla. And our next speaker is Diego Olmo from IRFAP, Spain, and he's going to talk about an overview of 10 years of Xylella fastidiosa in the Balearic Islands, impacts on crops and lessons from a multi-subspecies epidemic. The floor is yours, Diego.
Well, hello everyone. Thank you to the organizers for inviting us to give us this talk, to give you this talk. We think that after 10 years living with Xylella in the Balearic Islands is a good time to summarize an overview of our main highlights, main findings of our outbreaks. Well, this work is the results of this work, of this presentation, is the result of the work of many people, many collaborations, some research projects that I will comment. And then we start with the first detection in October 2016 in 3 cherry trees at the garden center in Porto Cristo at the east coast of Mallorca, the main island. Then afterwards, we found also some polygala plants at the same garden center. And at first, it appeared to be an isolated interceptor, but soon we find a lot of new detections in fields, but not only in Mallorca, also in Ibiza and Menorca. Actually, that moment, well, and soon in 2017, in 2017, we realized that the disease, Xylella was widespread across the archipelago. Also, we detect a remarkable genetic diversity. As you can see, in Ibiza, we only had Pauca ST80. In Mallorca, we have fastidiosa ST1, multiplex a few samples with ST7, a lot of samples with ST81. And in Menorca, only multiplex ST81. That leads to the containment strategy approved by the Commission in December. This is the list, the current list of the 38 species where we detect Xylella so far, but in which we most frequently detect Xylella is this list. And as you can see, among them, there are the major crops from the Mediterranean, also from Balearic, of course. And that concerns us, of course. And the next slides, I will show you some symptoms. I think it's interesting to see that the characteristic symptoms of Xylella in the Balearics are the same around the world. But, well, in grapevine, typical symptoms of Pierce disease are observed in Balearic Islands as leaf scorch, green island, magnification. For almonds, we see leaf levels, we see the typical leaf scorch, but in advanced stages of the disease, we can also see the symptoms of golden death, as you can see in the right. And for olive trees, the situation was different if we are in Ibiza or in Mallorca and Menorca, because severe symptoms were confined for several years in Ibiza, because they have Pauca and we have in Mallorca multiplex and also in Menorca. But that changed recently. In 2024, we found our new outbreak in the village of Sencelles in the central island, the new outbreak of Pauca in this case, and the ST53. So this is the current situation up to date. Now we have the disease spreading, the bacterium spreading. And the main finding, or the most important, is the new outbreak in the center of the island. Well, now I will explain some of the findings, highlight some of the findings within the some projects we have participated during these years. The first one is one on a national level, and the risk assessment work package, they demonstrate that entire Balearic archipelago at a high risk of the establishment and the spreading of Xylella, the three subspecies of Xylella, as you can see. Another interesting result from this project came from dendrochronology and genomics, and they found that Xylella are present in the Balearic Islands since the 90s. Other results, other interesting results, is that in that moment the European introductions represent independent events, and our multiplex and fastidiosa were originated in Almond in California. And on another project, there was some work package. One work package focused on natural vegetation. They found 13 novel hosts. This is important because natural vegetation surveys can uncover hidden reservoirs. It's interesting. Another work package focused on early detection, validated an aerial detection system for Xylella and almonds. Regarding vectors, the most important finding for me was the Balearic climate favors the bacterium over its vector, and so climate change may reduce transmission. And regarding varieties, almond genetic diversity has not limited the spread, even though there are some varieties that perform better than others, but the spread is continuing. And we estimate that more than 80% of almonds today are affected by LSD. And finally, I want to comment Big Silpred that ends in August. We are participating in many tasks. Some of them they are listed. But the most important for us is that during the project, we found a new outbreak, and we take a profit in doing some tasks, some trials inside the new interesting outbreak, this small area, because analyzing this case, this particular case, we obtained that Pauca outbreak, the new Pauca outbreak, is highly localized, less than 2 kilometers. The three species, Pauca, multiplex, and fastidiosa, are confined in a small area. And very interesting, we have been detected co-infections. As you can see the map with the colors, you can see the situation, and the closer that the different species are. And analyzing the genome of these strains, we are obtained that they are virtually identical from Napoleon ones. So our hypothesis is a recent introduction similar to those that you here in Napoleon happened here in Napoleon. Yeah, and thank you for your attention. That's all from my side. But I will leave you with this photo. It's a photo taken during a visit of researchers from Australia and New Zealand. We have with us Luciano. And this is an example that Xylella could be, Balearic Islands could be a good place to visit for research on Xylella. It's a kind of natural laboratory to make trials, but also for visiting nice places like this one. Sorry. Thank you very much.
Thank you, Diego. Next time with Elisabetta Poeta from the Institute of Bioscience and Bioresources of Silner in Reggio Emilia, Italy. Thank you.
Good afternoon, and thank you for the presentation. So today I will discuss the application of a rapid, portable, and undestructive tool to support phytosanitary monitoring. In this study, we use a device equipped with a tailored metal oxide semiconductor sensor to study soil volatile profile. So sensors are robust, compact, low cost, and give us information in a real time or after a few minutes. They don't require complex pretreatment, and they can be integrated into a portable system for analysis outside conventional laboratories. Overall, the responses can be tailored based on the product analyzed and the volatile organic compounds released by the sample, modifying the key parameters such as the sensing elements and the operator temperatures. So in this project, we analyze soil obtained from lecino olive trees, comparing the uninfected soil with soil associated with Xylella fastidiosa subspecies pauca infected plants. All the soil were prepared in an authorized laboratory in the CNR IPSP. So we use two different approaches: a device equipped with a tailored metal oxide semiconductor sensor to have information in a real time regarding the generic volatilome, the volatile fingerprint, and a traditional technique, the gas chromatographer mass spectrometer, which allow us to have information regarding the specific compounds released by the sample to support sensor data. Regarding the gas chromatographer mass spectrometer results, each sample was analyzed in biological triplicate, and we noticed that there were differences in chemical compounds through their relative abundance. And these variations suggest that the infection status of the plant may influence the volatile profile through the root activity or microbial metabolism and so on. Regarding the MOX sensor results, all the analyses were conducted in standard and control condition, and for each sample we analyzed both biological and technical replicates, because we would like to verify and to improve the robustness of the sensor, the data set, and verify if sensors were able to distinguish the volatilome, the volatile profile. We noticed that there were two clusters in three-dimensional PCA, one, the blue one, more compact, related to the uninfected soil, and the red one with a great variability due to the infected soil. And as for the gas chromatographer mass spectrometer result, this variation suggests that the infection status of the plants may influence the volatile profile. So actually, we notice that sensors are able to distinguish the volatilome, the volatile profile obtained from the uninfected soil and infected one, and then we would like to compare all the results obtained, and in the next future the idea is to create a prototype and test it directly in the olive orchard to verify if sensors are able to distinguish and to detect the infestation. Thank you.
Thank you very much, Elisabetta. And our next and last speaker is Florian Kunze from the European Food Safety Authority with the talk Horizon Scanning as an Integrated Approach for Monitoring Xylella Fastidiosa. So Florian, the floor is yours, and you can use the other microphone.
It's already on. Questo? Ah, sì. Okay, so yeah, hello and welcome also from my side, from the plant health monitoring team at EFSA. I will present to you today the horizon scanning activity on the example of Xylella fastidiosa. So, So basically, what is horizon scanning? Voilà. We are obviously not just watching the horizon for some pests, but what are we doing is doing intelligence work on a variety of pests in the internet using a web scraping platform, and like this collecting intel on these potential emerging threats for the EU agriculture. And of course, one of the main objectives is to do this in a manner as fast as possible, and also to report as fast as possible. Basically, we started with the horizon scanning activity in 2017, following a mandate of the European Commission. And since then, we've been cooperating with the JRC and also ANSES, for example, for our tools and the daily work. We publish our monthly results in a newsletter dedicated to the horizon scanning on plant health, which you can also access over this QR code here. And this newsletter gets, on one hand, sent to the Commission and the PAF committee, but also is made available for the general public. Of course, we also share the results over social media and the horizon scanning dashboard, which you can also see here in the slide. Our basis for the horizon scanning is the EOS platform, which is a really powerful tool hosted by the WHO. And this tool allows us to scan the internet for evidence, articles, news, social media posts, which might indicate a future or emerging threat, and also to monitor current already regulated pests. On this platform, we receive between 7,000 and 10,000 articles on plant health per month. So it is quite a big amount of data that we have to deal with. Okay, so basically here we see the development of the number of fetched articles on Xylella fastidiosa over the last years. And on one hand, we see that there is the general increasing trend. On the other hand, we see that there's quite a lot of variability. And we managed to backtrace those peaks, basically, and link them to the real-world events. And what is also important to see here is that it's not only linked to plant health, but we also receive, of course, a lot of articles to the social and economic dimensions of this problem. Yeah, so basically, in conclusion, the horizon scanning is being used complementing other procedures from EFSA and the NPPOs since 2017. There had been a lot of cases where the importance of considering the weak signals has been demonstrated, because often it is really the first signal of an emerging threat, and sometimes we were able to already raise the awareness and assess the risk on something already years before the arrival in the European Union. With regards to Xylella, we were able to track the development and the movement of Xylella globally. And we were, of course, also able to provide the timely information on new outbreaks and spread to the risk managers and the decision makers. So we are still, of course, always developing more and better procedures and improving the tool. So, yeah. And maybe see you afterwards at the poster 115, and thank you for your attention.
Okay, thank you very much, Florian. And before going to questions, I would like to highlight that two of our speakers, Carla and Elisabetta, both were granted by the Young Research Initiative, so congratulations. No, the funding that you got from EFSA for coming here. No. Okay, so now the floor is open for questions. Okay, I see one there. Wait for the micro one moment, please. It's coming.
Sorry.
And if you can introduce yourself, that will be helpful. Thank you.
Hello, Elif Olkun from Germany. I wanted to ask a question to Florian Kuntz. If, for example, the monitoring that you are using Hello. The monitoring that you're using, have you ever thought, or theoretically, can you use it to, for example, look into the past social media posts or articles before the outbreak happened at all in Europe, and see if there was a correlation or peak in some information or information that you see in the social media, for example, if there was any, move towards some symptoms or indication that this disease was visible?
Yeah, thanks a lot for the question. We also received quite a lot of similar questions 2 weeks ago. So basically, in general, it is possible to even go back in the past on this platform. However, the only condition is that we have to set the keywords which we used on the tool before. So if we are implementing one category, for example, today, we cannot fetch or see articles that were published the days before. However, if we set it last week, we can go back until last week. So the category and the keywords are basically determining it. But in case you're interested, we also have a community on EOS, so the tool, where we are also exchanging a bit knowledge and the use of it. So, yeah. Thanks.
Good afternoon to everybody. I'm Ayla Daudbasic, a representative of National Plant Protection Organization of Bosnia and Herzegovina. I have actually, there are two questions for colleagues from Italy and Spain, probably. So based on their experience, which part of eradication procedure was the most challenging? And does those eradication procedures differ from EU procedures based on the experience that you have on your national level?
Well, difficult for me to answer to this question, but I think that in general the program of eradication for Pauca was already something that was not really realistic, because the area that was interested was already big. Too big for a pest, for a pathogen that has also flying vectors and a list of hosts that were unknown at that moment. When it was discovered, we didn't know anything about the subspecies and also of the list of the hosts. And in addition to this, there are all the problems related to the plant, the trees, the kind of trees. For all things that has been already described this morning, I'm not going to repeat. So this has been the most challenging tentative of eradication. While, just to make a comparison, now is going on a program of eradication for the fastidiosa near Bari. Up to now, the program can go on more smoothly because in addition there is also a good experience that has been gained by the regional authority. So now they have been already prepared, ready, also with a good program of compensation, because a program of economic compensation is crucial to try to have results from a program of eradication. I don't know if I answered more or less to your question.
Yes. Well, in the case of Alicante, which is under eradication, I know that many hectares have been eradicated and many almonds removed. Maybe I'm not the best person to answer this question. Maybe in the next session, Vicente Dalmau from the Plant Health Service can answer better. But in my opinion, and now they are putting a lot of effort on this eradication trying to contain the disease in the area. And I know that at first it was difficult and with many social and cultural difficulties, but now the process is softness. And well, I don't know if I have answered well.
Thank you. More questions?
I have a question for Donato and maybe Juliana also. So you mentioned that lavendula is most highest risky plant. And when do you find many lavender positive in Apulia? Because Juliana didn't mention lavandula in his presentation. And after I have another question.
Well, lavanda, according to just to the statistical analysis of the data, in lavanda we found the higher percentage of positives among the plants that have been sampled and tested. Of course, the numbers of the lavanda tested are much smaller compared with those of olive or other things. So maybe the data is not really statistically significant. However, the small experience that we have, because in Apulia we do not have lavanda as a crop as you have in Provence, in some place of France. So they are mostly ornamental plants in some garden and in an area under pressure inoculum has been quite easily found. Also in some gas station out of the infected, so on the forefront of the epidemic, often lavanda was one of the plants that were identified, detected as infected.
Can I? Sorry,
no, the lavender was not the.
Please.
I have
not
a
in my
question.
presentation, the lavender, because the results that I showed refer to 2024 and 2025. Infected lavender are previous detected in the year.
Okay, I have a question from Florian Kontze. As you are already working with the horizon scanning and integrated approach for the monitoring, and I am already working on the similar approach and trying to integrate a singularity point for all the research. My website is xylellafastidiosa.com. So I want to know, can I access the data and bring all those research and the academic publications on this one common point where we can bring all the research and make it more accessible?
So your question is, if I understood it right, if you can access the data on EOS, for example? Yeah. So generally, I think it would be possible. I mean, I would definitely have to ask my bosses, but generally, it would be possible to join the community and also extract data from the platform via the API. So.
Okay, I would like to have a collaboration because the website xylellafastidiosa.com is with the same approach, with the same aim, and we are trying to bring something which is a hub for the integrated research for all the publications or all the work that is with the aim of Xylella related things.
Yeah, sure. I will pass the contact information that we would
Thank you.
consider that, yeah.
Okay, Emilio?
Emilio Montesinos from the University of Girona, Spain. Up to now, there has not been too much talks about nurseries, especially nurseries in the areas like, for example, the Puglia and Alicante and Portugal. I think there is a talk, I think tomorrow, from José María Pagés that will give us the opinion from the stakeholder nurseries. But what has been the impact of the Xylella in this area, in the Puglia, in Italy, or, for example, in Alicante? Because I think nurseries are the primary suffering the problem because of exportation, sending the material, and the regulation is very strict because if the Xylella bacteria has hundreds of hosts, probably in a nursery, or for example, of ornamental plants, it may be that 100% of the material is susceptible. So what is the experience on that?
For you, Donato.
So this is a critical point because, of course, as you can understand, the impact of Xylella, not of Xylella, but of the regulation, so the management of Xylella on nursery, it's almost dramatic. Okay, I understand, I don't know the detail, that there are good compensation also to support, to help the nursery. But in any case, the impact is dramatic. And there is a general feeling that probably in some case there is an excess of precautionary measures because I don't know, just to give an example, now as far as I know, the movement of the plants in a nursery that are in demarcated area is depending from the list of the host, the plant, the susceptible plant species belonging to each specific subspecies. And I don't know, for instance, I know if you look at the list of the multiplex, you have more than 100 hosts. And it means that are more than 100 species that cannot move. But just to give another example, among the host of the pauca, there is, of course, as it is well known, citrus. But citrus is not host of the strain of pauca that we have in Apulia. However, I understand that if one nursery is in the demarcated area, cannot trade citrus, despite the fact that so far in over 15,000 citrus trees tested in infected area are all negative. The pathogenicity tests were negative. But this is just to give you one example. In addition, we have, as I show in my slide, if you remember, with the different risk of infection, there are plants like lavender, etc., with high risk of infection. But there are others for which the risk is very, very limited, very close to zero, for a very few plants that occasionally have been found infected in area under very high pressure of inoculum. But all of you, I think, is aware that in the nursery, the condition, I will say the risk, the epidemiological condition are not the same of the open air that is out of the nursery, because the nurserymen, they apply strong, severe protocol to control the pest. So there is, in general, a problem that needs to open a debate. There is another additional point that I want to raise up. For instance, in Apulia, there is an important district of grapevine nursery, and grapevine is immune to the strain, to the pauca that is in Apulia. In addition, the nursery, they apply since 10 years ago a protocol of thermotherapy with water for their plants. But despite this, several third countries, they do not accept to import grapevine produced in Apulia. So I understand that often the nurserymen, they claim that there is not enough support at institutional level to negotiate with this third country to convince that those plants can be guaranteed as Xylella free.
Thank you, Donato. Okay.
Yeah, this is Roberto San from the MPPO of Spain. My question is to Diego from the Balearic Island. In your presentation, you have mentioned that in the area of Centelles are co-living the 3 subspecies. So I was wondering if you have assessed the possibility of any kind of recombination in the area, and if you are assessing that in this area.
Yeah, not yet, but it's something that we want to work in the future, considering the co-infections and study deeply. But not yet. The work is, we are discussing these days with colleagues for how to do this kind of works with because it's very interesting, of course. Yeah, but not yet. We have not yet this work done.
Thank you, Diego. Okay.
I have a question for Elisabetta Porta. Which is the sensitivity of the method? If the infection is recent, plants are asymptomatic, the instrument can detect the presence of Xylella through the change in volatile compounds in soil?
Thank you for the question. Yeah. Yes, because the sensor gives information regarding the volatile fingerprint. And if there is the pathogen inside the trees, the olive trees or other plants, there will be a modification inside the soil activity due to the metabolism, the root activity, and so on. Due to the presence of this pathogen. So the volatile fingerprint change based off this cause. So yeah.
Hello. My name is Leticia. I'm from the National Research Council from Madrid. My question also goes to Elisabetta. And regarding the question before, my question is whether the volatilome also changed depending on the pathogen. Imagine that it's not Xylella fastidiosa, but another one. And I've also have another question, and from the point of view of a person who doesn't do surveillance of Xylella in the field, I would like to know what is the added value of analyzing the volatilome versus more conventional validated techniques such as conventional PCR or qPCR.
Okay, thank you. Così? Okay, thank you. See, the aim of this project is to create a portable sensor system and put it in the olive orchard in the field and give information regarding the Xylella before visual symptoms appear because of the variation of the soil activity. Then the operators can have information and make specific target treatment and analysis on this topic. Okay.
Okay, thank you. Well, I have a question for our first speaker, Sibren Bosch, because you explained very well all the efforts that EFSA is doing for the surveillance of the territory, but we are talking like Xylella of introduced pathogens, so there are plans also to do this kind of work or similar approaches for the border inspections as well, or?
Thank you, very relevant, a lot of echo. So, the work we are doing and what I explained was very briefly explained around the detection surveys. So the pest is not there, we want to substantiate absence. We do also a whole set of tools for delimiting surveys and also we develop tools for supporting buffer zone surveys. It is exactly the same statistics, it is exactly the same methodology that can be applied and with some tailoring can be very simply applied for supporting border inspection, for supporting a systematic sampling at borders to maybe ensure that's our first obstacle to introduction in Europe. Maybe it would be nice to also experiment and to go further in that. Of course, this work will never be initiated by EFSA directly without having the clear mandate from the European Commission and the member states. If they find that this topic is of particular interest, we will of course do the effort to try to adapt, tailor these tools to another target population, a target of inspectors that are dealing with border inspection. Yeah, it's not a very difficult step to do. It's exactly the same methodological framework that can be further developed.
Thank you very much, Sibren. So it's 4 o'clock, and now we have half an hour for coffee. We have to be back here at 4:30. And please, big applause for all our speakers. Thank you very much.
Okay, hello everyone, and welcome to session 7 of the conference, which is on monitoring and modelling Xylella fastidiosa epidemics. It's volume 2 of that. My name is Nick Cunniff from the University of Cambridge in the United Kingdom. I'm here with Marta Sendoya of IVIA in Spain. And our first speaker is Rachel Russell, also from the United Kingdom, the University of Cambridge, and she will be presenting a talk entitled Modelling the Effectiveness of Xylella fastidiosa Monitoring and Control Across Pathosystems and Spatial Scales.
Okay, good afternoon, everyone. As Nick was saying, I'm part of Nick's group in the University of Cambridge. I'm an epidemic modeller, and for the past 18 months, I've been working as part of the Xylella fastidiosa working group, doing some modelling work. So a little bit of context. So the working group was set up to update the 2019 scientific opinion on Xylella fastidiosa, and this was mostly driven by the desire for the updated regulation to take account of potential differences between Xylella subspecies and hosts. So the previous opinion had focused primarily, particularly on the modelling side, had focused primarily on olive, and we wanted to extend that to represent what was going on across Europe more widely. The modelling work, the reason why people like to use these mechanistic models is that you can explore what-if scenarios. So we can take what's happened in Puglia and say, well, what happens if you'd applied this different mechanism? Or we can make up more generic landscapes and say, okay, well, what happens if you apply the legislated minimum control in these scenarios? Matteo Crosso, who's standing over there, is going to have a more detailed introduction to this at the end of the conference, but we are going to be putting out some details about this for review. So please either come and talk to us in person here at the conference, that would be great, or Matteo will have more details about a more formal mechanism later. Okay, so I'm going to talk about this in 2 sections. So first off, I'm going to talk about the model itself, so the structure of the model and how we parameterize it, and then afterwards I'll talk a little bit about the results. So the model we're using is a stochastic individual-based model. So the picture you can see on the slide at the minute is showing what the computer sees in its brain whenever it's running the model. So this is a snapshot at a particular point in time, and the square is showing a 2 kilometre by 2 kilometre region in the middle of one of our simulations. Each of the dots, in this case, is an olive tree, and the colour of the dot represents the epidemic state of that particular host. The model will run through that with time, and you can see the development of the disease. So as time progresses, so if we start back here, as time progresses, we can see the disease spreading. So you see patches of blue followed by red, and then finally hosts starting to die in the middle of the outbreak. So that's what's going on in the computer's brain whenever we run these models. We don't explicitly model vectors. So we model the transmission of the disease between individual hosts using a transmission kernel. So this describes how the probability of the disease being spread from one host to another host changes with distance. So on the left-hand side plot, we can see the most zoomed-in version of that. And this shows the red line, which is our short-range kernel, which is representing processes like a vector moving naturally from one tree to another within an orchard. In the middle plot, this highlights more the blue line, so this is our long-range kernel. So this is talking about rarer spread events, so things like a vector hitchhiking on a car and moving over a much longer distance, and you can see the distance scale on the left-hand side is like up to about 400 meters, and then the distance kernel in the middle, the long-distance kernel plot is looking up to about 10 kilometers. So we combine those 2 kernels together to show these combined types of spread. We also account for some aspects of seasonality and also the change of host infectiousness with time. So the type of seasonality that we look at, again, because we're not modelling individual vectors, we use a multiplier on the spread rate, which changes depending on the month. So this is an example that's shown for table grape, and we can see that in the winter months we have a 0 multiplier on the spread rate, and then over summer we have a kind of maximal spread rate. So it's a relatively simple model of seasonality, but it lets us see those kind of effects. And then for the time after the host is inoculated, we model a kind of up and down profile. So the concept here is that on the left-hand side of the graph, the y-axis here is looking at the relative infectivity. So how infectious is the host as time goes on? And at the point when the host is inoculated, we model that as zero infectivity. So the idea here is that the vector has just come and landed on the plant, and it's inoculated the plant with the disease. The disease is present in the tree, or the pathogen is present in the tree, but it hasn't spread significantly. So if another vector comes and feeds on the host plant, then it's not likely to transmit the disease. We then have an upwards phase, and this is representing the growth of the bacteria in the plant. And finally, we have a downwards phase, and this is where the growth in the bacteria in the plant is balanced out by the development of symptoms, which makes the tree less attractive for the vector. So we have a flat and then up and then down profile for the host infectiousness after inoculation. So I said we wanted to look at different case studies across Europe, and these are the four that we've chosen as the representative studies. So we are still looking at Pauca ST53 in Italy. But then we're also looking at Alicante, the ST6 on almond, Majorca for grape and almond, but only at ST1, and then we're looking at Ritigliano, again a local issue with table grape, ST1 and table grape. Okay, and for each of these systems, we need to parameterize the model. So the full-fitting flow is relatively complex. It's not something that I would want to explain in a short presentation, but this is a simplified version that shows the information source for each of the parameters that we're using. Sibran earlier was talking about the expert knowledge elicitation process. We've also used that to help us get information about this infectiousness profile, so bacterial spread up, vector attractiveness down, and we also use shared assumptions about the seasonality to feed into the model. For the local spread rate and scale, we ground that parameterization in some outbreak data that was kindly provided by the Puglia Phytosanitary authorities. Claudio's sitting at the back, looking shy. So they were able to find us an isolated outbreak of where we were able to parameterize our model, and then we were able to generalize that across the different pathosystems using an ECHE focus on wavefront speed. For long-range spread, we also use survey data and information about the length of time that the pathogen had been present in the region, and that was using data primarily from Apulia, but also a little bit from Alicante. And then for the host locations, as I showed at the start, in this kind of model, in the computer's brain, it has an idea of where all the hosts are. So in order to run that kind of model, you need a map. So Martina led this work, and this was done by characterizing the different host layouts per land use, and then we were able to extrapolate that out to much wider regions by using land use maps. Okay, so moving on to some results. So if we're going to say how successful control is, we need to say under what scenarios we're going to test. So we tested three main scenarios. We looked at eradication, and we tested that on a relatively small 10 by 10 kilometer landscape. And we initialized these scenarios by taking a single random host around the center of the map. And then for containment, we had two scenarios. So we had, both of these were run on a 50 by 50 kilometer region, and the two scenarios are illustrated in figure A and B below. And the figure A is the small scenario, and each of the little pixels in that diagram are kind of a 2.5 kilometre by 2.5 kilometre square. The grey area is showing regions where there's no infectious material at all, and then in the regions where there's white and red, white means there's absolutely no infectious material at all, and then red means everything, all the hosts in the region are as infectious as they possibly could be. So that's what those diagrams are talking about. So the small scenario is just a few kilometres across, and then we have the well-established scenario, and that's representing something where the epidemic has had a little bit more time to spread. I feel slightly embarrassed talking about this in the current audience, but just as a reminder for people of the current legislation. So for eradication, this is a little bit simplified, but if you find an infected host, then you have an infected zone of a radius of a minimum of 50 metres around that host, where you have to remove all of the relevant host plants. And then out beyond that, for a further 2.5 kilometres, you have the buffer zone where you have increased inspection. So the idea here is that you have the radial removal, which gets rid of hosts that you might not have detected, but that could still spread the disease. And then the buffer zone gives you, you're looking harder for infected hosts where you're more likely to find them. The containment zone is obviously for when it's been determined that eradication is no longer feasible. So you have a large number of infected hosts, in this case at the bottom of the slide, and the disease is progressing upwards up the slide. And this, the edge of that containment zone is determined by the furthest detected infected host. So that's kind of labelled with the little arrow. And again, out in front of that region of infection, you have a buffer zone where you look harder for new infections. But behind it, you have a containment zone. And the legislation here says that you have to do increased inspection, but at any time you find an infected host in this region, you have to do radial inspection around that infected host. So you're not required to remove it, you're required to do even more full inspection, basically, of that region. Okay, and then we effectively tested the minimum legislated required inspection and control, and then did more extreme versions of that. So for eradication and containment, we did no control, so a baseline without any human intervention, and then we also tested the minimum legislated scenario. For eradication, if we jump a row, we tested a scenario of full inspection. So this isn't intended to represent something that would be workable in real life. It's a bounding case where every host is inspected every year. And then we also did an intermediate case where you have a kind of middling case for eradication. For containment, we looked at a stringent scenario, so it doesn't really make sense to do full inspection in this case, but we did high levels of inspection aiming for the very high bound of what might be feasible, and then we also extended that by doing removal and containment. So this is the same as the stringent scenario, but it's kind of a hybrid scenario with the eradication type measures where you do radial removal in the containment zone. Okay, and here's a brief summary of the results. So what we found for eradication is summarised on this plot. So just to break it down, the metric that we've used for success here is the proportion of the epidemics that are eradicated after 10 years. We've looked at that across our 4 different case studies. So we've got Apulia, Alicante, Mallorca, and Rigigliano. And then the different colours are the different management regimes. And the first thing that is probably sensible to point out is what happens with the red bar. So the red bar is the most stringent level of inspection that we tested, and across all the case studies, even with this very stringent inspection, we weren't able to reliably see eradication after 10 years. The other thing that might have caught your eye is the blue bars. So the blue bars have no control, but we're still seeing eradication, which might not make sense. So to clarify that, because we randomly select the starting host and because we test a range of different parameterizations, this is representing the scenario where you're lucky. The seed host has landed on an isolated host, or the first host has happened to go quickly through its epidemic sequence without infecting any further hosts. So it's not unexpected that we would see eradications without control in these types of model. The slightly more meaningful thing is that if we only do the minimum legislated surveillance, then the difference between no control and doing that level of surveillance is very, very minimal. And this is a combination of the problem that we see in full inspection, where the control is really struggling as it is, but also that it's being found a lot later. Finally, moving on to the containment results. So just to break this down again, so the metric that we've used for these results is slightly different. So because we're looking at containment, we're looking to reduce the amount of the landscape that's affected by the disease. So we've used a metric which we call final affected area. So in this metric, we've split up the 50 kilometre by 50 kilometre map into 1 kilometre by 1 kilometre squares, and if any of those squares have any infected or removed hosts in them, we've deemed them as infected squares, infected area. So we've looked again at three different case studies in this case. So Apulia, Alicante and Mallorca for the small outbreak size. So this was the figure A on the earlier slide, and for the large outbreak size, which was the figure B on the earlier slide. The first thing that I think you'll probably notice is that the range of outcomes from these models is very large, but that's okay. What we want to see is what the effect of the management is on these distributions. And what we see is that for the small outbreak sizes, the different mechanisms are having an effect. So if we go from blue to orange to green to red on the Apulia small outbreak size, we can see that it's reducing those distributions. It's bringing each of the distribution points down to a lower level and reducing the impact of the outbreak. And although it's a little bit difficult to see, those patterns are repeated for Alicante and Mallorca. Unfortunately, if the containment measures don't start until later, so if you're starting from this larger outbreak initialization, then the measures are much more challenging. So we can see that between the blue, the orange, the green and the red, there's not a meaningful change between the different measures. Okay, so in conclusion, we've built and parameterized an individual-based model, and we can run it on these regional scale landscapes. We've been able to parameterize it for a range of different European pathosystems for Xylella, and then we've looked at these different scenarios for eradication and for containment. What we've seen is that radial removal with the 50-meter radius isn't able to reliably eradicate outbreaks, but that doesn't mean it's impossible. So it can be successful if you have these high levels of inspection, if you have conditions where it's not favourable to disease spread, or to be frank, if you're lucky. The containment measures, they are able to slow the spread of the disease, particularly for these cases where you start the measures when the epidemics are smaller, if there are high levels of surveillance, and if there is radial removal behind the point of furthest detection. Okay, and a couple of acknowledgements. I'm just the person who can drive the computer, so this was all done in collaboration with the Xylella fastidiosa working group, and I want to particularly thank the EFSA team and the experts who contributed to the ECHE, because we couldn't have parameterized this model without them. Thank you very much.
So the next speaker is Rocío Calderón from EFSA, that will be present enhancing Xylella fastidiosa spread modeling and surveillance optimization through remote sensing.
Okay, thank you, Martina. So first of all, why do we need a epidemiological model for Xylella fastidiosa? Xylella fastidiosa is difficult to manage because it can infect more than 700 host species. It has no cure, and it can spread for long period of time before the symptoms can appear. Thus, early detection and timely action are critical for successful eradication and containment. In this context, epidemiological models can simulate how the pathogen spreads across the landscape, allowing us to forecast the spread in the landscape and evaluate interventions before implementing them in the field. Current control relies on surveillance, host removal within a radius from a detectable infected host, and vector control. This model approach can support decision-making and plan health policy, informing by where and when and how we can implement control strategies. The predictive power of epidemiological models rely heavily on the quality of the data we use to estimate their parameters. Traditionally, epidemiological models rely on ground survey data on the disease distribution. Although ground survey, they are very valuable, they are expensive and slow. So it generates a bias in the spatial distribution of the disease. Moreover, it is very difficult to detect asymptomatic infections because of the long asymptomatic period of Xylella fastidiosa infection in olive. Remote sensing, it is an opportunity to overcome these limitations by providing continuous spatial and high-frequent data on disease distribution. Moreover, and more importantly, they have been demonstrated to capture some of those asymptomatic infections that escape from the human eye. Thus, the questions here, there are two. One is if remote sensing can improve the parameterization of epidemiological model, and if remote sensing can be a more effective surveillance method to guide interventions in the field. So to respond to these questions, we define three objectives. The first one is to parameterize a epidemiological model for Xylella fastidiosa spread using remote sensing data. The second one is to compare it against parameterization from field survey data. And the third one is to compare field survey and remote sensing as surveillance methods, evaluating containment performance across different revisit intervals and roughing ready. First of all, we have to gather the data to parameterize these models, and in this case, we have two different sources of data that they are complementary. One is FAIR survey, and the second is remote sensing campaigns. FAIR survey campaigns were conducted in summer 2016 and 2017 across 15 olive orchards in Puglia, and each tree was visually scored from 0 to 4 according to the proportion of symptomatic canopy. Across both years, more than 7,000 trees were assessed. At the same time of the visual inspection of symptoms in the fair, airborne campaigns were conducted over more than 1,000 hectares within the Xylella infected area in Puglia. We acquired hyperspectral and thermal imagery from a main aircraft, and we have to highlight that the high resolution of this imagery, which enabled us to identify each individual tree. So from this imagery, the objective was to obtain the infection probability, and this approach was in 2018 published in Nature Plants. It is basically based on a very severe tree crown cementation, where we only use the pure vegetation pixels of each crown. We then retrieve plant traits that they are based on pigments, water stress, and a lot of other physiological processes that they are altered by the infection. And these were input into machine classification models, which had the output of the probability of Xylella fastidiosa infection. This is our input that will be used in our epidemiological model. So in this case, the modeling framework for Xylella fastidiosa spread and control is based on a spatially explicit stochastic discrete time spread model, where each individual tree can transit from susceptible to infected status. The transmission of the disease was driven by a heavily tailed dispersal kernel, and new infections per susceptible tree in a step of time follow a Poisson distribution whose rate is proportional to the transmission rate and the proportion and the number of trees that are infected nearby. This process was simulated using a Gillespie lip loop. The calibration of this model was used to estimate three parameters: the transmission rate, the dispersal scale, and the kernel shape. And this model was fit independently for field survey data and remote sensing data to, in another step, compare the posterior distribution. The final step is to test surveillance and control scenarios by combining the detection methods, the revisit interval, and the roofing radius. So moving to the results here, we start with the model calibration results using field survey and remote sensing data as data source. And on the top two figures, I show the log likelihood across the parameter states and also the posterior distribution of the three parameters. By using the two sources of data, we can see that they both converge to the same region of the parameter space, providing a comparable model calibration performance. Also, it's very important to look at the bottom right figure when I show the estimated epidemic versus the observed data. And we can see that, for example, in the case of remote sensing case, at the beginning of the epidemic, the model estimates a higher number of infections. This is because remote sensing can capture those asymptomatic infections that escape from the human eye. Once we have this model parameterized, we predicted the epidemic spread in the sense of control, and here we got two key results. The first one is that the epidemic grows very rapidly, so we can have the 50% of trees infected by 2.4 years, and then 90% of trees infected by 3 years. The other result, and most importantly, is that there is a very huge detection gap. This is that the epidemic remains largely cryptic. So by the time we have only 10% of trees infected undetectable, in the reality background, there are 51% of trees already infected. So in the second slide, I test a containment using two different ways of surveillance. One is remote sensing, and the other one is field survey, across the same range of roofing radii and interval of revisit. Here, the difference of these two types of surveillance methods is the detection probability. While both are they perform as very good in terms of detecting symptomatic trees, remote sensing is the only method that could detect asymptomatic trees that escape from the detection in human eye, with a rate of 0.25. This data was captured from the paper of Zarco Tejada in 2018. The figures on the right, the heat mass, they show the probability of containment and the cost in terms of trees removed as function of rouging radius and revisit interval. Both approaches show that the probability of containment is higher when the visit interval is lower and the rouging radius is higher. However, remote sensing could contain the epidemic with larger revisit interval and lower rouging radius. In addition, field survey, in order to have the same containment as remote sensing, they had to remove a higher proportion of trees in comparison with remote sensing. So the theory thus far is that remote sensing disease must robustly parameterize spatial spread models and are consistent with field-based estimates. In the absence of control, the epidemic spread rapidly with a large cryptic function. The surveillance strategy is the main determinant of control of success, and remote sensing outperforms field surveys, achieving broader containment and lower removal costs under the same effort. This advantage is driven by the earlier detection capacity of remote sensing. And finally, the effective containment requires frequent surveillance in terms of 1 to 3 month revisit, while it is impossible to achieve with aerial remote sensing. Satellites offer a most effective manner to do this. So the next step in our group will be to validate the detection of Xylella fastidiosa infections with satellites and to validate the use of this data for parameterized epidemiological models. Thank you.
So thank you, Rocio, for a talk which is right on the state-of-the-art, linking models with new sources of data. Our next speaker is Francois de Alaye from INRAE in France, and he's going to speak on adapting surveillance design to outbreak characteristics, a dynamic framework.
So I thank EFSA for the invitation, and I will present a work that is a joint work with people from BioSP and IVA in Spain, and it's about how to design the parameters of survey designs. So let me give some context. So Cybrain talked about the different tools that you can use to do survey designs on pests and things like that. So there are the EFSA guidelines that helps you designing surveys. And so to do the survey design, it's the process of defining where, when, and how you should make observations. And so more precisely, the surveillance is the observation of little n inspection units among a population of big N individuals. And so these inspection are used to infer properties of the spatial distribution and support decision-making processes. So the question is how to design the sample size n. So classically, there is the formula that Cybrain introduced earlier with different parameters, like the method sensitivity. And we see that there is another parameter, the design prevalence, DP, and it's the prevalence that we should detect. That is the maximum level of infection that we would like to detect in the space. And we see that the sample size doesn't depend on the spatial structure of the phenomenon. And so we would like to make this dependence explicit. So here we are two examples of outbreak, and the first one is more aggregated, and the second one is less aggregated. And we see that in the aggregated case, a sample is more informative than in the other case. And so in this case, we need more samples in the less aggregated case. And so what we do is we will adapt the design prevalence to the spatial structure of the outbreak. And so to do that, we introduce the propagation function f. And Martina already told about the range of this function. That is the distance beyond which there is no spatial correlation. And we have here two examples. On the first row, we have an example with range 10, and below an example with range 50, with three different levels of infection. And we see that the perimeter of the outbreak depends both on the level of infection and also on the range. That is, the more there is aggregation, the less is the outbreak perimeter. And so what we propose to do is to adapt the level of infection we want to detect. We want to adapt this design prevalence to the aggregation level R. And more precisely, we do such that for two different levels of aggregation, we select the design prevalence so that the perimeter of the outbreak are the same for both aggregation levels. And so the outbreak perimeter will be the invariant. And so here it's on the first graph, it's the adaptation of the design prevalence. So 100 is the most aggregated range. And we see that we decrease the design prevalence. And below we have the corresponding sample size. And we see that it increases as there is less aggregation. And on the right, there is an example with 3 different propagation functions of 50, 25, and 10 ranges. And the first is the same level of infection. And the second row is the adapted design prevalence. And we see that the perimeter is the same across all aggregated, across all propagation functions. And so what you should do in practice to specify the design prevalence and the range in the most aggregated case. And then for the true range of the true outbreak, you tune the design prevalence so that the outbreak perimeter is equal to the outbreak perimeter of the aggregated case. And so we compared this to the when you selected the same design prevalence with 3 criterions. That is the accuracy of your total prevalence prediction, the A. The B is how you cover the space of infected individuals. And the C is the accuracy of a prediction of unsampled individuals. And we see that the standard method is less efficient where there is less aggregation. And so finally, to summarize, we adapt the design prevalence. So the maximum level of infection we tolerate to the level of aggregation of the outbreak. And so we saw on the results that there is the same level of efficiencies over the spatial structures. And when you don't know the true parameter of this propagation function, either you can learn it from literature or you can estimate it in sort of a 2-phase approach or something like that. And so here's the link to the paper if you are more detailed. And I look to your remarks later. Thank you.
Thank you, Francois. So the next speaker is Anna Simonetto from the University of Brescia, Italy. And the title of the presentation, 12 Years of Models of Xylella fastidiosa Epidemics in Europe, Lessons Learned.
Good afternoon. When we start our journey in the history of the modeling of Xylella fastidiosa, we found more or less 6 papers focused on modeling. And you can see the history of the number of papers during these 13 years. We observed a peak in the last 5 years when most of the evidence of the results starting to be available for the public and so also for the modeling part. But interestingly, we observe also a slowdown of the model in the last 5 years that probably could tell us something. Maybe in the second part of our talk, we can discover what can be learned by this. We try to define the topics afforded by this model. The first one that have been afforded by the modeling part was risk assessment surveillance. So the problem was how and where Xylella could establish. The second one that has been afforded is spatial epidemiology and spread, and you can see different kind of mathematical and statistical models. The third one is vector and transmission ecology. We shift a little bit before there were regional and area-wide management scale, and then we went in the crop field and with the implant. Then we have host pathogen and mechanism. And then the last one that appeared are eco-epidemiological models. And the last is models that change a little bit. We are not focused on the processes, but the objective. So our models that have been developed to support policymakers and decisions. What we learned by this model tried to get an answer to these three main questions: risk assessment and surveillance. So they provide climatic suitability maps, establishment risk assessment, and climatic change risk projection. The second one was spatial epidemiology and spread. So model tried to answer to problems like how rapidly the epidemic can expand, what are the processes that affected the transmission, and what is the effect of the containment measures that have been tested. The last question was the interaction and the processes between host, pathogen, and vector. But there is also some knowledge gaps, so some question that is not being afforded by the model right now. So what are the influence of the environmental factor on the epidemics? So focusing, for example, on the temperature and water stress on bacterial growth. How we can transfer the model between different pathosystems, because most of the model have been focused on olive agroecosystems. And we know a little bit less on almond, coffee, and so on. And what is the role of the plant community biodiversity? So, for example, the sink, the reservoir, and so on. Surprise. Okay. So we need to discover why we observe this slowdown in the modeling, because we need probably to change the approach to our modeling strategies and try to afford the problem of the beings in a more comprehensive way. So we need to think to who will use our models. So the strategy that we suggest is the idea of I can say, a comprehensive modeling approach. We work in the Big Seal project. One of the winning key approach of the project was to integrate different components, for example, molecular biology, vector ecology, plant physiology, environmental management, and the work package on the modeling part support this kind of integration, providing two kinds of tools. Digital laboratory, so a modeling that can be used to fully exploit all the very huge amount of results produced by the project. And the other kind of the modeling approach are knowledge-based decision support. So how to get these results useful for the final user. One of the idea that I proposed is the digital lab, so a model where we can collect and create a relationship between all the different components that also the previous speaker already presented. So where we can use as inputs field monitoring, remote sensing, weather, laboratory data, and so on. And we can elaborate them and to produce something that is really usable by the final user, both risk assessor or farmer and people working in the field. How to do this? We not only need a very, we can say, comprehensive model, like the one that we call core model. The core model is the part that working on the overall dynamics of the epidemiology, but we need also something that we can call as a plugin, a sort of interface, and we call them satellite models. What are satellite models? They split the epidemiology in different components and became the interface with lab field, lab experiment, field data, and so on. They focus just on one part of the issue, and they can directly be fed from the result obtained by different researchers. They make some elaboration. They are usually mechanistic, so they are based on real dynamics. So we try to, I could say, to make the less assumption and the less elaboration, and to keep the process quite well. And then this kind of a satellite model transform the input from field and lab data in something that is usable for the core model. So we have create a sort of tools, suite of tools. The left part that you see is the plugin with the real data, and the right part is the modeling part to create connection and to try to get together all this kind of information. And it could be done at field level, in the central part, or at area-wide management. On what we are working now, we already finished within the Big Seal project, the olive ecosystem that is being published in a paper on ecological modeling, and how you can use this kind of approach. For example, for the scenario analysis, you can test, as you can see, 2 different IPM schemes. So not just one single element. So you can try to change the vector control strategies and something on agronomic practices. Or you can change other components of the scheme and try to evaluate what is the impact of 2 different IPM schemes on the same location on the epidemics. We are working also on the grapevine. You can see the poster 120 and 111, where the model has been presented, and we submitted a paper. And the next step will be the almond agroecosystems. So what we learned in this 13 years journey, modeling approach and modeling will not have not exploited their potentiality right now. We need to change a little bit our mind and try to approach differently the modeling, try to think at them to virtual lab, so they can be an opportunity for a dialogue between researcher, risk manager, and also final user. The idea is that in one framework, just one big not for the complexity, but for the idea of the comprehensive framework, you can manage with different scale, you can manage with different objectives. So thanks for the attention, thanks for our colleague. The mind behind everything is Professor Giglioli, but we have a huge work team on that. Thanks.
So thank you very much, Anna, for an interesting talk and for dealing with the slight technical problems so gracefully. The next speaker is Vicente Dalmau from the regional government of Valencia in Spain, and he's going to give a presentation entitled Evolution and Consequences of 9 Years of Application of Phytosanitary Measures to Control Xylella Fastidiosa Subspecies Multiplex Outbreak in Alicante.
Thank you very much, Nick. Thank you all, good afternoon, and I would like to thank the organizers for having selected this oral presentation to explain which is the evolution of the situation of Xylella fastidiosa subspecies multiplex in Alicante, which is the area where I am responsible of the plant health service. Starting, yes, the map that you have seen in several times during these conferences, I locate the area where we are going to speak in the following minutes. This is the evolution of the demarcated area. You can see in red line the limit of the demarcated area, and in blue you can see which is the limits of the demarcated area of the previous year. So you can see how the demarcated area has been increasing year after year, and the evidence are that we have a lot of positives, but the demarcated area is since 2020 year is quite established, apart from the increase of the demarcated area shown in 2023 that was also explained just a few minutes before by Martina. This is the results of the monitoring density shown as well by Martina. This slide has been already shown, but what I want to highlight is that we are focusing our sampling not only in the demarcated area, but also in the surrounding 10 kilometers away from the demarcated area, in the close vicinity of the demarcated area, to be sure that the demarcated area is well delimited. This is the increase of in percentage, the demarcated area. So we can see that since 2020 to last year, the annual increase of the demarcated area average is below 5%. And this is a result of the sampling and analysis done in the demarcated area, but you can see also in the picture in the map on your right that we are taking samples all in the whole territory where the hosts are established in our region. This is the figures of which is the state of our demarcated area. We have about 1,700 square kilometers of demarcated area, 4,400 hectares of infected zone. We have already eradicated about 4,400 hectares, and we have about 190,000 analyses performed in all these years. Up to now, we have detected 30 plant species different affected by only Xylella fastidiosa subspecies multiplex XT6. So it's quite homogeneous, the outbreak that we have in Alicante. About the prevalence, we can see that the infection is mainly for almonds and also for polygala, and that some other species, like the forest species, the prevalence is much lower than the one found in agricultural environments like the almond production areas. Also, a positive point is that the prevalence in insects is quite low in our area, on average 3% after more than 20,000 insects analyzed in all these years. These are the aspect of some eradicated area done in these past years since the first outbreak. Here you have an orthophoto. In the left, how was the area in 2017 and in 2025 after successive positives found in this area. This is Tarbena, which is a highly infected area, and how do we remove mostly the almonds in these zones. This is also shown by Martina, that this shows that when we are detecting positives, which are the orange squares, then after we are applying the eradication measures that are applied in the whole infected area of the Alicante. Here I want to explain some results of our essays done in testing how is the prevalence of forest species, that not only almonds has been present and infected, but some forest species. So we selected 4 places for the eradication areas in Alicante, in different areas, in the coastal area, also in the midland and the inland area. And we conducted surveys and analysis in the infected area and also in the surrounding area. And what we see is that the prevalence of the insect infected and the plant material has been reducing in this period. So if we can, that's it. So you can see that in the infected area. You can see that in the infected area, the prevalence was higher. The infected area is the 50 meters around the positive plant, depending on the location of the eradication area. But we can see that in the following columns, which are in 1 year later, and in the vicinity or further than the infected zone, no insects were detected, and the prevalence in plants was much lower than the one found in the eradication areas. This is an effect on the impact, because there was a question just before on how is this disease impacting in almonds in Alicante or in Balearic Island. Since 2020, the statistics show that there is a 82% reduction of surface of almond trees in Balearic Island. This reduction in Alicante province is about 43%, and if we consider only the 3 counties of the province of Alicante where the Xylella has producing the highest impact, this reduction has been of 57%. So after removing 25,000 almond trees in the infected zone, we can see that the reduction of the area of almonds has been also produced by other reasons. But we are not the only responsible for this. Also, there are EU funding for plant health measures. In the EU Commission, you can say that the plant health is essential to ensure sustainable and competitiveness of agriculture. The impact of priority pests, it's thousands of millions of euros. Like you can see here, for Xylella, you have between 5,000 million euros to 7,000 million euros, but also you have the impact of job losses, export losses, impact on the environment, etc. But the European Union, to support the surveys and control measure for 400 quarantine pests in 27 member states, we have only 8 million to share, which is, in my personal opinion, is not enough to face all this potential impact that can produce the 400 quarantine pests. This is the one example I want to follow. The one example of which is the percentage of investment of funds received by the European Union and the remaining amounts that are paid by the regional Ministry of Agriculture and the Central Ministry of Agriculture in 50%. And this is the last funding received, that where about 14% of the cost of all these measures were paid by the funds of the European Union. And why I do explain this? Because as we don't have enough resources to apply the regulation in homogeneous, because our resources are limited, what we do is that we prioritize our actions from the outer part of the demarcated area, and then we start eradication measures from the outside to the inside, because our main goal is to avoid the spread of the disease to other parts of pest-free areas. And finally, I want to highlight the economical impact that has the nursery sector in my region. We have more than 1,600 professional operators that are selling plants. The region of Valencia is the main region for the sale of living plants. And the EU regulation has strong restrictions about how the plants can move around Europe in the European territory. And the problem is that Xylella fastidiosa multiplex has the highest number of host plants, and the impact for the nurseries close to Valencia is the highest in terms of the number of plant species that has been restricted to the movement outside the demarcated area. And also, one thing highlighted by Donato is that sometimes third countries doesn't understand that there are alternatives like thermotherapy, and they oblige to declare pest-free areas different from the one that we have established following the rules of the guidelines of EFSA during all these years. And thank you very much. I would like to thank to all this financing organizations like the European Commission, the Ministry of Agriculture, and our colleagues from Grupo Traxa that are executing the measures, and IBIA and Generalitat Valenciana and CSIC. Thank you very much.
So the next speaker is Gianvito Ragone, but he's not here. Ah, okay, sorry. So Gianvito Ragone is from the Regional Plant Health Service from Puglia region, and he's going to present integrate surveillance and phytosanitary measures against Xylella fastidiosa subspecies fastidiosa ST1 experience of the Apulia region.
Good afternoon, everyone. Thank you for this opportunity. So today I want to show you our experience in control of Xylella fastidiosa ST1. So we should spend some words about the economic sector of table grape in Apulia region. With more than 70% of Italian production, it generates more than 400 million euro per year. So with the detection of Xylella fastidiosa ST1, there is the rise immediate concern about the economic stability of this sector. So all the things and all the activity that we have done are based on regulation, European regulation, but also national and regional risk and action plan, and also the guideline of EFSA. So the goal of our action plan is to assess with a monitoring survey the presence of vectors in Apulia region for the detection of Xylella fastidiosa ST53. But during this monitoring, during 23, we find 30 positive insect vector in a pest-free area near Bari. The name of the small town is Trigiano. After that, we started the limiting survey with the finding of 20 positive almond trees, and with that we characterized the ST1. So during 24, we analyzed for each hectare in the buffer zone of 2.5 kilometer, 7 samples per hectare. It depends on the host plants. So at the end of 24, we analyzed more than 44,000 samples with more than 400 positive plants, of which 339 positive for ST1. So during 25, we have done a monitoring survey. So we find only 20 positive plants for ST1, and during 26, only 13 positive plants. So in the figures, you can see also the overlap of different demarcated area for ST1, ST26, and ST53. So we have a different eradication measure with different layers. So step 1, we remove all positive plants and all the host plants in infect area. After that, we have done a supplementary measure to remove all the inoculum, all the reservoir that are like abandoned or marginal almonds. So in the high-risk zone of 400-meter buffer radius. So farmers can accept also the eradication of table grape vineyard in the 400-meter buffer radius to also reduce the inoculum. This is an example of the eradication measure in table grape vineyard. And at the right, we can see in yellow colors the table grape that are eradicated, but also positive plants that are eradicated in infected zone. These are in the marketed area, an example of abandoned almonds. So all these activity are destructive measures. So for this reason, we assess the effect of this measure, and we help farmers with the compensation scheme that is calculated with the difference between gross production value and production cost. With we financed €58,000 per hectare for table grape vineyard. So we also financed the approved of table grape vineyard. So another thing that we are doing is the control of insect vector. During the spring, we monitor the juvenile stage, and during also May till November, we have done the monitoring of adults with sticky trap and sweep netting. So this is used to assess the best period for tillage operation and insecticide operation that are a mandatory practice to reduce the population of vectors. So to guarantee the health of the propagational material, we analyzed more than 11,000 samples. So we find 0 positive plants in our certificate system. So the key lesson of our activity are that with more 50,000 tests, we can say that there is a stability of the outbreak. And with the vector, the monitoring of vector, we can early obtain a result of positive plants or, of early outbreak. So mandatory practice like the removal of table grape and mandatory practice like tillage and insecticide operation can reduce the inoculum. The certificate system can guarantee the health of the propagational material. So with all of this work, and we should do a lot of work in the future, we can guarantee the health of European viticulture. So I want to thank you, all my colleagues that are involved in the control of Xylella fastidiosa ST1 and all of Xylella fastidiosa. So thank you.
So thank you, Gian Vito, for a very clear presentation and a positive story. The last talk in this session is Vale Egea of CSIC in Spain, and she will be presenting on a hyperspectral phenotyping platform for early detection of Xylella fastidiosa infection in woody hosts.
Good afternoon. First of all, I want to thank the committee for giving me the opportunity to present this work. As we know, the detection of Xylella fastidiosa is challenging because it's long incubation period. In this aspect, the early detection is essential to before visible symptoms appear. In this context, the high-resolution hyperspectral imaging is a powerful tool to determine the response of the plant. The objective of this work are to develop a phenotyping platform, plant spectral information, to analyze the spectral signature of woody hosts, and we will try to identify vegetation traces before the development of the symptoms. This is the methodology that we will develop, and the first step, this is the creation of the platform. In this case, the phenotyping platform has an hyperspectral sensor which is able to acquire different imagery across, in this case, different woody hosts in different times post-inoculation. The next step, we will try to calibration different images from this calibration to extract the different vegetation spectral signature. Later, we estimated the 101 vegetation indices, and in the final step, we selection this representative group of these indices, and we will train in validation with machine learning models. In the last step, we create an ensemble disease detection model with these machine learning models, and we can see in this slide the high capacity to the predicted infected and non-infected plants. In this slide, we can see the different heat map, that this map represented the contribution of the selected vegetation indices in each goodie host. As we can see, there are, in this case, 5 common vegetation indices, to diseases very important in these models. The conclusion of this work, hyperspectral phenotypic platform has been successfully developed for imaging woody pot plants. A robust image processing workflow was established to extract vegetation pixels, the discrimination between Xylella fastidiosa infected and non-infected using an ensemble machine learning models, and species-specific spectral response of service the distinct vegetation indices to disease discrimination in different woody crops. And for the last, the vegetation indices are predictors of the infection related with pigments and water. And without brilliant people, this work, it wouldn't be possible to do it. And thank you for your attention.
So thank you very much, Vale, for an excellent talk, keeping exactly to time, so very well done. I'd firstly like to thank all of the speakers we just heard from. So we've heard a wide range of perspectives, ranging from modelling to phenotyping to experiences of control in the real world. I'm sure it will have sparked some questions in your mind. So I turn it over to you to ask questions of our speakers. Please don't be shy. There's one there.
I'm Massimiliano Morelli from CNR Bari. I have a question basically to Anna Simonetto, but maybe it can be also answered by the others. Given this pathosystem, Xylella pathosystem or better pathosystems, how wide is the lifespan of the model you are releasing? I mean, these are, I guess, predictive models which are projected on a future temporal scale. But in the meanwhile, you said that, at least in our expectation, these are continuously fed with new knowledge, with new languages, new data sets. So, in your expectation, nowadays, when you release a model, how big will be the order of magnitude in a temporal scale of your model in the hands of risk assessors, consultants agency? So when you will change your model, or this will be still valid?
Very interesting question. In fact, probably we need to separate models by results. The architecture that I try to design is being developed to overcome the problem of expiration. The core model will be alive. Satellite model can change according to the available knowledge. In fact, we are in new outbreaks period, so we are increasing our knowledge on different processes. Many of them have been already studied, and there are others that are not effort still now. But the core model will be alive. probably the results that we obtain now will not be perfectly representing the situation in 2 years, but the model structure has been developed to overcome the problem of expiration.
Okay, thank you.
Hello? Yes? You don't see me, I'm behind the pillar. Yeah? Okay, I'm Sibren from EFSA, and I had a question for François Dalaye. Very interesting to see how you are trying to estimate the design prevalence. My first question is about, have you tried to implement it in a survey design, or have you tried to calculate it and implemented it in a real survey on Xylella fastidiosa? And the second question is it only focusing on monitoring surveys, or you're as well trying to estimate these levels of aggregation or degrees of aggregation for performing further detection surveys in areas where the pest is not present?
So thank you for the question. I didn't quite hear the second question, but so for the first question, we tried on simulations how the behavior of the sample size with the adaptive design prevalence and without with it, and we computed the different efficiencies. But we didn't try on real data yet. But we tried on multiple host configurations, and that was supposed to represent the real world. So yeah. And, but
the second question, thank you for that answer. The second question is about the type of surveys that you think you can design using this system for estimating design prevalence. Is it for monitoring surveys in areas of outbreaks, or is it as well something you think you can apply for detection surveys in completely different areas where maybe the situation is completely different, the ecosystem is completely different?
Yes, so in the simulations, we tried different survey designs, like two-phase and cluster and something like that, to see if there were some different behaviors according to the survey design. And but the origin was to for the early detection to see if the pest was present. But there are other work to design survey designs for the early detection, but not the sample size, but how to do the survey for early detection.
Thank you. We will continue discussing. Yeah. Thank you.
Thank you. I think we have a question at the front. And if you have questions, please don't be shy. If you stick your hand up, we'll come to you.
So thank you for the excellent presentations. I have a question, well, or a comment or a reflection for Rocío, Gianvito, and Vicente. I think Rocío presented very impressive results that with remote sensing, I mean, the surveillance efficiency can be improved a lot. And I mean, the technology is there. There are plenty of papers already published. There are private companies running these kind of systems for different applications in agriculture. So my question, or I would like you to reflect on that, why this is not used or is not widely used by the plant protection services on the daily, let's say, survey activities. There are some constraints, something, because apparently this may help a lot on reduce the number of inspectors, the number of, cars driving to the place, etc., etc. So if you can comment on that.
Yes, thank you for the question. For table grape vineyard, it's difficult to use remote sensing. Also for the cover, the plastic film, that not be able to, for the drone, it's not be able to understand the difference potential, the stress potential, and other things. So it's difficult.
this is something that we will take into account because always I express how scarce of resources we are and how efficient we should be, and whatever we can reduce the cost of surveillance by using these methodologies. The constraint is that, for instance, that this, for the diagnosis, for instance, that this technique should be certified, let's say, by EFSA or by EPPO diagnosis protocols, but for the surveillance, it's a support technique that we can apply. Yes, we will think about it.
Maybe I can add or repeat that EFSA is now starting to work on how to integrate directly the whole remote sensing approaches within the toolkit to support better. No? It will hopefully help you to reduce resources or better use resources.
And now it's my turn to answer to Antonio Vicente. I think that remote sensing has a lot of potential to be used as a surveillance method. We have demonstrated for more than 10 years that it is applicable. It's true that we now are more focused on aerial imagery, that it is more challenging to use over a high-frequent acquisition of imagery, but I think the future is now satellite imagery. Now, for example, the European Space Agency here in Italy with Prisma, with the MMAP, they are deploying hyperspectral satellite imagery. That is now a huge step change. And now we have to take the opportunity to use this imagery in order to try to see if we can detect the disease. I think that we can detect the incidence, but the challenge will be in detect early the disease, that it is only possible with aerial imagery because of the higher resolution. But we have to investigate it.
Okay, so there was another question there? Yeah. Hello, I have two very small questions. Thank you very much, first of all, for all excellent work that you present today. I am very, very interested in all the modeling. I'm sure that you use the real-time data to train the model, so it can give us a prediction for what's coming. However, could you mind to tell me, or maybe in your data, what is the accuracy of the prediction? First, second question is, in terms of surveillance design, for the moment we do have the available tools to design it like repest. If we use your model, is any advantage that your model bring to us compared with the available model like repest?
So thank you. That was a very general question. It was slightly unclear who that was addressed to. I don't know if anyone wishes to take that question.
Okay, so the accuracy was measured with 3 criteria. So the first one was when you measured the prevalence and you computed the confidence intervals and the size of the confidence intervals. And it was less when you adapted the design prevalence. And after, there was a coverage criterion that would tell you how much you cover infected that you didn't sample. And this was a better coverage for the adapted design prevalence. And the last one was the similar, was when you predicted the infection status of unsampled plants. And so you measured the balanced accuracy of this prediction. And so it was better also for the adapted design prevalence. But
the second one is how better the model in design of the surveillance programs in compare with available programs like RIPET. Because we use RIPET to design the area for survey number of sample. If we use your model, for the moment you are still in simulation, but if we use it in simulation environment, what is your advantage? What you bring better than rabbit?
What is my...
if your model can give us any better result for prediction, for the predictions and design their survey program in terms of area of survey, number of sample compared with rabbits. If I have to choose between your or rabbits, how you convince me to pick yours and not rabbits? Simple as that.
Okay. So the rabbits advises the sample size to take, but here we use the same formula than rabbits, but just we modify the design parent parameter. But on all other cases, it's the same. That's the rabbits tool.
So you say that your model is an improvement of rabbits?
Yeah, it's
beyond rabbits, but improve it?
Yeah, just another parameter that you can tune to have a better usage of the surveys.
Yes,
so it's
thank you. Yes.
Yeah.
I hope that someday when you implement it with the real data, and I can talk to you again about that.
Okay.
Thank you.
This is a question for Rocío and Valle about remote sensing. How accurate is the system for distinguishing between abiotic stress, for example, water stress, and Xylella symptoms?
In my case, the spectral signature is with pixel without symptoms. In this case, we will separate this model, this ensemble model, is be able to discriminate infected and non-infected with pixels in same plants, okay?
I'm going to talk about at the canopy scale with remote sensing. So the data that we use here was data, it was a modeling framework that we developed in 2018. But then always the challenge was that, for example, Xylella fastidiosa is a vascular pathogen, and it shows similar symptomatology to other pathogens, for example, verticillium wilt, and another challenge is water stress. So at the beginning, it was very difficult to detect, to distinguish between the three dysbiotic and abiotic cases, but then we came across to another modeling framework when the objective was to detect specific indicators to each pathosystem and water stress. And having this it is like a hierarchical modeling, and using only this specific indicator, we could distinguish between water stress, verticillium wilt, and Xylella fastidiosa in olive, even in almond. So it was also published in Nature Communication in 2021, and now is one of the most proud results that we had in our group.
Okay, so I will keep it short, and maybe you said it, and it's the day, the heat. When we talk about remote sensing, it could be satellite images, plane, or drones. Drones have the advantage that they could monitor below covered crops. Has any of you done some research on drones? Because somehow I missed it, maybe?
For example, I started my PhD using drones in verticillium wilt years ago, but then with the regulatory issues in Spain, we couldn't use it when you want to acquire imagery over large areas. So we finally we decide to use airplanes, because finally the objective that we have in our study is to develop more operational ways of surveillance. But now we are not using drones because of the regulatory issues instead.
While you're thinking, I have a question for Anna, if it's okay. So thank you for your talk, Anna, which I thought was really interesting. And you talked about a wide range of factors that you want to include in models, and I think even have done for Olive. So which one of those was the most important? What's the key missing one?
Sorry, I didn't get the point. Sorry, can you repeat?
So if I understood your talk correctly, you've been working on a model targeting Olive, and you talked about having a core model to which you insert various sub-models with more detail on certain processes. So which one of those was the most influential on your result? What process is most important?
In fact, we didn't perform right now this kind of analysis because we are interested in the sensitivity analysis of parameter. We are more interested in the relation piece of both of them because we strongly believe that the solution is connecting each other and not just focuses on one aspect. We also performed in the past some specific model for different components, but right now I don't have the results according to the most relevant. Sorry.
I understand, that's very clear. Is it the case that that pause has given anyone in the audience some time to formulate a question? It is the end of a long day. We could wrap up slightly early if there are no more questions. There's a couple of announcements that I need to make if we do that. So the first, obviously, is I'd like to thank all of the speakers for their excellent talks. So thanks again to all the speakers.